So once again, a very warm good morning to one and all present here. I welcome you to the 66th Annual International Conference of Association of Microbiologists of India, supported by DBT, ANRF, DRDO, CSIR, and UCOST. Yesterday, after the inaugural ceremony of this 4-day conference was beautifully been done, and we are starting with the day 2 of this enriching conference. Before I invite the chair of this first session, I would like to introduce you the chair of this session. Professor R.C. Kuhad, a Pro-Chancellor of the HSC University, Gurugram, a founding chairman of AMSE. Professor R.C. Kuhad is a renowned microbiologist, academician leader, and a visionary in the higher education. He currently serves as Pro-Chancellor of the SGT University, Gurugram, and he is a founding chairman of the Academy of Microbiological Sciences with the illustrated career in spanning over the decades. He had made an outstanding contribution to the research, innovation, and academic governance of India and abroad. Professor Kuhad has previously served as a Vice Chancellor at the Central University of Haryana and as a professor in the Department of Microbiology. Now I request our chair to please join us on stage. Give a big applause for the chair of today's first session, Professor R.C. Kuhad. I welcome you once again, sir, to the Dev Bhoomi Uttarakhand University. Can we have a huge applause for the chair for this session? Now, it is the time to introduce the co-chair of this session, Professor Prince Sharma. Professor Prince Sharma is a distinguished professor in the Department of Microbiology at the Punjab University, Chandigarh, and a fellow of Academician of Microbiological Sciences, FAMSC. With extensive teaching and research experience, he had made a significant contribution to the field of molecular microbiology, enzyme engineering, and industrial biotechnology. His pioneering research focused on the development of thermostable and eco-friendly enzyme, addressing challenges in the industry process, such as those in the paper, detergent, and the food industry. With this, I invite the co-chair of this session, the Professor Prince Sharma, the President of AMI. Can we have a huge applause for our co-chair of this first session? I welcome you once again, sir, to the Dev Bhoomi Uttarakhand University. Now I take an honor to invite the co-chair and the chair to please introduce the panelary dialogues being done by our first session.
Good morning, everybody. Let's start with a good beginning. As you know that we are beginning with 4 days conference. And yesterday we had inaugural session that had been very successful. Congratulations to the organizers. Congratulations to the delegates. Dear friends, theme of the conference is immediately relevant, and I'm sure it will be very beneficial to each one of you. And the conference, the theme has been decided after due deliberations, keeping in view that most of the places, most of the conferences, we have been focusing on research, innovation, skill, and whatnot. But we are leaving education every time. And this time, Center Council decided that Association of Microbiologists of India and Academy of Microbiological Sciences should also focus simultaneously, parallelly, how the education in microbiological sciences should be. And that's why the theme has been focused on next generation microbiology research, innovation for economy, environment, and energy. Dear delegates, you are lucky enough, specifically the young or budding microbiologists, that you will come across the distinguished, speakers throughout these 3 days, 13th, 14th, and 15th. Make use of that. Let me introduce to you the distinguished scientist, our good friends, Professor Rooplal, who is born in Hamirpur, Himachal Pradesh. Professor Rooplal is a distinguished Indian microbiologist and educator with over 4 decades of experience. I would request Professor Rooplal to come on stage so that our young students can see and take your photograph और अपने अपने मन में और दिल में उतार लीजिए इनको कि ये प्रोफेसर रूपलाल हैं. You'll enjoy his say the whole address. फोटो आपका ही है. He is a renowned educator with over 4 decades of experience in teaching, research, and academic leadership. Ladies and gentlemen, he began his teaching career in the year 1979 as a lecturer in zoology at Sri Venkateswara College, University of Delhi, and later served as professor of molecular biology in the Department of Zoology, University of Delhi, until 2018. I'm also happy to let you know that currently he is an INSA senior scientist at Acharya Narendra Dev College, University of Delhi, where he continues to mentor young researchers and promote microbial science globally. And he is ambassador of Academy of Microbiological Sciences as well to popularize microbiological sciences. He also serves as senior ambassador from India for both the International Society for Microbial Ecology and the Federation of European Microbiological Societies, and as advisor to Emily SAC, a global initiative advancing microbial literacy in India and abroad. Dear friends, Professor Lal's pioneering research spans microbial diversity, genomics, and metagenomics, focusing particularly on environmental and industrial microbiology. His groundbreaking work has led the discovery of over 50 novel bacterial species and the sequencing of more than 30 genomes. He has, yes, ये तो सुनिए आप, कल से मैंने कहा था आपको क्लैप याद रखना है. क्या याद रखना है? CLAP, clap. And while you clap, then understand the meaning of clap. Okay? तो बजाइए ना ताली तो. His team has successfully developed cloning vector systems and rifamycin analogs effective against multi-drug resistance tuberculosis, contributing significantly to global health research. And an inspiring mentor, Professor Rooplal has supervised 75 PhD theses. That's remarkable. That's remarkable. He is not himself 75 years, but he has graduated, say, 5 doctorates. And he has mentored over 150 students and authored more than 250 research papers with 9,479 ISI citations and S-index of 48, reflecting his sustained scientific impact. Give him a big clap. देखिए, I only can identify that those who are happy with the achievement will certainly clap. Those who are unhappy won't clap. I want to see that how many of you are happy. Another benefit is when you will clap, you will get gray hair like me later, because earlier I never used to clap. That's why I got. Ladies and gentlemen, his exemplary contributions have earned him numerous national and international honors, including the Alexander Humboldt Fellowship, Germany, Department of Biotechnology Overseas Fellowship, Indo-US, American Society of Microbiology Professorship, Andover Executive Fellowship from Australia, and the Moselio Chester Distinguished Service Award from the American Society for Microbiology. He is a visionary in microbiological sciences. Professor Rooplal continues to inspire the next generation of scientists through his dedication to education, innovation, and global collaboration in the biological sciences. And today also, he will be speaking about next generation microbiology. I invite on behalf of you, on my own behalf, and on my co-chair behalf, Professor Rooplal.
Thank you, Professor Quad, and for a very nice introduction. देखो इतनी तालियां बजने के बाद जो बोलने वाला है वो नर्वस हो जाता है. But it's always pleasure to speak before Professor Quad and Prince, with whom I had been associated for the past almost more than 30 years. In fact, Professor Quad, actually, I'm not microbiologist. And Professor Quad actually pushed me to microbiology. He came in the year 1995 to my lab with the slip कि भाई आप अब AMI के member बन जाओ, life member, और मुझे पैसे दे दो. And I became AMI life member of such, I didn't know that time that such a great organization, which is known not only in India, but almost in the entire world actually, so many members and so many activities AMI is performing. And then, in the year 2006, again he came to my lab and said कि रूपलाल आपको IGM का एडिटर इन चीफ बनना है. मुझे पता भी नहीं था एडिटर इन चीफ होता क्या है, और इन्होंने बना दिया. Frankly speaking, when I did my graduation, I did not know anything about microorganisms. The only thing I knew was Plasmodium falciparum and Mycobacterium tuberculosis. Two organisms which I mugged up actually, क्योंकि एग्जाम में ये क्वेश्चन आते थे कि भाई कौन सा ऑर्गेनिज्म मलेरिया कॉज करता है, and which is the one that causes tuberculosis. Even in my MSc, I only studied Paramecium. That was the only organism I thought, microorganism. But when I did my PhD with the impact of pesticides on ciliate protozoans, when I was writing my thesis, I realized I had done a mistake by using my ciliate protozoans. I would have used bacteria actually to explore the degradation and metabolism of pollutants. So that's how I subsequently entered into microbiology, and then in the coming years, I only realized actually sometime when we started doing metagenomics that microorganisms are so powerful that they really rule the planet. In fact, when we did metagenomics, again actually we were, myself and Yogender were sitting after a meeting at Kolba beach in Goa, and after having a glass of beer, Yogender said, "Yaar, I wanted to enter into gut microbiome." And Yogender said, "यार मेरे पास तो सैंपल हैं लेकिन सीक्वेंसिंग करने के लिए पैसे नहीं हैं." I said, "मेरे पास पैसे हैं, दोनों मिलके करते हैं." And that's how I started. When we published the paper, I started reading about human gut microbiome. और तब मुझे पता लगा that each one of you is carrying so much load of in your gut of microorganisms. Nearly 1.5 kg, 3 pounds of bacterial load in my gut, and they decide about 90% of my functions. Not only this, the recent reports are telling that my oral cavity contains microorganisms more than the entire population of human beings in the world. So microorganisms believe it, and this AMI, they are so important. And microorganisms, now we are coming to the conclusion that microorganisms, they rule the planet. This realization came to all of us because of the revolution in sequencing technology and also in computational biology. Perhaps all of you know about it. And you also know it, that plants and animals cannot survive under many harsh conditions under which microorganisms, you can find them almost everywhere. And this realization, as a result of computational biology and sequencing technology, as of today we know that the total number of microorganisms on this particular planet is something around 0.9 to 3.2 into 10 to the power 31. You will not believe it, but these are the data, these are the facts, that microorganisms constitute 20 to 30% of the total living biomass. If that is the situation, now these are actually the facts, that if you look into the total living biomass on this particular planet, plants, they actually comes first, actually having 80% of the living biomass, followed by microorganisms 20 to 30%, and then animals and human beings. आप देखोगे ह्यूमन्स आर नियरली 0.1%. अगर 2 जीरो के बाद बाकी सबको हटा दो तो हम तो हैं ही नहीं. And public microorganisms, they perform functions which probably most of you know about, routine functions, but I'm telling you, they generate about 25% of the total nitrogen. They fix 2/3 of the oceanic carbon dioxide. They generate 50% of the oxygen which we breathe. पहले मैं zoology में पढ़ता था that we evolved from monkeys and apes. And when I started teaching actually the 3 domains of life, the phylogenetic tree, I realized हम तो microorganism से evolved हुए हैं, which appeared on this particular planet sometime 4 billion years ago. और वो हमें इवॉल्व भी नहीं किया, जिंदा रखने के लिए ऑक्सीजन और सब कुछ वो ही दे रहे हैं. If believe it, if microorganisms are not there, we will not be there on this particular planet. That is the importance of the microorganisms, and that's why I'm saying that microorganisms, they rule the planet, which we have neglected, of course, for a number of years. I'm speaking on, with this brief introduction on a topic which I mentioned actually, the metagenomics of the stressed environment. I just wanted to convey to those who are, and most of the microbiologists, they are doing metagenomics. And when you do metagenomics, better you select stressed environment. Because by selecting stressed environments, you get actually novel organisms, novel pathways. You start knowing actually the evolution of genes. And also you get to know and you get organisms that you can use for, as Professor Kuhar mentions, for biotechnology and for a number of innovations. But remember, before you plan any of your experiments for doing metagenomic, be very careful for the selection of a site. That matters. Sequencing, depth of the sequencing and the place or platform that you are using, and annotations. You need to be very careful. If you do some mistake over here, your hard work of many years will not be or will be of no use. Now I'll explain what I have mentioned, that stressed environments are very important from our experience of nearly 20 to 25 years. The first example I will take, metagenomic and our contribution to chemically stressed environment, which we call it as HCH dump site. Now we wanted to study actually the degradation of HCH, and we were looking for a place where there is HCH load because when linden is formed by the chlorination of benzene, nearly 1 ton of linden production lead to the formation of 10 tons of HCH mark that industry actually throws in the open. And we were looking for a dump site where actually the industry has already thrown this particular HCH mark. But हमें नहीं मिल रहा था. And finally we found out that there is an industry in Chinat in Lucknow that makes linden. And we went there, and for me it was very difficult to enter into the premises of the industry. But Mrityunjay Swar that time joined my lab, and he said, "Sir, चिंता ना करो, मैं तो सब कुछ पता लगा लूँगा." And that's how we entered the premises of the industry. He interacted with the scientists, they showed us everything, and by the side of the industry you find out tons of HCH mark which they had been putting on the government land. But they were not ready to tell us actually the dump sites they have produced. So what actually we did, and Mrityunjay also helped me, we bribed the Chowkidar, and he took us during night time actually to the HCH dump site located in Umari village in Lucknow. And it is around 40,000 tons of this HCH mark that was lying there. Almost half of it was HCH. Now, we started working on that particular dump site, highly stressed with chemical. To our surprise, we got lot more organisms that were degrading these chemicals, which were put into that particular dump site. But we wanted to do the metagenomics. And we planned a strategy. We actually isolated the DNA, did the sequencing, and when we did the sequencing, we found out that that particular site where there was half of it was chemical, was highly dynamic, and a new microbial community got evolved over there. Number of organisms like Spingomonads, Spingopixie, Novus Spingobium, Pseudomonas, they actually got evolved into that particular niche and started degrading or eating those chemicals which at one point of time scientists thought probably there will be no living system or microbial system that could degrade all these pollutants. But microorganisms evolved pathways, and when you look into the information obtained from shotgun sequencing and analysis, we found out it's a very dynamic site. Have got so many actually catabolic genes, IS elements, plasmids, and so on and so forth. When we further actually looked into it, we found out that Lin genes that are responsible for the degradation of these chemicals, they were highly enriched in that particular environment. And a new pathway came into existence into those organisms. I'll not get into details, but tell you that almost these organisms acquired under that particular stress genes and got them clustered, which were associated with IS elements. And when we stopped working on that particular dump site, we found out that organism had evolved complete mineralization pathway for the degradation of 2 of the compounds. For rest of the compounds, they are still working. The second example, actually, one of my students actually getting, Professor Kohar mentioned actually 75 students, but they were really very brilliant. ये काम मैंने नहीं किया, actually they had been working for me. And one of the student actually, he combined genomics and metagenomics. And as a result of that, he was able to construct the core genome into which, under that particular stress, these catabolic genes, IS elements, and plasmid genes are getting into the genome and making it more flexible for the degradation of all these pollutants. And that paper got published in ISMI journal. And he could also make depictions how these catabolic genes are getting into these genomes of these organisms under a particular SGS process. After having done that work for nearly 20 years, we wanted to actually, in association with Yogender and then Manikam and also many other colleagues actually, to get into the dump site. We thought that the single organism will work. So we actually produced in the fermenter in Tari lot of these tanks of these organisms and then did bioaugmentation, but we failed. And the lesson that we learned was that single organism will not work for the degradation or biocontamination, but what will be helpful is biostimulation and that is what we had been trying. The second example of the stressed environment, which I said we were interested actually to work on gut microbiome, and mention how we got associated with Yogendra Singh, where we took the samples from tuberculosis patients and also those patients that were treated with antibiotic and those which were not having any tuberculosis, took the samples, isolated the DNA and studied the gut microbiome. This particular paper was featured on the cover page of Environmental Microbiology. Our conclusion was, in fact, they were that there is a population of microorganisms that increases in tuberculosis patients, and those organisms are known to produce short-chain fatty acids and also propionate and butyrate. And all these chemicals, they are known actually to impair immunity, alter appetite, and also blood lipid profiles. The conclusion was, and many people are working on this, how to restore the healthy microbiome, looking into the studies which they are now doing extensively. The third example which we took up was take up the stressed environment, that is the Manikaran hot spring. Now perhaps you know it actually, world over there are hot springs, and Yellowstone hot springs in USA have been extensively worked on. The TARC DNA polymerase that comes from Thermus aquaticus is from the organism that was isolated from Yellowstone National Park. So we wanted to look into the hot springs in India. There are many, but one of them that is located in Manikaran in Himachal Pradesh at an altitude of 760 meters is the hottest spring in the country with a temperature of something around 96 degrees centigrade. And we could isolate. It's a very scenic place. If you get a chance actually, of course Dehradun is also very scenic, but you can also visit Manikaran. We have a Gurdwara there and a temple, and people probably believe, I believe that life originated from microorganism, but many people, they believe that it originated from Manikaran. Probably they say Manikaran because they don't have any knowledge of microorganisms. So Manikaran का उनका मतलब माइक्रो ऑर्गेनिज्म से ओरिजिनेट हुआ है मणिकरण में लाइफ एंड देयर इज ए गुरुद्वारा यू गेट फ्री फूड एंड एवरीथिंग अकोमोडेशन भी फ्री है वहाँ पर सो एनी टाइम व्हेन यू गेट एक्चुअली यू मस्ट विजिट दैट पर्टिकुलर प्लेस सो व्हाट वी डिड वाज आइसोलेटेड एंड वन ऑफ द इंपॉर्टेंट ऑर्गेनिज्म दैट वी आइसोलेटेड इज Thermus parbicensis, and when we sequenced its genome, we found out that the TARC DNA polymerase sequences which are present in the genome, they are really very unique actually in this particular organism. And this particular paper actually हम भगवान मंदिर में माथा भी टेका था कि ये बड़े अच्छे जर्नल में पब्लिश हो और कवर पेज पे इसकी फोटो आए. सो दिस जीनोम गॉट पब्लिश इन JBack एंड कवर पेज पे फोटो आ गई थी. भगवान कितना मेहरबान होता है कई बार. Right. So that's what actually when you looked into the sequence of the TARC DNA polymerase, you can see in the motif A and C, there are a couple of amino acids which are different from those that are already being used in the amplification of the DNA from different Thermus strains. So we have codon optimized it, cloned it in E. coli, and subsequently taking it. I will mention that later, and one of my students is presenting actually details how exactly we are going to commercialize this if it works. But we also did the metagenomics from the hot spring and did the physiochemical analysis and found out that there are, again it's a very dynamic site, 96-degree temperature, but completely the viruses are there, archaea, then firmicutes, protobacteria. Apart from that, we found out that two bacteria, Enterobacter and Bdellovibrio, they got highly enriched in that particular environment. We couldn't do anything with the Enterobacter, but we wanted to look into it, what is this Bdellovibrio doing? And when we looked into we could also, because it was an enriched environment, so we could assemble the genome of this particular organism. And we compared it with other, but when you looked into literature, what this Bdellovibrio does, This is a gram-negative bacterium, and it feeds on all gram-negative bacteria. Like enters into the cell, then multiplies, and then after there is a lysis of the cell, a new progeny is being released, and then that again swims, look for another host actually, or a gram-negative bacterium. Now, we actually looked into the genome and found out that like all other Bdellovibrio, it has a hit locus that makes this Bdellovibrio to sit and penetrate into the gram-negative bacterium and finally eat on the host and multiply. So when you looked into the genome of the Bdellovibrio from the metagenomic data that we got, we found out that one of the genes that makes this particular Bdello to penetrate into the E. coli was missing. So what we actually wanted to see was whether we could culture it. We could not. But we enriched that particular sample and added E. coli into it. And you can see over here, Bdellovibrio sitting on E. coli, not penetrating. So it's a different mode of action, which we call it as apibiotic mode. The another message that can be inferred from this, that by doing in silico studies, you can plan your wet lab experiments and do it. We didn't stop over here and looked into the viral genomes. We could assemble nearly 65 viral genomes from the data that we got, and also identified some bacteriophages to which these viruses they belong. In addition to this, again we did enrichment, and you can see in the next slide the viral particle sitting on the bacteria. So just from the in silico experiments which we did, we confirmed that yes, these communities are highly enriched with Bdellovibrio and also viruses. So we didn't stop over here. And we wanted to go ahead actually to another hot spring which was located at 3,000 meter above the sea level in Manikaran, some 20 kilometer from Manikaran. We call it as Kheer Ganga. It was a very difficult track. And most of the students actually who accompanied us, some of them they stayed actually in between, could not reach to the top. That's what I'm saying, that selecting a site, doing metagenomic is very important. But आपको उसके लिए थोड़ा सा हिम्मत भी करनी है. बहुत सारे बच्चे एक टेंट में ही ठहर गए, दिल्ली से थे मेरे पास. उनको पता भी नहीं था कि ये इन्वायरमेंट कैसा होता है. एंड व्हेन ड्यूरिंग नाइट दे वर स्टेइंग ओवर देयर, एक कुत्ता आया, उन्होंने सोचा शेर आ गया. भगवान की दया से वो बच गए. बट देन एक्चुअली couple of us we could make it to Kheer Ganga. हमारा टेलीफोन सब कुछ बंद हो गए थे. My wife was telephoning, and when I came back she said कि ऐसे काम दोबारा मत किया करो. But we got the samples actually and did the analysis. That's what I'm showing it over here. It was a very difficult exercise. We did the physiochemical analysis and we found out almost 4 years of analysis that we did actually found out that it's again a very unique site which gives us very unique organisms, sulfate degrading organisms. We could assemble complete metabolic pathway responsible for the degradation of sulfate, and probably this particular site also has got organisms that could be of highly biotechnological importance. So what I mentioned was, when you work with the stressed environments, you get very important bacteria. You come to know a very unique diversity. You also find out IS elements and plasmids and so on and so forth responsible for the horizontal gene transfer. In addition to this, you get organisms that you can use for biotechnological applications. And recently, I'm coordinating a project which has been sanctioned only last month by DBT involving TARI, IMTech Chandigarh, IHBT Palampur, and Asha is there, IIM Jammu. And what we are doing, taking actually for large-scale production this DNA polymerase Thermus parvitensis that we have isolated, and also this particular organism produces protease, and we also want to clone it and subsequently upscale it and see whether we can commercialize these two molecules. Apart from that, rest of the bioenzymes are being upscaled by my colleagues in IHBT and IMTech and also IM Palampur. as I mentioned and Professor Quad mentioned, actually this work which I'm presenting is almost a story of some 20 to 25 years, and large number of my collaborators, including Yogender, Jiten, Banwarilal, Manikam, Atya Kapale, then Prashant Pale, and many collaborators from Switzerland, Australia, US, and also from Germany. But apart from that, large number of my brilliant students who did this particular work, and we could publish more than nearly 60 to 70 good papers. They were really very hardworking and in fact guided me most of the time. Of course, to deal with them was at times was really very difficult, but now I see they are at very good positions, and you can see Mrityunjay who is there, he is DG actually, KIT TBI, and also chairperson of the Bhubaneswar Knowledge Park cluster. So they are, I feel very happy when I find them, they are doing so well. But in addition to this, I want to thank my colleagues from ANDC, Ravi and Seema, allowing me to work over there, and INSA providing me INSA senior scientist position, my colleagues from AMI for taking me to this fascinated field of microbiology. I will remain grateful to you all actually. भाई आपको अच्छा काम करते देख के मुझे भी खुंदक आती थी, मैं क्यों नहीं करूं. So that's how I got into microbiology, and Dev Bhoomi Uttarakhand University organizing this particular conference, particularly Nabil and Nirjara. Nirjara is also one of my brilliant students who is doing all this organization. उसके लिए तालियां बजा दो, मेरे लिए तो बजाई नहीं आपने. Thank you very much.
Over to chair and co-chair.
Okay, that was awesome, sir. I mean, I can believe that the force with which you talk and speak, I mean, it's lots of confidence which is showing up in your work. Awesome work, sir. And let me tell you, all of you, that it's not just a work of few years, it's, I think, his lifetime work. It is speaking now. A lifetime work for several years. And this is really awesome, sir, with the work which you have done. I believe that when COVID came, before COVID, people were not knowing much about microorganisms. And after COVID, when COVID came, everybody was knowing then what is PCR. Even the roadside people were knowing what is PCR, what is RT-PCR, what is CT value, what is PPE. People started knowing this. And the same way, when we came to this Dev Bhoomi University a few months back, I don't think so much of the people were knowing about microbiology. But by the time we will leave after these 4 days, I think everybody is going to know about the microorganisms. We are talking so much about the microorganisms and their role in this planet, everywhere. And as Dr. Rooplal is also saying, he has told that how important the microorganisms are and their role in this planet, much more important than any human beings, what they are doing actually. He has been talking to you that our body, how much weight the microbes carry. Let me tell you the other way, which we teach to the BSc first year. Our body has approximately 10 raised to power 12 cells, and our body has approximately 10 raised to power 13 bacteria in our body. So I always tell my students, really, are we only 10% of ourselves? 90% we are bacteria. That's what is the power of microorganisms is there. It's a wonderful thing that to isolate bacteria from different places. You have been, I think, going to various different places, isolate bacteria, Kheer Ganga and Manikaran and all that. Same way, I have also been going to different places. Manikaran is a very rich source of microorganisms. The hot springs are very rich sources of the thermophiles. Way back, Dr. Rooplal, we also isolated in 1995 thermos species. I was working on the thermostable restriction enzymes. We isolated several thermostable restriction enzymes, and one of them was taken up, bought from us by New England Biolabs also. And still we are getting, till now we are getting the royalty out of that. The point is, Manikaran is a very rich source of the thermophiles, which are waiting for you people to get exploited for biotechnological purposes. Go there, visit there. It's not just those hot springs. It's places like Kheer-Ganga. It's places like deep in the ocean where you can find microorganisms so important for industry. Let me tell you that microorganisms, Indian soils are very rich. The Indian habitats are very rich in microorganisms. Let's follow the footsteps of Dr. Rooplal, who has given an awesome and very comprehensive, in a very less time, he has given us the knowledge about what microbes are and what microorganisms can do. One thing I have always been telling you, Dr. Saab, this is not Badello, this is Dello Vibrio. We always talk about that B is silent. That somehow the bacteria are there to be named. And let me thank again Professor Rooplalji. He's not just the name is Rooplalji, his mind is also a Rooplal. He's really a very good friend and a very good human being, a very good friend of us, and a very good scientist. Thank you so much, sir. And one last thing is that, as Professor Kohad yesterday said, that we have in place, there's a box where you can put up your ideas, innovative ideas into that, and that box is still waiting for you. All of you are invited to put your innovative ideas into that box, sir. Right, sir? Yes. Thank you so much.
Thank you.
Now I request our chair, Professor R.C. Kohad, and the co-chair, Professor Prince Sharma, to present a token of gratitude to our plenary speaker, Professor Rooplal. Can we have a huge applause for being this amazing session? A person who already has 75 scholars under me, and that's so enthusiasm. Congratulations, sir. Now I request our Honorable Vice Chancellor of the University, Professor Ajay Kumar, sir, to please join us on stage. Thank you, sir. Can we have a round of applause for Professor Rooplal? Now I request our Vice Chancellor, sir, to kindly present a token of gratitude to the chair of today's session, Professor R.C. Kohad. It was a beautiful and rich session that we have witnesses today by Professor Rooplal, which was truly enthusiasm. It was really learning for us, sir, being a scholar of this university. You already have 75 scholars been underdone you a PhD, and you have been so enthusiasm. Congratulations to once again you, sir, for that. With this, we would like to felicitate our chair for this session, Professor R.C. Kohad. Thank you, sir. Now I request sir to please kindly felicitate our co-chair of this session, Professor Prince Sharma, President AMI. Thank you, sir. Now, after this session, we have a high tea for all the people being here. So for the students and scholars, we have the high tea at the Pandal outside the hotel management. Faculty members at the Ayurveda hall, which has been available next to the hotel management itself. All the delegates and speakers will be joining us in the hotel management restaurant for the high tea. After the high tea, we'll be meeting once again for the session 1 in the main Pandal, which will be next gen microbiology education, innovation and research. Session 2, current and future trends in antimicrobial resistance to the promise of vaccines will be happening at the Ayurveda seminar hall. And oral presentation, poster presentations will be there at the respective schedules, which has already been shared to everyone. And we'll be joining back once again here at 11:50. So I request everyone to please join us, who will be the part of session 1 in the main Pandal. Thank you. Everybody has to reach here by 11:50. We have just a high tea break of 20 minutes. Thank you. It's just a 20 minutes break. I request everyone to kindly, please, as soon as possible, they can reach to their respective places for the high tea so that we can start the sessions once again back at 11:50. Hello. I welcome you once back for the next session 1 of this today's session. I request all the participants and delegates to please join us as soon as possible. We are starting with the session 1 very soon. All the dignitaries who are sitting here, I request everyone to please pass on this message to your colleagues and your friends to join us as soon as possible, because we need to start with the session 1 of today's. Volunteers at the back, make sure everyone is seated well before the session starts. Hello. I request everyone to kindly seat it well. We are starting with the session in the next 2 minutes. I request everyone to kindly please be seated. I am starting with the session first for today's session. I request all the dignitaries standing in front, please kindly be seated. I was about to start with the session. I request the dignitaries standing in front, I request you to kindly please be seated. We are ready to start with the session. So we are starting with the session 1 now. The session 1 is Next Generation Microbiology Education, Innovation and Research. The session on Next Gen Microbiology Education, Innovation and Research focused on the latest advancement in the microbiological studies, emphasizing modern teaching methodologies, technological integrations, and innovation research practices. Experts highlighted the importance of interdisciplinary collaboration, digital tools, and sustainable approach in microbiological education and research. The session aimed to sustain an approach in microbiological education and research, inspire students and researchers to adopt next-generation techniques and addressing the global health, environmental, and industrial exchange challenges through microbiological innovations. Before I invite the chair on the dais, I would like to introduce the chair for this session with you all. Professor Mrityunjay Swar, Director General at R&D and Innovation at KIIT Bhubaneswar. Professor Mrityunjay Swar is a distinguished microbiologist and extensive research experiences and promoting affordable diagnostic in rural healthcare. He has been successfully led multiple national and international projects funded by prestigious agencies including DBT, ICMR, DST, World Bank, and US Optimum Foundation and Hindustan Unilever Limited, covering areas such as microbial floral characterizations and vaccine development, the rural innovation. His contribution is extended to industrial collaborations and innovative initiatives like establishing technology business incubators and rural women technology parks. With this, I would request our chair of the session to please join us on stage. Can we have a huge applause for our chair, Professor Mrityunjay Swar? I welcome you once again, sir, to Dev Bhoomi Uttarakhand University. It is the time to introduce the co-chair of this session, Professor Azam Ali Khan. He is a Chief Executive Officer at TIDES Technology Incubation and Entrepreneurship Development Society, IIT Roorkee, one of India's leading business incubators, and an MLS of IIT Roorkee with BTech and MBA in Finance from the University of Mumbai. Professor Khan plays a pivotal role in fostering innovation and entrepreneurship. His specializations is in startup ecosystem development, idea generations, business model canvas, faculty entrepreneurship, and startup financial management. A frequent guest speaker, jury member, and national advisory committee expert, Professor Khan is known for promoting the lab-to-market approach and monitoring early-stage startup innovators and faculty entrepreneurship, especially in emerging sectors like renewable energy. With this, I invite the co-chair of this session on dais, please. Can we have a huge applause for Sir? Now, it is the time to listen from our keynote talk by our speaker, Professor Mrityunjay Sir, Director General at R&D and Innovation at KIIT University, Bhubaneswar. Over to you, sir.
Yesterday about microbiology, and Professor Kuhar mentioned about this year more towards education in microbiology. So I worked with Professor Ruplal as a PhD scholar, learned a lot how to be grounded and work on good science. But today I'm going to speak for next 15 minutes that science is important, but most important is don't make science a parasite. It means can science builds technology which builds enterprise. You might build enterprise or you might transfer your technology which is full of science and technology, but it brings commercial viability. If you don't do that, science remains parasite in nature. So with that note, I thought I'm in Dehradun, Dev Bhoomi University, and wherever I go, I always meet my friends. So I've met so many friends today. But let me tell you, the mindset needs to be changed of the academic research. Otherwise, education, research, innovation, and entrepreneurship cannot coexist together because each one of these subjects are very different mindset. So today I'll speak to you with a lot of facts and figure. As a scientist, what you can do, as a researcher, what you can do, but also how can you build a new culture of innovation ecosystem which promotes science as the backbone, but the front faces technology and enterprise creation. And everywhere innovation is there. So changing the mindset and building the innovation ecosystem, entrepreneurship-driven ecosystem in this institution. And I thought this will be very, very important because this slide, if you look at it and understand, all the great scientists of the globe, even I met 17 Nobel Prize winners, brought them to my university kit. Each one of them have their own companies. And that's the message I want to give you. Doing science is great, but the great thing will be if you convert your science into technology, not only at the laboratory, but gets into the field and builds commercials. If you do that, I think things are going to be much more exciting for you, and every day of doing science will be exciting for you. So look at this slide. All of us, those who have published 500 papers, H-index 200, everyone, I think all are in the bottom of the path building exercise. That's the message I want to give you, that we have been celebrating our doing science, but what will happen after completing your scientific career? Is it going towards an innovation-based ecosystem or just like publication and patents? So that way, all of us are busy in academics, research, knowledge creation, knowledge dissemination as a professor. But then the next path is quite exciting. That's what I'm going to speak to you afterwards with a lot of facts, that you do science, but you do science for a purpose. So what is next part is technology enabling, intellectual property, design thinking, building proof of concepts. You might venture it as startup, you might co-develop, you might tech transfer, you do trials, certifications, validations. These things are missing in our lab because when I meet my friends and even seniors and juniors, they celebrate of a publication that's very good because you must do good science, basic science is to be strong, but then your mindset should be, how do I translate some of my scientific experiments to build interesting product, process, or even service which is for the mankind. And by doing this, you can build commercials. So this path building exercise is very, very important. And it looks like only a simple slide, but if you pick up 20% of it, you will be the greatest scientist of the globe because one of your product, if it comes to market, whole country, world is going to celebrate. So I'm going to speak to you about, not about science, what I'm doing, but the process of doing interesting technology development exercise. So I'm here in university, all of your students, faculties. Now look at what is missing apart from education and research. The missing part is a culture, a mindset of platform creation, which we call technology business incubator. So we need to build a culture in this microbiology world because microbes are so tiny to handle it so nicely. But you need to really build a new culture of using microbes to create interesting things, which I'll give you a couple of examples afterwards. It's a new mindset, it's a mission program, it's a process, which I'll tell you very recent now, just now, and it's a platform exercise. So you're doing PhDs, you're doing masters, but you should do it for a purpose. So you find a lot of challenges in this, but let's convert into opportunities, which you'll realize that how students are doing exciting work. Great science, great PhDs, but building great companies, hiring 500 people. That's what science should do today. So the process is very, what you call, structured here. And people like us who are building now startup ecosystem, promoting entrepreneurs from scientific community, the first and foremost thing, and you realize in my slides how we have built it up. The regional mapping, the strength, the opportunities, the need and demand is very important. So let's not do science what we have been doing in postdoc or in PhDs. Let's do science, first focus on where you are, what interesting science you can do, which might be important for the region. So that's what I think we need to build as a part of our path and process. Identify the problem statements. I'll give you an example. We have a company called BabyQ who says, very simple, is a pharmacy student who says when patient goes to a doctor, they give a cocktail of antibiotics without knowing the infection is due to bacteria or virus. How can you tell doctor in 10 minutes, not more than 10 minutes, this guy is infected with bacteria, not virus? Imagine, simple, basic microbiology, but nothing is there. Everywhere you go, eye infection, any infection, they give you a cocktail of antibiotics. They don't know what it is. Now, he comes up with a BabyQ, simple microfluidics technology, hardly cost you 70 rupees. Now, it might be 20 rupees in scale. 10 minutes tells you what kind of, not what kind of bacteria, it's a bacteria or a virus. Next is it's a gram-positive, gram-negative. Imagine, you as a microbiology researcher and students must think in this lens. Do research, but do that for a purpose. What is the unmet need? How do I bring more design thinking processes into it? Prototyping, we never talk about prototyping. We always say experiments. But build prototypes, look at those things, and create right team, and look for patent or tech transfer, or look for different kind of strategy of investments. And you have to be a good management guy without a degree, but do a lot of these outreach activities. And I said this in a simple way for all of you, because this process, if you follow, I think your science will be quite exciting, and you realize your language will change completely, and always you'll be funded. I'm very happy to tell you, when I came back from Zurich after PhD from Professor Ruplal's lab, went to Switzerland, did a postdoc. That's what the mission, the ambition of people like us to come back to get a position. I was placed in KIT University, the director, at a very young age. But then I realized what I need. What I'm saying today is that's what I followed. Build processes what Odisha needs. Build process what biotech school needs. Today we are very fortunate to be one of the top class in the country. I think good students, good faculties, and our students getting into Cambridge, Harvard, is all very normal process. And many of them build their own companies, and they have really close to 10,000 employments they have created through incubation processes. Great companies. That's the process I am talking about here. Now we need to also change our language. All students, those who are doing PhDs, I know you have to follow your professors. But if you come to my lab, my students are all professors. They behave like me because they have to be independent. So they're given problem statements, they bring problem statements, we discuss, and they bring solutions, they write manuscripts, we only review it. So the language, what I'm going to tell you today, most of us don't know about it in the scientific world. We must talk about what science you are doing, if you're building a technology, what TRLs you talk about. And we go to institutions like premier institution, I'll not name them, be it Bangalore and Delhi, they talk about, I have built a new technology. Dear students and faculties, believe me, they are not technology, they are scientific experiments in laboratories. They cannot be called as technology. So when you pitch it or present before somebody, they don't like it. And then we say we are not funded. So our language of new science-driven entrepreneurship is more towards technology readiness level. I'm not going to speak on more on that. These are very metrics. I think I'll keep the slides for all of you. The technology readiness level should be our next language in all scientific institutions. If we don't do that, I think we'll never talk about technology. We'll only talk about science. Market readiness level is very, very important. We always say, I have built a technology. It's not for market. It's only for your laboratory, just to showcase in a poster. It is not for applications or deployment. So, dear participants, please change your mindset, change your language in terms of TRLs, MRL. Very, very important. What I'm saying, do good science, but also bring a new mindset of TRLs and MRLs or even IRLs. So I'll give you an example for next 5-7 minutes that what I'm talking about here, maybe I have talked to the leadership of Dev Bhoomi University, we'll also talk to them today. You don't have probably an exciting incubator, technology business incubator. I joined KIT University, started building it up. I have my own group having scientists, PhD scholars. My H-index is going and flying like, I think, you know, I'm proud at this age, it's going up to, I think, 55 probably, and 9,000 odd citations. It will go, 30,000 probably. But it is not exciting for me. I never celebrate this, never wrote any message on those things. What I like about how we are promoting through a platform, the exciting science leading into exciting technology, validations, piloting in the test beds, and getting into enterprise mode. And they are hiring students, not in form of PhDs, but in for employees. So TBIs are just like an institution. It's an ecosystem which never works in isolation. It brings science, technology, development, validations, IP, tech transfer, regulatory framework, design thinking, all what I said, including investments. Dear students, those who are getting into this domain of doing science, please bring some of the mindset of creating exciting things out of these TBIs. So that will be very exciting. I told you in the process, first one, that you need to look at the local problem statements. So when I came to Bhubaneswar, Odisha, we have a lake, Chilika Lake, it is suffering. We are microbiologists, we have some responsibilities. That's what we talk about, the need of the region. So we now help some of the students, faculties to build exciting technology, which is the phragmites, which is a weed species, now being converted. I think with the scientist sitting here, Dr. Chinmay studying from IMMT, converting those phragmites into particle boards and papers. Imagine if science can lead such kind of impact, this is going to create huge economy for the region. Now we as a scientists have a lot of responsibilities to do good science for a purpose. This is what I'm telling you, it's a purpose which is important. This, I love it because a material scientist who has done PhD and says, I want to help farmers through my scientific experiments. So what he says, a phase transition material, which is once you charge, you can put in a container, you take the whole container for next 12 hours without any cold chain, without any energy management. Imagine, if you really want to help farmers, you need to build infrastructure of cold chain, very expensive. Government cannot do everything. So he said, let's decentralize cold chain, and this simple box is so interesting, simple innovation which helps farmers to take their good from the, from the farm to the market for next 10 to 12 hours in cold chain without energy. Imagine, this has huge impact. The company is growing and growing. Great material science. But a similar professor might have published 50 manuscripts on that. But somebody thinks with a different mindset, let me convert into application which has commercial viability. Impact, commercials, what a satisfaction the scientist would have. The professor is so happy from an institution. Very simple guy. He says, we are talking about, this is a technology from SN Bose. SN Bose celebrates patents, technology, but they are not into commercials. So somebody comes up and says, I want to pick up technology from SN Bose institution, which is a premier institution for doing great science. But he says, let me build a device where you put your fingertip without taking blood, it can measure hemoglobin. Today hemoglobin, tomorrow creatinine, bilirubin, all can be done under simple device. Everybody was laughing on this. He says, by physics, this is not, the principle why this is not be very possible, not accurate. Today, the company is in the market, invested by Mankind Pharma, Sun Pharma. We also exited in the company, made good amount of money. What it says, a device, you put your fingertip, 30 seconds gives you hemoglobin. Imagine, 10,000 pieces in the market. and Mr. Maselkarji, everyone talks about, you can Google called EZDRX. Any company I'm talking about, you might Google it, you'll see what level they are now. They're all scientific principles. Same SN Bose Institute, professor, innovations, patent, limit. Then next phase, think, bring a new culture, new mindset. Similarly, I will not talk about it, I don't have much of time. This is what is very interesting. Great chemist scientist from Cuttack, from Odisha place, and he did PhD and postdoc from Japan. He was coming back, wanted to be a faculty. I said, being a faculty is the easiest job. What else you want to do? Somehow she got good coaching in our TBI. I have a lady, Dr. Namrata Mishra, will be speaking maybe afterwards on this. The TBI, what we have been in KIT, has really helped her to build one of the most inventive company of the country now. Everybody is honoring her. So what she makes a protein, which once you take it, it drills your, let's say, liver cells, gets inside liver, release drug molecule. Everybody says this is Star Trek. It says some sort of science fiction. Everybody, I think I will not name him, a very famous chairman of a committee who always gives bigger money and said, grade C, not feasible. Ladies and gentlemen, we must change our mindset to do science which are very high risk in nature. Same gentleman, I convinced him somewhere after he said it is grade C. I said, sir, I know this lady. She will do wonderful job. Please support. Little money of 50 lakhs rupees that time from BIDAC today. I see Sandhu sir is probably here. Everybody talks about her. Honorable President of India, Prime Minister Office. If she's successful today, let's say it will take a lot of time for her. Pfizer, Novartis, everybody is watching her. You can Google the company called Scigenica. I think every award she has won today and huge amount of money she's raising. What I'm saying, she was a great chemist, got a platform, new mindset. Today, everybody talks about her in India, in UK, in Ireland. If she's successful, the regimen of cancer therapy strategy will completely change, very precision therapy. Then you can imagine she will be more than Biocon in future. Think about it. Very young man, PhD in our good school of biotech. I'm just giving you examples, seeing faculties, students here. The professor is a material scientist and doing some protease inhibitor experiments. Professor is busy in publishing paper, PBAS, promotions, professor became professor. What is next? The student who's getting PhD says, I want to do something else. Coming to Innovation Center, you can Google the company, even Panacea Biotech buys this from them as a protease inhibitor, which is world's first thermostable protease inhibitor, and is growing and growing. That's what the science is very strong with publication patents, but it's a product which is in the market. So dear students, faculties, we need to change our mindset and create enabling ecosystem to promote great education of microbiology, not just from textbook or from paper, but also to apply it to create such exciting things. So 2 minutes, I'm from KIT TBI, which is India's one of the last, Azam also will speak about that, one of the largest incubator, not by size, but the impact we create in the Bhubaneswar, in the east and northeast. It is growing and growing. Everybody says incubation is not sustainable. Let me tell you, this is not only sustainable, this can be highly, highly profitable. And the best part, we are creating a lot of impact. I'll tell you, 9-10, when you start doing this, as Professor Rupalal said, microbiology, everybody saying not possible. For him, today he is a great one. Similar, I learned from him, started TBI in KIT in 2009-10. The chairman, former DGCS, was laughing at us that ODS people are very lazy. They take a lot of rice, fish, and sleep. And it's not feasible, entrepreneurship from science. So today, we are well known. Every ministry supported, growing and growing. 490-odd companies, 10,000 employment. Imagine students, where you will go? There is only few companies, biotech. Our students are employed in startups. They are paying even more than bigger companies. So the culture has changed now in many parts of the country. I want here through 60 AMI, Dehradun should learn this and Dev Bhoomi can deploy this. So in all sector, these companies are raising funds not from government only, but also from private. It's a large ecosystem, very modern laboratories, but these laboratories are certified by NABL, which are not generally in science laboratories, and they are also earning, people are learning new mindset. We bring ideas from you, help you to build team, build proof of concept, MVPs, product, enterprise, all are based on science. Everybody does experiment. They use HPLC, LCMS, whatever you do, they clone, sequence, but at the end, they have something in the mind how to translate it. We are becoming very infectious these days, getting into all east and northeast region of India, helping those states. As I said, if you do it with a strong team, that's what one of my team member, Dr. Namrata here, you can, I think, do great things for the country. Science-wise and also the entrepreneurship-wise. We got national award by Honorable President of India, then Minister, the most IP incubator award. It's all going on. I'll not talk about it. Startups are doing great. And this is all because of my team, 106 team in our incubator who are the employees. And many of them are PhDs. Many of them are PhDs. They are not, they have done postdoc somewhere, but they are enablers of science-led technology. All startups coming forward. I wish this conference will bring this culture of great education of microbiology, which you have learned past, but today, let's not be very superficial. Learn it for a purpose, do it for a purpose, and do PhD for a purpose. With this, I thank everybody for listening to me. Thank you so much.
Thank you, sir, for sharing your valuable thoughts with us. Now I request Professor Dr. Nabil Ahmed, the organizing secretary of this conference and General Secretary of AMI, Professor Namita Mishra, to please join us on stage and share a token of gratitude to our keynote speaker, Professor Mrityunjay Swar, Director General at R&D and Innovation at KIIT University, Bhubaneswar. Can we have a huge applause for our keynote session and a cheer for this session? If anybody has a question from the audience, you are free to ask. Students at the backside, if you have any question related to the session, what sir has delivered, you are free to ask those questions. Sir and ma'am, I request you to please stay back and share a token of gratitude to our co-chair, Professor Azam Ali Khan, for this keynote session of this day one. As a chair of the session, I request once again Professor Mrityunjay Swar sir to please accept the token of respect and gratitude on the behalf as in chair for this session, sir. Thank you, sir. Thank you, ma'am. Thank you to our chair and co-chair for joining for this beautiful session with us. Circle of Science, a first panel discussion of today's Bench to Business, the Startup Journey. The panel discussion on Bench to Business Startup Journey focused on transforming innovation research ideas into a successful business ventures. Eminent entrepreneurs, industry experts, academicians shared insight on the commercialization of laboratory research, funding opportunities, incubation support, and challenges faced by the startups. The session highlighted the importance of mentorship, collaboration, and innovation-driven strategies in bridging the gap between scientific discovery and market application, inspiring young innovators and pursue entrepreneurship. For today's panel discussion, the moderator would be Professor Namita Mishra. Dr. Namita Mishra is a distinguished life sciences professional with the MSc in Bioinformatics from Banaras Hindu University and PhD in Life Sciences from CSIR IIMT, followed by a postdoctoral fellowship from NPDF DST SERB in Bioinformatics. And she is presently serving as a head Bioinformatics at KIIT Bhubaneswar and the General Secretary at AMI. With this, I request the moderator for today's panel discussion, Professor Namita Mishra, to please join us on dais. Pardon? And I would like to invite the panelists. I request Namita Mishra ma'am to please join us on stage. With this, I would like to invite the panelists for today's panel discussion. Dr. Shivani Sharma from TIDES, Dr. Tanvi Govill, she'll be joining online with us, Dr. Anvita Gupta, Dr. Shikha Dhawan, Dr. Rakesh Vig, Dr. Urvashi Kuhar, and Dr. Vinita Jindal. I request all the panelists to please join us here for the panel discussion.
Called collaboration. We all want to sit together.
Over to you, ma'am.
Very good afternoon, everybody. Good afternoon, students. A little bit louder. A little bit more louder. Very good afternoon. We know that we have all become a villain. We are standing in between lunch, but we'll try to make it interactive, okay? And just a note that there were two panel discussions. One is bench, next generation microbiology innovation, bench to business, the startup perspective, and the second panel discussion was industry perspective. But because of the paucity of time, and most important than that, because when we are talking about the innovation spectrum, both industry, academia, startup, I think all have to come together to have a very holistic perspective. That's the reason we have merged both the panel together. And now, when we talk about microbiology, I always say it's very transformative. A small microorganism creating a very big impact, whether it's good or bad. But definitely today we are talking about another kind of transformation, that is how a research idea transforms into sustainable business. And I'm sure all of you would agree that over the years, the science, we are witnessing a paradigm shift in the science, from research labs to startup incubators, from culture plates to boardrooms. Science is no more confined to publications and patents. It's more moving into products, policies, and people life. So that's the beauty of thing. And coming to the second panel, that is industry. I think there's a lot of debate that happens around industry and academia collaboration. They are both the side of the bridges. one creating knowledge, other creating impact, and sometimes collaborating, many times criticizing. So let's hear the perspective from this panel members, and I'm very glad that we have a very holistic panel who are representing all the vertices of innovation. So we have Dr. Shikha Dhawan, who has an industry, yes? Okay, wonderful. We have one more panel member who has joined online. So there's Dr. Tanvi Govil from South Dakota Mines, who is bringing a global lens. Dr. Tanvi, I hope we are audible to you. Yes? I think the organizers have to see, probably she can listen to us, but yeah, check the audio. Then we have Dr. Shikha Dhawan, who has years of academic experience, industry experience, startup experience, now mentoring a lot of startups. I've been working with her for the past couple of years, so I know she has a broad experience which she is going to share with us today. We have Dr. Vinita Jindal, who is leading the technology transfer and IP vertical of BIRAC, helping a lot of startups, immense knowledge in IP, immense knowledge in technology transfer and commercialization. Many years from BIRAC, she has been working with a lot of startups. Thank you so much for joining. And we have Dr. Anvita Gupta. She's herself is an entrepreneur and is working on a very interdisciplinary science, how to fuse AI and science in solving some of the very important diseases. We have Dr. Rakesh Vig, who is MD of Microwin. With him, he's bringing the industry perspective. And at the end, we have Dr. Anvita Gupta. We have Tanvi Govil. Sorry. Yes, Dr. Shivani Sharma, who has been working from IIT Roorkee incubator, so has a lot of incubation and startup experience. So before we open for a panel discussion, I think let's open up with a quick round of a rapid fire round. I'll pose 2 questions to each one of you, maybe 30 seconds, and then we'll go one by one with the speaker. So the question to each one of you is, what is that aha moment? You know, what pushed you to move from the conventional path and promote innovation, entrepreneurship, or being the other side of the table, supporting a lot of research, translational research happening? That's my first question. Second question is, how do you define the next generation microbiology? The term next generation, whether it is a time for deep tech, whether it's defining collaboration, or it is all about mindset, as Professor Mrityunjay Suar just spoke about it. So I'll start with me. For me, my aha moment was always that when I was doing my research, like many of the students here, I also got a couple of questions when we were having a break, that are there any opportunities in pharma sector? Are there any opportunities in science? So that is the time in the master's and PhD were always crib. But the moment when I got to know that yes, there are immense opportunities, it's just that awareness is lacking. I think that was my changing period, that I said that let me be on the other side of the table and support researchers in bringing their ideas into some dreams, into some products. So that's what's my aha moment. I'll start from you. 30 seconds, very quick. Yes.
Thank you, Namrata. Life every day is an aha moment. I'm Dr. Shikha, and if you have found the ikagai or the purpose of your life, my purpose is to serve the underserved. With that passion, I've been spreading entrepreneurship from farmers, self-help groups to startups in tier 2, tier 3, tier 4 cities also, and sky's the limit and the world is my oyster. That is my aha moment.
Wonderful.
And the microbes rule the world, as Professor Rupral elaborated in the morning. So this world has always fascinated me, and next gen is about blending microbes with AI, taking microbes and AI closer to benefit mankind.
Wonderful. Over to you.
Yeah. So for me, I come from a very conventional background when I completed my PhD in botany. So as such, when you want to do something for the society, but you don't know where to go other than the academic field. So then that time I got an opportunity where, Department of Science and Technology, they had a very good program for the women scientists. Basically, they were inviting applications to start their career in intellectual property rights. So that was something I feel actually changed my whole, career. So I did, completed PhD, and I was teaching in a private college. So that time I thought I want to do something, but what is there? So that was the opportunity who actually completed or who actually completely transformed me as well as, career prospects. So when I joined BIREC, so basically the organization fulfills if you want to do something for the society. So that's where you connect yourself. When you have the ambition to serve the society, that kind of places you join and you get an opportunity that actually, fulfills your dream. So that's where I think that, opportunity which I got that time in 2008, that opportunity kind of a aha moment for me. And of course, when we talk about the deep tech or microbiology, next-gen microbiology, or even when, everybody is talking about the AI, artificial intelligence, of course, they are going to rule the next, maybe a couple of decades or maybe more than that. So in BIREC, we are also gearing up, providing funding support on deep tech science. Thank you.
Hi, my name is Anvita, and I also think life every day is an aha moment, and there have been many aha moments in my life. So my story is a little unique. I grew up in the US. I wanted to be a doctor for many years. My parents came to the US from India for grad school. My dad went to IIT Kanpur, and I didn't want to be an engineer. I thought they were working too hard all the time, right? But somehow I started doing research at a pretty young age into in biology because I wanted to be a doctor, and started using AI to do some work with drug discovery. At that time, we just called it machine learning. And so this was work that I did in collaboration with Harvard Medical School, and we were developing new machine learning algorithms to find new drugs for Mycobacterium tuberculosis. And that algorithm was immensely successful. Harvard then took these drugs through preclinical trials, and they found that 80% of them were actually active against the drug-resistant bacteria. There's a huge success rate difference, and so for me that was a huge aha moment, and that work Harvard is getting a patent on, and I was invited to present that at the White House to President Obama, and there the president talked about how personalized medicine would only be enabled through AI, how AI was crucial for microbiology, for drug discovery. So I think for me that was a big aha moment, that this work can have a huge societal impact, and that's what you think about from an early age. You want to see that's what you care about, right? How can my work help society, help other people? So that was, I think, one aha moment for me, and then I felt that the best way to bring my work to the world was through startups, through industry, and so that put me on that path. And then your second question was about the next stage.
Next generation microbiology, what is that?
What
is that next generation?
Yeah, so people have mentioned AI a lot. Definitely I am also a big AI fan, and I've worked a lot in it for 10 years. But I think the biggest thing for me will be how AI makes microbiology more productive. Right now, a lot of students here would have had that experience that they sit 10 hours in the lab, or go nights, go early mornings, and they get tired, and the research sometimes gets stuck in circles. So I think AI can really help with making research more productive. So we do the experiments, only the experiments we have to do, we see huge success rates, and then we can take that forward. So that for me is the future of microbiology.
Wonderful. Dr. Rakesh.
Thank you. Myself, Rakesh Veg. See, as a student of microbiology, and everybody knows that microbiology student is a student of the laboratory, right? He sits in the laboratory, do all those practicals, experiments, and do his thesis extra. For me, a journey from laboratory to starting my own enterprise and building up a company from scratch. I, being student of microbiology, didn't know anything about entrepreneurship. Now it is a buzzword, but when I started it, it was not that buzzword. And secondly, you see, when you enter into business, then it is a different horizon, different ball game altogether. You start an enterprise from the scratch. You build your infrastructure. Then you become a cost accountant also. Then you become a finance accountant also. Then you are the supervisor. You are the security agent also. You do the marketing for your company. So starting from, just imagine a transition that an entrepreneur goes through from a laboratory, doing all the experiments, and then marketing his product. So for me, transition from a laboratory to building up an enterprise, to starting an enterprise, and then making it a successful enterprise is a aha moment for me. And future, yes, I think microbes has the largest future. Before that, let me just share it with you. The name of my company is MicroWin. See, we are microbiologists, and we offer winning solutions. So that is how this name MicroWin came into existence. And as far as the power of microbes are concerned, world has seen the power of microbes during COVID-19. So microbes, they can rule, they can bring the entire universe or the world to a standstill. There's huge power of microbes. The future belongs to microbes. Future belongs to microbiologists. And with the changing, regulations and newer and newer non-tariff barriers, microbiologist has a wonderful career. So for me, being a microbiologist is an aha moment. Starting my own venture is another one, and scaling it up to the next level is the third one, ma'am.
Dr. Shalini.
Hello. Yeah. So myself, Dr. Shivani from IIT Roorkee Business Incubator TIDES. So
A little bit louder.
Hello.
If we can just...
Hello. Yeah. It's fine. Yeah. So myself, Dr. Shivani from the IIT Roorkee Business Incubator. The aha moment for me, like I'm again from the research background, I'm a synthetic organic chemist. So when I have joined TIDES, I see how we can, like in the research, we are usually focus on the technology development. Our focus, how we can build a new technology which is really impactful for the society, but we don't know how to execute. So incubator play a very important role because they are telling you, if you have a good idea, how you can execute this idea to in a perfect way so that you can transform into a successful venture. So that is the aha moment for me, where I am now guiding various early stage startups to how they can transform their product to a successful venture. And for the next generation, like everybody is talking about the AI, the integration with the microbiology, but I thought like this circular bioeconomy is also playing a very important role in the term of sustainability, so we should focus on that also. Thank you.
Dr. Tanvi, can you hear us?
Yes, I can hear you guys. Can you hear me?
Yes, we can hear you now. So very quickly,
hi,
I hope you could hear the question that was posed to all. I'll repeat.
Yes, I can.
Yeah, please. So what was the aha moment for you, and what is that the next generation microbiology defines in one word?
Okay. So myself Tanvi Govil, and I'm an early career assistant professor in South Dakota Mines, USA. But just to talk about, just bit of my journey, I started with working with extremophilic microbes in Punjab University, India itself, with Dr. Prince Sharma in his lab. And then I continued that journey working more with extremophiles now with Dr. Rajesh Sani, who is also present in EM at this point of time. And for me, the most aha moment is that In my current position, I have been able to have my own entrepreneurship venture along with again Dr. Sani. That's a Vermigenics. And our aha moment really has been that whenever we are actually looking for working with industries, we had spent years working with extremophiles, thinking their value was obvious. They had unique enzymes, stability, heat tolerance, etc. But when we started talking to companies, nobody asked us about biology. Every single person asked us, can this survive in my process? Can this cut my costs? Can this reduce my downtime? And that was the moment when everything shifted for us. We realized that industries are not looking for solution you are bringing to them. They're still looking for solution that they want to see moving forward and the relief from a problem. And suddenly all of the science changed for us. So in the past years, we have been able to contact a lot of market people here, and main aha moment for me was that US market is quite open and they are letting us also enter the entrepreneurships. And when it comes to next generation of microbiology, I feel interdisciplinary science that could be done with microbes, that is the next generation of science. So for example, if some companies may be making a living insulation foam, basically merge bacteria with material science, with chemistry, physics, that's the next generation.
Wonderful.
Thank you.
So the reason why we opened this panel with this question is for you girls and boys sitting right here, because there's a lot of doubt as what next after your masters and PhD, what is next? Please understand that there is no more that conventional path that after masters you have to go for PhD, you have to go for higher studies, you can either be a professor or a scientist. We have seen a lot of examples. Many of us have already shared their journey that now IIT graduates, a lot of academic are turning into entrepreneurs, right? So please utilize this networking platforms very judiciously, because you never know that when that aha moment for you will come, when the door of opportunity will come. So that is up to you. Please look around. There is interdisciplinary, this is this era of interdisciplinary science, AI, healthcare. When you talk about healthcare, it's no more about only clinicians or medical science. We need biosensors, we need biomedical engineers. So that is something, girls and boys sitting out here, please, this is a very important takeaway message for each one of you, and you have examples right here. Now with this, I think we'll open with a proper panel discussion. I'll be posing 2 questions to each one of you, but let's be precise, because what I want is that the last maybe couple of minutes, let the spotlight be on the audience, the students out there. If you have any questions, you can ask. So I'll start with my left, Dr. Shikha Dhawan. You have been a living example of innovation and action. You have supported a lot of startups, you have mentored, you yourself has also into a couple of businesses. So what has, being a woman, what has changed over the years, then and now as a woman entrepreneur? Because we see very few women in the leadership position, but tell me your experience as an entrepreneur, as a mentor.
Thank you, Namrita. That is the question I always get asked. We as women, how we are different. I'll come back to that, but first I want to say, like Dr. Kohad said in the morning, research, innovation and entrepreneurship is shaping the economy of the country. So women, startups and founders, they have increased from 17% to about 26% in the last 1 decade. So if you talk about women in the topmost positions, this is happening. And I would want to say to all the students who are present here, this is the best time to be in India. A startup ecosystem, innovation and research is so conducive for you to do a tech transfer and form a commercially viable product and earn from it. As a woman, I've been working and developing others' portfolio, others' company, others' enterprise. So with all the opportunities that are available, I won't say only to women, to each one of us as citizens of India, and with special focus on women-centric proposals and grants that are being given by Government of India. Please use the innovation and entrepreneur system to grow yourself. Like, I switched myself from public health to being an entrepreneur myself, and my Ikigai being serving the underserved. So I have used self-help groups and farmers as a production unit, if I may say, for startups. That's the value chain. I close the loop because farmers and self-help groups find it difficult to take their product to the market. So marrying them to a startup help push their produce to national and international markets. So as a woman, I've been able to connect the dots, find the talent, and help everybody thrive. So एक बात बोलना चाहूँगी, जो मेरे mentors और teachers यहाँ बैठे हैं, प्रोफेसर रूपलाल, प्रोफेसर कोहार्ड, प्रोफेसर अमूल्या पांडा, इन लोगों ने ना हमें पंख दिए उड़ने को, बट सबसे पूछा जाता है कि हर कोई क्यों नहीं उड़ पाता. क्या आपने अपना इनर पोटेंशियल समझा? सबके अंदर वो चाह है उड़ने की, जिसने अपने पंख फैला लिए. The sky is the limit. So all those students out there, you have the potential with you. This is the best time to be in India. Grants and funding are lined up. Take use of the opportunity. ये मत आके बोलना हमें पता ही नहीं है, तो मैं बोलूँगी आपने ढूंढा ही नहीं है. Thank you.
Rightly said. I think I always wondered that why do we coin this as a woman entrepreneur? We never tag as a male entrepreneur. I think this gender disparity is diminishing in a big way. And today, it's not about whether it's a female-led founder or a man starting a startup. It's all about the problem statement, the unmet need, the talent, the team that you are building that is more important. So girls and boys, the time is ripe for innovation, entrepreneurship. If you have an idea, please seek mentorship. There are a lot of enabling ecosystem around you. Don't feel embarrassed that it's a very easy question, why should I ask? Just go, there are a lot of mentors sitting right away here, and ask for the help, and definitely there is no looking back. Thank you, Dr. Shikha. The second question that you have worked in very difficult terrains. You have worked in Orissa, you have been in Orissa, you have been in Northeast. So how do you envisage, like, is startup or innovation the buzzword of bigger cities like Delhi, Hyderabad or Bangalore, or now tier 2 cities like Bhubaneswar or Dehradun are slowly and strongly carving the niche in innovation ecosystem? So does geographical boundaries define innovation and technology? What is your view on that?
So that's a very interesting question. As a woman, we are always being told, आप travel बहुत करते हो, आप काम दूर दूर जाके बहुत लेते हो, तो घर कैसे चलता है?
I can relate.
So be empowered enough, empower your family enough, empower your children enough, and live guilt free. This is what I want to say. सब कुछ हो जाता है. आप जितना घर पे हो उतना आपका need होता है. आप नहीं हो तो पीछे सब कर लेते हैं. So having an empowered family really helps. And I would want to tell you something. Northeast और उड़ीसा और ये terrains में लोग जाना नहीं चाहते हैं. But my strength has been go where others refuse to go. So these are barren areas where I've built my own roads. I have been to remotest parts of Tripura जहाँ roads भी नहीं है. So, you know, if you want to break the barriers, create your own roads. अगर बनी हुई roads पे चलते रहोगे तो common रह जाओगे. When it rains, the eagle always soars above the clouds. बाकी सब बारिश से छुपते हैं. और हम में तो इतना जुनून जज्बा है कि जितना दूर जाने को बोलोगे उतना दूर जाएंगे. जहाँ कोई नहीं जाता, जहाँ किसी के पैरों के निशान नहीं है, वहाँ जाके निशान छोड़ के आओ, तभी इम्पैक्ट आएगा. उड़ीसा में मैं फुलबानी में इतना रिमोट एरिया में गई हूँ जहाँ मुझे बहुत खुशी होती है कि फ़ोन और इंटरनेट नहीं चलता. तो आप सारी ज़िंदगी गैजेट से क्यों जुड़े रहना चाहते हो? वहाँ जाओ, लोकल से समझो. एक लोकल इंटेलिजेंस होती है. अगर आप लोकल्स में लोकल चीज़ें प्रमोट करो, जैसे उड़ीसा में ट्राइबल आर्ट है, उनका जो फूड हैबिट्स है उन्हीं को प्रमोट करके आगे लेके जाओ। और हमने उसमें उड़ीसा ट्राइबल टेक में एआई और कैमरा विजन भी डाला, क्योंकि जो वो पेंटिंग्स बना रहे थे वो यूएस में बैठे लोग भी देख पा रहे थे और उनको वहाँ से भी सेल्स मिल रहे थे। सो इज़न्ट दैट पावरफुल नमिता? अगर बोलना कि मैं ट्रैवल नहीं कर पा रही तो एआई और ऑटोमेशन यूज़ कर लेते हैं, ट्राइबल टेक को भी हम ऐसे प्रमोट कर पाए। उनकी पेंटिंग्स यूएस में लोग खरीद रहे थे। तो अगर आप सोचना चाहो, आप अपने आप से लिमिटेड हो। अगर अपने माइंड को कंट्रोल कर लो, एक डॉक्टर मृत्युंजय ने बोला एक माइंडसेट होता है। यू कैन बी अ विनर इफ यू हैव अ विनिंग माइंडसेट। गो एंड कॉन्कर द वर्ल्ड एवरीवन। इट इज स्पेशली फॉर यंग पीपल। 1 घंटा इंस्टाग्राम पे नहीं बैठोगे तो 1 घंटा फ्रेंड्स के साथ चाय और कॉफी पे बिताओ। ह्यूमन कनेक्ट लाओ, नेटवर्क लाओ, ideas thrive karo. You are there to change the future of the country and we have full faith in you.
Absolutely, yes. I will give an example. When I have done my master's, I went to Banaras. The first question that they asked me, you are from Orissa, how can you speak English? So that was the impression that we had given of a state. So I think, please, it's high time. But we always give an excuse that down south there is some person, there is a lot of opportunities over there. But I think opportunities are everywhere. It's just about the changing the mindset that is so important. It's from local to global, from national to global. That is the spirit that we have to carry on forward. Now, time is short, so we have to be a little bit more quicker and precise with the answers. Dr. Vinita, we have always seen that when we bring a technology from a research lab and bring it to industry, there are a lot of gaps. There are a lot of gaps and challenges because the TRL level which an academia claims is way behind of the technology, of the industry expectations. So what are the very important challenges that happens in the technology transfer?
Thank you, Namrata, for this question. And believe me, every researcher in a lab, they face this situation very commonly, very frequently, because industry wants to have the ready technology. They expect this has to be ready so that they can, go and scale up. But it doesn't happen in that way, and it doesn't work in that way, because as a researcher as well as a scientist, there is a thing, we are able to have the technology ready at a TRL 3, maximum at TRL 4. And after that, of course, we need a industry connect. So industry does not want to put their fund to, because they don't have a confidence at the very early stage technology. So they little hesitate to bring their fund. So that's where what we have done in BIREC, we have one program which we call it as contract research scheme. So with the help of this program, if industry feel this technology is good, but we will not be able to invest in that technology, but the researcher also feel, okay, somehow we are getting a fund from government organization and industry is willing to take it, let's gets validated with the help of the industry. So yes, this is the biggest challenge where, a kind of collaboration because of the early stage technology has become a very important challenge, and they are not able to transfer it, or they are not able to collaborate it. So if they are able to collaborate it with the help of the funding received from the government organization or any organization who are willing to put their fund, they will definitely collaborate. So yeah, TRL is very important, which Dr. Mrityunjay talked about in his presentation. So we have to be aware of the technology readiness level.
So my next question to you is, again, there's a lot of disparity among how do you see IP in terms from the academic perspective and industry perspective. Academia look into more of ownership, while the industry looks after commercialization. So how do you navigate that and bring it on the same page?
So basically what happens in BIREC, we also receive a collaborative projects. So we want to ensure that like any other funding organization, I mean, unlike like DST, CSIR, we do not keep IP ownership with BIREC. So it's up to the academic, scientist as well as the industry, how they mutually, collaborate on that part. So it depends, of course, on the contribution by the industry, by the academic institution. If it is already owned by the company or owned by the academia, so as such we don't see any problem. But of course, they will sign a tech transfer agreement. They will submit to, they will have the arrangements how this technology transfer with the help of the agreement will follow. But if they don't have and they are still discussing, yeah, most of the time because of any clause, of course, on the modification or the improvement of the technology, that's where, the industry and researcher, they have a different views on that part. So that's where, of course, we always guide them. Maybe you can have the joint ownership, who is going to do the improvements in the technology. So depending on the contribution, intellectual contribution, it is, we help them to decide, okay, you can have the co-ownership or it can solely belong to the industry. So of course, industry people, they will look from the, they see the technology in terms of the product development, taking it to the market. Of course, they would also like to have the ownership. But from the academia side, because they have developed this technology, they would also like to see the ownership. So that's where sometimes, they have a conflict. But of course, if they discuss about it, it can be resolved.
Yes, please.
Can I just add to it? We are talking about startups, but Dr. Vinita and Dr. Namrata, the new thing which is coming up, because Niti Aayog and Government of India is pushing entrepreneurship from state level universities also. So just wanted to tell the audience that a policy decision at the country level is going to be formed soon regarding faculty and student-led startups and who owns the patent, what will be the university share in it. So state-wise and a national level-wise, the policy is going to get constituted very soon.
Absolutely, that is very important. Until and unless at the policy level there's a reformation, it will take its own time. So Dr. Anvita Gupta, you please tell us about, you have been working in AI. So how is AI now reshaping microbiology research or disease? Because now we see AI everywhere. In many proposals that come to funding agency, I've been in selection committee in a lot of funding boards. I've seen that, many times the startups, they use this AI even though it is not needed. So what is that, current scenario of AI in shaping the microbiology research?
Yeah, it's pretty interesting because AI has almost become a hype word for a lot of people. So they say, oh, we're using AI in this, AI in that. And I'm someone who has worked 10 years in machine learning and AI. I did my bachelor's from Stanford in computer science, my master's there also in computer science. And so you really see the nitty-gritties of AI, and then when you go and ask, so I think for investors or for the government evaluating these startups, I think as soon as you ask them a few questions as to what's the algorithm you're using, what's your data set, how are you evaluating your models, usually anybody who's just using it as a hype will not have the answers to those questions. So that's a pretty easy way to differentiate. But there is a lot of promise out there in AI and microbiology. And I have a talk later this afternoon where we talk about what we're doing. But as an example, we have a long collaboration with Professor Prince Sharma and Professor Nina Kaplash at PU. And we have been using AI to design new antimicrobial peptides for drug-resistant infections. And those peptides, it was really interesting because we designed them, got them synthesized, sent them, and then Professor Nina Kaplash, she said to me that this is the ranking of the peptides. Actually, all three are active, but the ranking of the peptides in the lab matched exactly what we had predicted in using our AI model. And usually that doesn't happen. So that is an example of the stunning level of success that you can get if you actually use AI to its full extent in microbiology. And that collaboration has been immensely successful. We're bringing this to market extremely soon. And so that's one example. Drug discovery, I think, is the big one. So drug discovery in microbiology.
So what are the challenges and limitations of AI? What do you see?
So it's a pretty broad question, I think focusing on microbiology though. The challenge is that AI requires a lot of data. So that's the first thing people think of, and high quality data. So a lot of labs, they have maybe 5, 10 data points on some microorganism. Maybe they have 100 if they've been working for a while, but AI requires on the level of millions of data points, right? But you can be very smart about how you train your algorithms so that you use a lot of data that's available publicly and then just really be very specific and fine-tune on the data sets that come from your collaborators. So one limitation of AI would be the amount of data you need. I think another limitation would be basically the person applying it, if they're skilled enough to know where to apply it or not to apply it. And for that, we need computer scientists to work very closely with actual microbiologists. So because we have the expertise in AI and we work with PU who has the expertise in microbiology, or if we do, previously I've done work at ETH Zurich, there I worked with medicinal chemists, so they have the lab experience. So the AI people need to work very closely with the scientists who are actually there in the lab. So that's what I would say.
Wonderful. Dr. Rakesh, so again, there is a lot of confusion regarding the time frame, how academic research works and how our industry expectations are. Generally, academic research, we say that, okay, it will take its own time for R&D, 4 to 5 years, and industries, they need it fast because a lot of money is being invested. So how to navigate that time frame disparity?
I think this is a wonderful question that you have asked. This is a fundamental difference. Actually, this difference is in our thought process. Institution is more interested into fundamental research, more interested into high quality research, and maybe breaking certain newer paradigms. However, on the other hand, industry is interested into a technology that they can take it to the market. A technology that can be easily transformed into the saleable products. Technology that can give them, ROI. We as an entrepreneur are more concerned about ROIs, return on investment, the money that we put and the profit that we, that we get and we reinvest into the business. So we have these two fundamental disconnects between academia and in the industry. So I think what I have seen over a period of time, now the academia has also realized the industry needs, and industry also realized the limitations of the academia and their thought process. To bridge this gap, what I feel is that I give you an example of Punjab University where they have established their TTOs, this technology transfer offices. That I think is playing a very important role. Dr. Prince Sharma and Dr. Naveen, they are aggressively, actively working on bringing technology to the industry. So what I feel is like, I give you a simple example. This is a platform. Here, all microbiologists across the country, they have come together under one platform. Ma'am, maybe in the next conference, if you gave us 10% time to the industry, let us say you brought 5 entrepreneurs today, right? I would request next time you bring maybe 20 entrepreneurs or 50 entrepreneurs. Give them, if you have, let's say 100 hours time, give us 10 hours time. So we will bring our perspective, we will put across our perspective to you. You listen to us, you listen to our requirements. Our requirements are very simple. One requirement is ROI. Another is how fast I can take that product into the market. And third is how fast I can take more product into the market. And any technology that you offer to us should have a potential in the market, ma'am. So if you can listen to us, if you can listen to our requirements, that will be very helpful. Secondly, building up maybe academia, industry partnerships. You can have more conclaves. Maybe you can have, we can have maybe one annual conclave where industry and academia, they come together. Maybe, when we were kids, we used to have lot of melas So can't we have maybe a technology mela where you can call entrepreneurs? You see, you have a lot of microbiologists, those who have successful business. I mean, I would request that call them, listen to their perspective, give them platform to interact with so great minds that that are there in this hall only. So we will come up with solutions, and that will be good for the industry and for our research fraternities.
Dr. Shikha,
Can I just add, because students are here, and very well said, sir. I just want to add for the students, please don't think in research, innovation and entrepreneurship is for science streams only. It is for management, commerce and other streams also. And when we talk about academia industry or startup industry connect, please go for internships, because we have seen successful tech transfer and commercialization happening if the industry has been mentoring students as interns. They've actually gone to the industrial setups and done immersion visits. So basically the idea which is brewing from them is already pre-vetted by an industry. So look for those opportunities as internship. Ask your university to build a cohort of these industries where you can go and do paid or unpaid internship. This is for students to start asking that they should be given internship in the local industries. Thank you.
Absolutely right. I think students just do not complain about your canteen food. Rather, look forward to such kind of networking where you go to industry, understand what is the industry requirement and make yourself prepared. Because now, I think as a student, this is your gala period. After this gets over, when you go to the real world, that's where the networking, collaboration and everything matters. Dr. Rakesh, my second question, you mentioned about industry, academia working together. And now this is the era of co-development. Even BIRAC have a lot of schemes where industry puts 10% of the money, rest academia puts, and rest BIRAC joins in. So how do an industry perceive such kind of co-development, early stage research? Because sometimes when industry has joined at the beginning, a lot of time and effort is saved. You don't have to reinvent the wheel and you do extra experiments. Rather, you know what is the requirement, what is the taker requirement, and you make yourself prepared for. So how does industry look forward to such co-development initiatives?
Excellent question, ma'am. Industry look forward to this. See, as an industry, if I talk from the industry perspective, we are always hard pressed for funds. So academia, universities, and maybe CSIR institutions, they have wonderful labs, they have wonderful minds. So if we put up our 10% share into them, and we can bring in some some good project forward, is always a welcome idea. However, madam, it comes with a caveat, and the caveat is that we operate in a highly regulated market. That is our major issue. What I mean to say is a highly regulated market is that we are controlled. We are controlled by a lot of regulations, very stringent quality control measures. And if we, let us say, in the early stage development of the product, that okay, we have a maybe preclinical product, that if that product is developed by the lab, we have to take it to the post-clinic stage, maybe phase 1 trial, phase 2 trial, and before commercializing this product. So it's a very long drawn process. And this is the major problem that industry, that particularly the pharmaceutical industry or the healthcare industry faces in India. And that is the reason that most of our industry, they just wait for the patents in US and the other market to expire, get a new process, register the product and sell it into the market. This is how we have built up a generic market. So bringing up these and these are regular. And one more thing I would say, at the ministry level, ma'am, for us, let us say the major contributor to the growth of our industry is biosimilars, or you can say biologicals, right? So the issue is that we are controlled by different agencies, different ministries. For example, Department of Pharmaceuticals. If we say Department of Pharmaceuticals comes under Ministry of Chemical and Fertilizers. And you talk about maybe your Department of Biotechnology, this comes into Science and Technology. However, now all these things, we operate into a pharmaceutical segment, and here we are controlled by a drug act called Drug and Cosmetic Act 1940 and Rules 1945, and the process owner for that is the DCGI of India, who comes under the Ministry of Family and Healthcare. So, madam, these 3-4 different ministries, this is a cocktail of ministries. So one ministry sometimes doesn't understand what the other ministry is doing. What DCGI is doing is not known to maybe what DST or DBT is planning. So these are the major policy problems that we as an entrepreneur face in bringing up technologies to the market, ma'am. So if these things can be, I can give you one more example also. Like we have 3 different armed forces. We have armed forces, we have Navy, and we have Air Forces, and above that we have a CDS. This is called CDS, who actually coordinate between these 3 armed forces. So why can't we have maybe a coordinating body which can coordinate between Ministry of Health, Ministry of Science and Technology, and Ministry of...
you rightly said. There are systems like this, the Science and Technology Cluster, which is from Office of PSA, which I'll be defining. But we have to cut because I have already got some reminders. But you mentioned, right, it is all about collaboration and connect, clarity, trust. These are very important whenever there's a scope of collaboration. Dr. Shivani, with your experience supporting a lot of startups, tell us what are the qualities that differentiates a good startup making business, and we have seen a lot of startups also diminishing at the prototyping stage. So what are those good qualities that a good startup stands out?
So the difference mainly lies in the execution and the adaptability. So many of the startups, they have very good ideas, they have like the brilliant prototypes, but the problem with that, they are just looking for the perfection of their prototype. They are not looking for to understand the market need, what is the market size, like who will be the customer, how they can refine their product to a better level. So if anybody want to be a successful entrepreneur, so they should be focused on these things like the market need, refining of the product depending upon the feedback, whatever they will get from their customers, as well as the main thing is the team building. So if you have a good team, like it will be a plus point for you to execute in this startup journey.
Absolutely. Team building, adaptability is so important. Any success example story that you want to share?
Yeah, so the one example I would like to share here is the Fermentation lab from the IIT Roorkee portfolio. So he's a PhD scholar from the biotech department. So initially he began with his PhD work. So he was working on microbial fermentation to generate sustainable enzymes. So we have supported him with the different funding support. We have providing them infrastructure like the dedicated wet lab, and then mentoring them in the business. And right now they have raised around 5 CR from different sources, including VC funding, and they have established their own pilot plant at the Roorkee campus itself. So that is one of the good example that I would like to share for the microbiologists.
I'm sure there would be many.
Yeah, there are many,
Thank you. Now moving to Dr. Tanvi, can you hear? Am I audible?
Yes, you are audible.
So now you have both a global as well as an Indian perspective. So tell us very quickly, what are the lessons that India need to adopt to strengthen the industry-academic collaboration or to bring microbial research, translational research into commercialization?
Okay.
Any
nice
global
question.
trends that you want to share? Yes.
Yeah, so it's a nice question, but let me start by saying that, this is not an easy question. And I'm not sure why, but there is always a very common belief that Western countries have stronger collaboration between academia, industry, and academia another world, because their academics are more entrepreneurial or their industries are more enthusiastic. Honestly, it works because both sides need each other. But for that to happen, one thing that at least the US universities do well is they understand that to enable companies to come and work with their faculties, the researchers need to be very visible and discoverable to industry. So if there is a technology inside a lab that has even a small chance of industrial relevance, here the universities immediately files their invention disclosures. And that simple step sends a signal to the outside world that there is something happening. From there, faculty, students are encouraged to engage with industry mentors, participate in early customer discovery programs, and etc. So one thing, like in short, and maybe I should add that there are even other several formal business support programs here, accelerators, market validation grants. There are ICOP style, as panelists have been discussing, like access the market very early. That help researchers understand the market before science is even finished. And universities also invest in platforms that automatically connect labs to companies. So, in short, coming back to your question, and honestly, if you ask me what are some of the things that maybe India can adopt in the future, that's one thing is visibility is the first bridge. 100%, if Indian universities, I'm not sure what their level they are in, they have to be public facing. All the technologies, briefs, clear capability summaries that make it easy for a company to understand what a lab can do, that needs to come forward. Also, as I have been listening to the other panelists, creating more spaces where in academy industry naturally cross paths, that is helpful. Not large conferences, annual summits. There could be, like shared pilot plants, maybe inculcating more regional bioprocessing centers, start up more joint testing facilities, incubators, where companies, faculty, students are physically present at the same time. And third, if if that is not being done currently, India should also strengthen programs that would bring market understanding into academic research, as one of the panelists has been just mentioning. So, yeah, that are some of the takeaways.
Thank you so much,
I would
Dr. Tanvi. I think now it is 1:50. I can see the gloomy faces of the students, so we'll not drag it from here onward. But yes, I think one message which is very clear from today's conversation is that when we're talking about Atmanirbhar Bharat, Viksit Bharat, it cannot be the effort of a single institution, of a single entity. Industry, academia, startup, government, everybody have to come together on a unified platform, leveraging the strength and resources of each other, and that's where the new India, the Viksit Bharat will lead to. Thank you so much for sharing your perspective. I hope all the students got a lot of takeaway messages. Thank you so much for your time and sharing your perspective. Jai Hind. Thank you so much. Okay, any students, if you want any questions, please raise your hand. Yes, I see one hand raised up. If organizers can pass on the mic to her. Can somebody please, you can come forward. Yeah, please come forward. Yeah. You can take the mic. Introduce yourself. Yeah.
Hello, ma'am. I'm Ishani. I'm from Uttar Pradesh. And, ma'am, my question is to Shikha, ma'am. Ma'am, it was very lovely and interesting to hear you. It was really, really amazing. And I have got a couple of questions. So I've written it down.
Ask one question. Rest you can ask in the lunchtime. Okay? That's where we have put it as networking lunch.
I'm having lunch with you, Shivani.
Yeah, absolutely. Go ahead with your first question.
Okay, ma'am. So my first question is the obvious one, that how can we join and connect to BIRAC? Like if we want to come out of India or go to somewhere else, then what are the procedures for that? Also, ma'am, the thing is that...
the question is a little bit confusing. What did you ask? How to connect to BIRAC?
Yes, ma'am. Like if we want to join, like what are the best ways? Like are there internship programs or something in which we can enroll?
Okay, okay.
Yes,
I think with Dr. Shikha, Dr. Vinita can also
I can answer
this.
that, it's okay.
Yes.
Yes, ma'am.
So, do you have an entrepreneurship idea? Do you want to form a startup?
Yes, ma'am.
So, it's not only BIRAC, BIRAC is for biotech related innovations, but you have to look for open call for proposals. BIRAC has lot of grants open, BIG is open currently, that gives a 50 lakh funding. It's not about internship, it's about getting your idea funded. Lot of government opportunities are there, UP startup policy is there. You would want to get incubated at one of the incubators. Where are you in Lucknow?
Okay.
Where are you in, in Uttar Pradesh?
Uttar Pradesh, I am from Jaunpur.
Jaunpur. So Jaunpur may not have an incubation system there, but you can actually discuss if IIT Roorkee can help you. So you can get incubated any part of the country. You can also get incubated in KIT, TBI, Bhubaneswar. You can also, yes, a lot of options are there. DST grants are there. BIRAC is funding a lot of initiatives. So depending on the stage of your growth, depending on your technology readiness level. I hope you heard what Dr. Mrityunjay talked about technology readiness level. So look for those grants which are open, and there are certain grants which are open throughout the year. Right? So see the idea which you want to nurture and take it forward. We also have special sessions on open grants. We tell you also how to get mentorship, how to actually write those proposals also. So get connected, seek out help, and I'll tell you more over lunch also.
So girl, just one thing I advise that I want to add on. Just jump into startup, because now it's a fab word. Okay, understand whether that's your cup of tea or not. So there are enabling ecosystems like TBIs. You can approach any technology business incubator. You don't have to go there. Just virtually connect with them. Understand about how BIRAC functions, how the incubator functions, what are the different schemes, what are the pros and cons of starting a startup, attend some capacity building workshops, and then take a decision. That will be the right way, to have an understanding and then get into one stream. Okay, I'll be happy to be connected to you and share my details. And Dr. Vinita is also there from BIRAC, so probably in the lunchtime we can talk to her. Okay?
Thank you so much, ma'am.
Thank you so much. If there are no questions, yes, ma'am. Yes,
she's going. Okay.
Yes, ma'am.
So from the BIRAC side, I can mention there are a couple of grants which is meant for the students specifically. One is the EUVA Fellow Program. So where the undergraduate and postgraduate students, they can apply, and they will get a chance to get the mentoring from the EUVA centers. They give you a exposure to pre-incubation, which Dr. Namrata just mentioned. At least you will get to know how incubation system works, how entrepreneurship, journey is like. So that will give you, or that will expose you to the journey of the entrepreneurship, even from the undergraduation. So there are a couple of programs, so which I will be, I have a presentation after lunch, so I'll speak about what BIRAC, doing for the students as well as for the startups.
Don't miss out the session, right?
Sure, ma'am.
Thank you for asking this question.
Thank you, ma'am.
If there are no questions, we are breaking. Again, I see a hand raised.
Good morning.
The last one, very quick, okay?
Good morning, ma'am.
Yes.
Thank you for giving this opportunity. Ma'am, as we know that there are many youngsters who leave the field of research or innovation just because of the cost. As we know that for a research or innovation, there is a lot of money required, and it is just because that India imports its machineries or devices from others country, which makes youth or youngster to go back from the research field. How can we come out of this problem?
Let me tell you, young boy, there is no dearth of funding today. There is lot of funding schemes across different ministry, be it BIRAC, DST. If you have a good idea, if you're really interested to pursue research, start a startup, or just do research and then go for technology transfer, then there is never looking back. Okay? Have a good mindset, build a good team, pick up a right problem statement, and there are a lot of enablers to support you.
Can I also add something really quick to that?
Yes,
And
please,
quick.
Dr. Anvita.
I think, so I've seen labs all over the world, the US, Europe, Switzerland, and a lot in India. And I would like to add that the infrastructure and labs in India, in many places, especially in the incubation centers, is better than what you see in Europe or in the US. And that is available to you for very, very low cost as a student here. So I would say take advantage of that. You know, don't think that just because it's in India, it's in my backyard, that it's not good enough. It's actually equivalent to the international infrastructure. So have that mindset that you can take advantage of that, and you're in a young, growing country, and that's a huge advantage that you have over the entire rest of the globe.
And also the greatest advantage that the money that you will get will go to your account, right? You will be the boss of your idea. It doesn't go to the institute. So you can buy your equipments, you can utilize the equipments that is there in the incubator. There's a lot of freedom.
Thank you.
Yes, thank you. So we'll break down here. Thank you everybody. Thank you to all the panel members for sharing the perspective. It was a wonderful interactive discussion that we had. Thank you so much.
Thank you, everyone. Now I request Professor Prince Sharma, the President of AMI, to please join us on dais to felicitate a token of gratitude to all the panelists and the moderator. It was truly a beautiful session by all the panelists, sharing a beautiful thoughts for all the youngsters being sitting here. And thank you to our moderator, Namita ma'am, for being there for us for today.
Congratulations to all the panelists. It's very heartening to see that out of 7 people, 6 are women here, and this is really great. And 3 of them, they are my students actually, who have done work with me, and it's I'm very proud that they are doing so well. One is in business, is doing such a great business. One is in US, and the third one is, I think she's at night, she's still awake, Tanvi, and I thank you, Tanvi, to you that we have woken you till late, and they have all joined. It's really good to have such a discussion. It's very good discussion for all the students. Thank you so much to all the panelists to come and you have come and joined this AMI session for this very particular, very important session. Thank you so much.
Thank you, sir. I'm starting with the first panelist, Dr. Shivani Sharma. Sir, I request you and ma'am, please join us in front for the felicitation. A token of respect and gratitude on the behalf of Dev Bhoomi Uttarakhand University and AMI. Can we have urgent applause for one of the panelists for this today's panel session? At the back side, already mentioned by cohorts in the morning, clap is one of the most essential things that you need to be have with you. Now I request Dr. Anvita Gupta to please join us from the front. And thank you for being one of the panelists for today's panel discussion. So it's next requires the next panelist, Dr. Shikha Dhawan. Thank you, ma'am, for sharing the insightful thoughts with us. And truly, I'm telling you, it was so energetic to listen to you, ma'am. It is. Can we have a huge applause for another panelist, Dr. Shikha Dhawan? Boys and girls at the back, there should be no stop in your clap. That will help you to actually boost up your body itself. Next, I request the another panelist, Dr. Rakesh Vig. And our next panelist, Dr. Vinita Jindal ma'am. Thank you for being one of the panelists for today's session. Now it is a time to felicitate a token of respect and gratitude to our moderator for this session, Dr. Namita Mishra. Thank you, ma'am, for sharing such insightful thoughts and being a moderator for this beautiful session. This cap is a respect and gratitude from the Uttarakhand region. It is a traditional cap of Uttarakhand. And it carries a Brahm Kamal on the top, which is the national flower of our region. Can we have all the panelists together to have a one group photograph, please? By the time after this photograph session, we'll be having a lunch for all the students at the SOHMT outside in the Pandal, and for the faculties in the Ayurveda area, and for all the delegates and the speakers in the SOHMT restaurant, the hotel management department. And we'll be back here in next 40 minutes for the next session. So I request everyone at the back, even the students as well, please join us back in next 40 minutes. It's a time right now, it's 2:05, and I request everyone to be here by 2:45 sharp. And I would like to say a thank you to Dr. Tanvi Govil as well, who joined us from US online for today's session. Thank you, everyone. Micro pitch ideas that inspire will be happening at main Pandal at 2:45. Computational microbiology, data sciences and digital health at Seminar Hall, first floor, engineering building at 2:45. Session 4, AI in microbiology research, bioprocesses and bioeconomy at Ayurveda Seminar Hall at 2:45. So the respective people and the speakers delegates can join their sessions in their respective places. With this, I request everyone to kindly proceed for the lunch at their respective areas where I have announced before, and will be joining back sharp at 2:45 for the next session. Thank you.
Hello. Check. Check. Hello. Hello.
All the volunteers at the back side, kindly request all the students who are standing at the outside, kindly get them inside because we are about to start with the session further. पहले इनवाइटी वाला देना है, फिर साथ साथ ही चेयर वाला। सारे उसी में लगे हुए हैं। हाँ। So good afternoon, welcome, and once again to the day one of International Conference. We'll be starting with another session that is Micro Pitch Ideas that Inspire. The theme Micro Pitch Idea that Inspire, focusing on encouraging innovators and aspiring entrepreneurs to present their creative ideas in a concise and impactful way. It emphasizes innovation, originality, and feasibility, allowing participants to pitch their concepts within the limited time to capture the attention of mentors, investors, and experts. The session aims to inspire young minds, promote entrepreneurial thinking, and transform innovative ideas into the actionable solution that can create social and economical impact. It is a time to introduce the chair for this session, Professor Shikha Dhawan. Professor Shikha Dhawan, with a 22 plus years of experience in the health research and public health, Dr. Shikha Dhawan holds a postdoctorate in biomedical research USA and a PhD in microbiology and PGDM in biotechnology business management India. She is the founder of Bonanzans Creating Bioenzymes as a sustainable, eco-friendly solutions for diverse industry. An entrepreneur cafe, eCafe, she drives a global startup success by connecting entrepreneurs with investors and enabling transformative business growth in a partnership with the Foundation of Innovation and Technology Transfer, FITT, IIT Delhi. She is a leading Niti Aayog national study on the research, innovation, and entrepreneurship in 108 states, public universities across 18 states, shaping India's higher education and innovation landscape with a lasting impact. With this, I invite our chair, Professor Dr. Shikha Dhawan ma'am, as a chair for this session. It is a time to introduce the co-chair of the session, Professor Vinita Jindal ma'am. Professor Vinita Jindal ma'am is a distinguished biotechnologist and science administrative, currently serving as a senior scientist at the Biotechnology Industry Research Assistance Council, BRRAC, a public sector entrepreneurship under the Department of Biotechnology, Government of India. With this, I would invite the co-chair on the dais for this session. Now over to the chair for the future.
Thank you so much for the kind introduction. Welcome my co-chair, Dr. Vinita also. So moving forward, we'll have 3 sessions today. Dr. Vinita, Dr. Azam Ali Khan, and I'll have a session myself here. So moving forward, I would want to invite Dr. Azam Ali Khan. He is Chief Executive Officer, TIDE, IIT Roorkee, and his forte is Technology Innovation and Development of Entrepreneurship Society at IIT Roorkee. May I please invite Dr. Azam Ali Khan to give his talk.
Very good afternoon, everybody, and thank you very much, Dr. Shikha and Dr. Vanita. It's been a pleasure to hear both of you in the panel discussion, and thank you very much, Dr. Vanita. We interact a lot with BIRAC, and we are running multiple programs. So, I mean, I must take this opportunity to thank BIRAC for creating an ecosystem for life science startups, which was, I believe, missing before BIRAC era. So, students, I hope everybody is now full. I'll keep my talk pretty short. I've been asked to speak about what exactly is needed or what could be a winning ingredients for the pitching of ideas to a VC. You know, everybody talks about VC. But before VC, there are also several other stages. VC is not a panacea of all your problems. Rather, he or she is an added problem. Because if a VC is sitting on your cap table, to a certain extent, you have to give up your own dreams of the reasons why you started entrepreneurship. But what exactly are we looking for a pitch? What is a pitch? A pitch is merely, you can say, a presentation a PDF in PowerPoint format, where you try to consolidate your ideas, encapsulate them in maybe 10 to 12 slides. They are typical hygiene standards for any presentation. You don't have to basically crowd it with 200, 300 words. Please understand that your pitch is not a paper which you are presenting. You don't have to show your technical skills only in the pitch. Technical skills are definitely important. But other than that, that pitch needs to be attractive. When you're making the pitch, or if I take the example of IIT Roorkee, when we are assessing a pitch, we are majorly seeing two ingredients. One, what is the market you are trying to address? And then there are sub-segments to that. People will call about some jargons, TAM, SAM, SOM, blah, blah, blah. But you just want to understand and show, are you solving a problem which is big enough to solve? And second thing, whether the problem is good enough to solve. So when we say market, we are just trying to assess that. The second thing which we are looking into in a page is who is solving the problem. What is the background of the founders? How well do they know their product idea? How much invested are these founders into the idea? Is it an off-the-cuff, lifestyle-based kind of idea, which you came on to pitch just because everybody else is pitching and everybody else is talking about startups? So you thought that probably you should also have this suffix to your name, that you are a co-founder? Or is it that you have the necessary technical as well as the commercial skill sets? When we, going through the panel discussion, I think somebody spoke about that your idea needs to be commercially viable. You need to have a commercially viable product / service / technology. Now, we need to understand what exactly does that mean. When we say commercial, we are talking about money. There must be a sufficient number of threshold individuals or entities who should be ready to buy your product in lieu of some money. That is what exactly we mean by commercial. The second word is viable. Viable means it needs to be practical enough in the real world to pull off. For example, we know there is a process of photosynthesis which happens almost every time in a leaf. But it is not possible to conduct a process of photosynthesis in a real life situation, otherwise we would have solved the problem of food in the world. So the idea is good, but it is not viable thermodynamically. So when we are talking about creating a pitch, please understand that you are not selling your research work. You are not selling your prowess as a researcher, but you are selling your prowess as an entrepreneur. We are interested in looking into deep tech ideas, deep tech solutions, but the solutions which can solve some genuine problem. So in all your 9 to 10 slides of pitch, please do not stray away from this notion that your idea should make money for the company, for you, and also for the investor whom you are pitching to. Now you have to make this thing extremely attractive for them. And you can make this thing attractive only by using as less jargons as possible. I once went into, there is a world-renowned startup conference which happens in Helsinki, and it is going to happen next week, called Slush. And in Slush, what they did, there were 3 foundations, Honda Foundations, Bill and Melinda Gates Foundation, and one more. And they had a first prize of 100,000 euros. And this prize was actually reserved for researchers. They called it researchpreneurs. What was the process to win this 100,000 euros? Every researcher has to come upstage. Now, this is one of the biggest life scare for a researcher, to come upstage and speak, because we are not training our researchers for that. Every researcher has to come upstage and talk about his or her research in 2 minutes. And that too in a way that somebody who is not from the relevant background must also understand what exactly you are trying to do. There were people from engineering background as a PhDs, there were people from biotech, but the biggest challenge was how to present your jargons in the most lucid fashion. I have seen many professors, if I give them a topic of their research, they will speak like hours, and they will not feel it because they know the ins and outs of their research work like anything. But the moment you ask them that, okay, try to improvise and consolidate it in 2 minutes so that I, who is not a background from a relevant field, can understand, they will be in a soup, most of them. And the problem is not in an individual, the problem is that the way we teach. So when you are making a pitch, please ensure that you are pitching for a commercial activity, you are not pitching for a research grant. Try to be concise. The hygiene which I was talking about, do not use more than 4 to 5 sentences in one slide. Do not use more than 4 to 5 words in every sentence. Now you would say that, oh, it is very easy. The problem is this is the most toughest part. Because when you have to choose your words so carefully and so delicately, it requires a lot of hard work. So 10 slides, 4 to 5 sentences, say for example 5 sentences every slide. So we are talking about 50 sentences, 5 words every slide. So we are talking about 250 words. And make it more graphic. And by graphic, I do not mean that you add all the graphs which you have generated in the course of your PhD. Please don't do that. Because, I mean, to a large number of people who are assessing this pitch, they will be dumbstruck. What exactly does this mean? How will I make money out of it? There are issues with the biotech or life science startups, even more than the typical engineering side startups, because they require or they deal with life. Simple. And if you are dealing with life, whether it is health tech, med tech, or something of the relevant field, you have to be extra cautious. I may not be that cautious if I am developing a solution for solar energy, but if I am developing a solution for checking my blood glucose, I need to be 110% sure the number which is coming up is good or not. Good in the sense accurate or not. And that is the reason the gestation period of developing a startup in life science is more than a typical non-life science startup. And so what typically is proposed or recommended is like, for example, Mrityunjay also was talking about, for the TRL level, and we generally talk in terms of TRL levels, right? This word has gained currency of the last 6-7 years. The technology readiness level. The TRL levels for 1, 2, 3, or 4, that is your growing up phase. And you try to get the grants which is possible, typically the grants from the government. But there are few grants which are there from TRL 4 to TRL 7. And that in terms of a typical deep tech life science startup is called the valley of death. And that is where typically you need to go for startups or for private funding. And my humble submission, my humble request to you is, please do not get into the trap of grants. Grants may be available, but they are not adding enough value because they are not adding market validation and customer validation to your idea. You use the grant to have some MVP, the minimum viable product or the prototype, and then start looking for private funding. It is the toughest thing to get private funding for life sciences startups, or for that matter, even any deep tech startup. But the kind of validation which you will get from private funding and the kind of push the private investor will give to you will be way better than any grant for that matter. So I think this in a nutshell, if I have enough time, I think I've already spoken 5 minutes, 4 minutes. So when you're going for a deep tech startup, there are typically few ingredients which you should take care of. A deep tech startup typically in India is seen to be somebody who is either developing an algo, if you're talking about SaaS-based startups, deep algo, whether it is DL, deep learning, machine learning, artificial intelligence, IoT, sensors, quantum, or the idea which you are proposing has some defendability. And when I say defendability, defendability in terms of patents, so that you can at least create a wall between you and your nearest competitor. Third thing which I would like to share with you, I mean, there are a lot of students over here, and they must be thinking that is it a mandatory thing to do a startup? No. Startup is not meant for everybody. It is not mandatory for you to do a startup and only then you can succeed in the world. It is only one of the way. It is definitely a way which can give you a lot of self-enrichment. It will give you a lot of satisfaction to yourself because you are creating an impact into the life of so many other people. More so, you are earning money. You are a breadwinner not just for yourself, but also for the people who are working for you. It will definitely give you a lot of satisfaction. But please don't have this notion that it is mandatory for all. You need to see the pitfalls before entering into it. The picture is not at all very rosy. It has its own share of thorns. Be aware about those thorns. Get into an incubation system. Ask people. Get mentors on board. And then I think probably if you have a good idea, an idea which can be worked upon, you have a long way to go. Yes, I got it. So it's a long journey, and I think I've just tried to share a couple of my experiences with you. But thank you very much, and I hope that you can take a bit of what I have spoken in the last 10 minutes. Thank you. Thank you very much.
Thank you, Dr. Azam Ali Khan. And with the passion you spoke, it really shows you have ears to the ground, the way you have been mentoring startups, and we can actually make out how you are developing India towards a Viksit Atmanirbhar Bharat, and wonderfully doing this at TIDES IIT Roorkee. So big round of applause for Dr. Azam and for his brilliant ideas for entrepreneurship, and especially for students who are trying to inculcate a culture to generate jobs and not to seek jobs. Dr. Azam, I would request you to be on the stage, and I would request Dr. Vinita, my dear honey and happy cohort, to please come on the stage and facilitate Dr. Azam.
I request Dr. Shikha Dhawan, the chair for this session, and Dr. Vinita Jindal ma'am as well, the co-chair for this session, to kindly join us for sharing the token of respect and gratitude to Professor Azam Ali Khan for sharing a valuable thoughts on how we can have a startups for all the students being sitting here. Can we have a huge applause for this beautiful session by him? Every time I need to make you remember that clap is highly important. Thank you, sir. Thank you, ma'am. Now it is a time to listen from the eminent speaker. Once again, I request Professor Shikha Dhawan ma'am to kindly share his valuable thoughts with us.
Thank you so much, Shubhang, and always a pleasure to speak. And I am somehow not fond of being at the dais, and Perry told me he cannot see me from the front. He had to go and sit behind. So is it okay if I steal the whole stage? Because I cannot get confined behind a dais.
Would be really great, ma'am.
So, ladies and gentlemen, always a pleasure to be speaking at AMI. And I want to tell a secret, because Honey and Happy were there with me, why I specifically called them on stage. We hosted the 44th AMI at University of Delhi. And this is after 22 years I have met them. They both have grown into beautiful individuals. So wanted them to share stage with them. And that time, Dr. Kuhad had full confidence in me. Like I said, they are mentors, they are advisors who have given you wings to fly. So he is one of them. A big round of applause to Dr. Kuhad. Because what I'm going to talk about is valorization of lignin. That is about value addition in lignin. But I want to quote my own value addition from a student doing PhD in microbiology, working on lignin degradation. उसका भी एक story है. Biochemistry में lignin degradation हमें पढ़ाते थे, and I used to shrug from that topic because the pathway was very complex. वो कहते हैं ना डर के बाद जीत है. I ended up doing PhD in the topic जो मुझे graduation level में और post graduation में बहुत difficult लगता था. So this is for all of you students out there. डर के आगे जीत है. Face your fears. There's nothing like fear if you ask me. मैंने तो जिस topic से सबसे ज़्यादा डर लगता था उसमें PhD कर डाली. So thank you all for being a wonderful audience. And I want to tell one more thing. 44th conference tha, so I was doing what Shubhang is doing. So it's an honor to see myself, if I can say, and thank you to Namita for recognizing that, and thank you to Nirjara for making me host this session impromptu. Always there, always present, always a pleasure to be with all of you. Now can you please have my slide? नहीं तो मैं बिना slide के ही बोलती जाऊंगी, which I can do very well. But अगर slide दिखा देंगे तो मैं talk structure हो जाएगा. Okay, so the slide, the session which I'm going to talk about, because I work with entrepreneurs. At Association of Microbiologists of India, we want to talk about research, innovation and its translation to business, which my dear friend Vinita looks after very ably at BioRack.
सो विनीता को वो स्टार्टअप्स अच्छे लगते हैं जो पेटेंट्स, इंटेलेक्चुअल प्रॉपर्टी, ट्रेडमार्क, कॉपीराइट और कमर्शियलाइजेशन की बात करते हैं. बट मैं आज अपना सेशन, क्योंकि बच्चे यहाँ बैठे हैं, हम लोग एंटरप्रेन्योर माइंडसेट की बात करना चाहते हैं. डॉक्टर शिखा यहाँ बैठी है जो बहुत अच्छा रिसर्च कर रही है लिग्निन वैलोराइजेशन में. तो मैं आप सबको बताना चाहूँगी कि एक रिसर्च और इनोवेशन आइडिया इन वैल्यू एडिशन ऑफ लिग्निन, आप उसको एक बिजनेस आइडिया में कैसे लेके जा सकते हो. सो दैट इज द स्ट्रेंथ वी वांट टू ब्रिंग टुडे, बिकॉज यू आर द फ्यूचर ऑफ द कंट्री. हम लोग आपको क्लासरूम में एकेडमिक सेशन दे सकते हैं, बट हाउ टू इनकलकेट अ कल्चर ऑफ टेकिंग रिसर्च टू एन एंटरप्राइज, दैट इज माई जॉब टुडे, स्टैंडिंग देयर चेयरिंग दिस सेशन. इफ आई कैन ब्रिंग दैट आउट इन यू कि एक सिंपल रिसर्च आइडिया को भी कैसे लेके जा सकते हैं आगे. आप में से कौन कौन जानता है कि लिग्निन क्या है? मैंने तो पीएचडी कर रखी है. लिग्निन क्या क्या है बताइए. लिग्निन क्या है? इज द मोस्ट abundant biopolymer which is renewable on earth and gives firmness to plants. Cellulose is the most abundant polymer और उसके बाद lignin आता है which gives sturdiness to plants. Normally when we talk about paper making, उसमें lignin को as a waste निकाल दिया जाता है क्योंकि आपको cellulose fiber चाहिए होता है पेपर बनाने के लिए. Conventionally in pulp and paper industry, lignin was seen as a nuisance. But when I started doing my PhD with Dr. Kohad, I realized कि lignin को भी आप value addition करके बहुत चीज़ें enhance कर सकते हैं. ये लोग अभी भी मेरी स्लाइड ढूंढ नहीं पाए हैं, बट मैं उसके बिना भी चल सकती हूँ. सो बात ये है कि बायोकेमिस्ट्री पाथवे में आई कैन टॉक टू यू अबाउट हाउ यू हार्नेस दिस abundant renewable source of nature into a startup idea. क्योंकि पल्प एंड पेपर इंडस्ट्री में इसको वेस्ट माना जाता था, बट नाउ वी कैन डू वैल्यू एडिशन टू लिग्निन and this would form the basis of lot of new business ventures, right? So आप lignin को अगर plant fiber से release करना है, depolymerization जिसको कहते हैं, तो आप lignin modifying enzymes use कर सकते हो. Lignin peroxidase हो गया, lacases हो गया. My PhD is in lacase from Pycnoporus cinnabarinus. Lignin जब usable form में आ जाता है तो आप उसको वैल्यू एडिशन कैसे कर सकते हो. स्मॉल मॉलिक्यूलर कंपाउंड्स रिलीज होते हैं जिसको आप, डॉक्टर शिखा ने मुझे बोला वो केमिकल ट्रीटमेंट करते हैं. जो मैं स्टार्टअप आइडिया बोल रही हूँ वो भी केमिकल ट्रीटमेंट कर रहा है, एल्कली ट्रीटमेंट कर रहा है, लेकिन उसके साथ साथ मैंने डॉक्टर कोहाट के साथ एक चीज़ सीखा था, स्टीम एक्सप्लोजन. तो वो हम एक फिजिकल मैकेनिकल मेथड उस स्टार्टअप आइडिया में लाए क्योंकि आपको केमिकल यूजेज भी कम करना है, बिकॉज टुडे वी ऑल्सो वांट टू केयर फॉर द एनवायरमेंट. ओके, सो मूविंग फर्दर, इफ यू यूज लिग्निन मॉडिफाइंग एंजाइम्स, आप मोनोमर्स रिलीज कर सकते हैं. दीज कुड बी फंगल, बैक्टीरियल, एक्टिनोमाइसीट्स, जिससे वैल्यू एडेड प्रोडक्ट्स बनेगा. These products will enhance the value of lignin per se because अब जो प्रोडक्ट बनेगा कन्वर्ट होके उसका बायोमेडिकल यूज है, बायोप्लास्टिक में यूज है, कॉस्मेटिक इंडस्ट्री में यूज है, बायोफ्यूल में यूज है. अब आप सोचेंगे इतना कमाल का मॉलिक्यूल क्या है, क्योंकि पल्प एंड पेपर में तो उसको वेस्ट समझते हैं. सो वैनिलिन निकल सकता है. इट्स एरोमेटिक कंपाउंड यूज्ड इन फूड फ्लेवरिंग एंड फ्रेगनेंसेस. Ferulic acid, phenolic compounds, they can be used in resins and coating, polymers, plastic and bio-based chemicals, जिसमें ये lignin को आप value addition कर सकते हैं. They can be also used to make resins, lignosulfonates, fibers. एक नया concept है startup idea का कि आप lignin nanoparticles बना सकते हैं ड्रग डिलीवरी के लिए. एंड बिकॉज ऑफ इट्स प्रॉपर्टीज अमॉर्फस, लिग्निन इज नॉट सॉल्युबल इन वाटर, यूवी ब्लॉकिंग फिल्म्स और कोटिंग भी इससे हो जाता है प्रोडक्ट्स पे. यू ऑल्सो मेक हाइड्रोजेल्स फॉर बायोमेडिकल एंड एग्रीकल्चर एप्लीकेशंस, मॉइस्चर रिटेंशन, स्लो रिलीज फर्टिलाइजर्स. सो कैन यू अप्रिशिएट विद मी कि एक लिग्निन जिसको हम वेस्ट समझते थे लेकिन प्लांट के लिए जान डालता था, सेलुलोज और लिग्निन की वेडिंग में हम लिग्निन को निकाल देते थे, बट अब हम लिग्निन को भी वैल्यू करने लगे हैं. समथिंग लाइक वी से अ वुमेन लेड स्टार्टअप और अ मैन लेड स्टार्टअप. लिग्निन को भी अब इज्जत मिलने लग गई है. उसको वैल्यू ऐड करके हम कमर्शलाइजेशन कर सकते हैं, प्रोडक्ट्स बना सकते हैं जिनकी मार्केट में डिमांड्स है, जो मैं आपको ये बता रही हूँ. सो इसको टेक फॉरवर्ड करते हुए आई कुड हैव टॉक्ड मोर अबाउट चैलेंजेस क्या है, लिग्निन डिग्रेडेशन में चैलेंजेस क्या है, माइक्रोबियल प्रोसेसेस में क्या दिक्कत आ रही है. बट आई थॉट बिकॉज स्टूडेंट्स आर हेयर, एक सिंपल से मॉलिक्यूल को आप एक बिजनेस प्रोस्पेक्ट में कैसे लेके जाना चाहेंगे, मैं वो बताना चाहूँगी. और ये एक स्टार्टअप है मेरा, सर्कुलो. ये लुधियाना बेस्ड है. दे मेक बायोप्लास्टिक्स आउट ऑफ राइस स्ट्रॉ. राइस स्ट्रॉ भी सबसे सॉलिड स्टेट फर्मेंटेशन में डॉक्टर कोहाट के साथ ही सीखा है. तो मुझे बहुत अच्छा लगता है जब स्टार्टअप्स ऐसे आइडियाज़ लेकर आते हैं और मैं उनको बिजनेस प्रोस्पेक्ट्स के लिए आगे जाके ले जा सकती हूँ. अब आपने स्टबल बर्निंग सुना होगा. नॉर्थ वेस्ट इंडिया इज बर्निंग. एग्रीकल्चर रेसिड्यूज बहुत बर्न कर रहे हैं. वी में से पंजाब और हरियाणा में 70% स्टबल बर्निंग रिड्यूस हो गया है, बट फिर भी वो 30% स्टबल बर्निंग से इन्वायरमेंट में बहुत पोल्यूटेंट्स है. सो ये जो स्टार्टअप है इसने 149 मेट्रिक टन्स ऑफ कार्बन डाइऑक्साइड इमिशन ग्लोबली रिड्यूस करा है बाय कन्वर्टिंग एग्री वेस्ट रिच इन लिग्निन इनटू बायोपॉलीमर्स. तो ये फोकस है उसका. बच्चों को समझ आ गया कि स्टार्टअप का आइडिया क्या है. लिग्निन है, बहुत अबंडेंट एग्री रेसिड्यू वेस्ट है, उसको आपने कन्वर्ट करा इनटू बायोपॉलीमर. उससे क्या हुआ, एक एग्री रेसिड्यूज का स्टबल बर्निंग बंद हो गया, आपका कार्बन डाइऑक्साइड रिलीज जो एनवायरनमेंट में था वो बंद हो गया, और ओवरऑल हेल्थ इंप्रूव हो गया बिकॉज ऑफ दिस पार्टिकुलेट मैटर स्टैग्नेटिंग इन द एयर. दिल्ली और एडजॉइनिंग एरियाज का एयर क्वालिटी इंडेक्स भी बहुत गिर गया था. सो आप देखिए एक स्टार्टअप के कंसेप्ट से आप इन्वायरमेंट और हेल्थ को भी कैसे बेनिफिट कर पा रहे हैं, राइट. मूविंग फर्दर, दिस इज द मैप शोइंग पंजाब एंड हरियाणा जहां अभी भी स्टबल बर्निंग हो रहा है, एंड देयर आर स्पोरेडिक न्यूज इन उत्तराखंड, हिमाचल एंड उत्तर प्रदेश ऑल्सो. अब सर्कुलो ने क्या करा, पैडी स्ट्रॉ को लेकर उसको कन्वर्ट करा लो स्पेसिफिक ग्रेविटी हाई स्ट्रेंथ बायोपॉलीमर्स, और उसको इंजेक्शन मोल्डेड करके क्या बनाया, प्लास्टिक ग्रैन्यूल्स बनाए, जिससे आप साइकिल पार्ट्स बना सकते हो, इलेक्ट्रॉनिक गुड्स बना सकते हो और किचन वेयर बना सकते हो. इजंट इट मैजिकल. एक प्लांट रेसिड्यू से आप ये प्लास्टिक्स कैसे बना सकते हो. सो दैट इज द साइंस ऑफ कन्वर्टिंग अ वेस्ट मटीरियल इनटू बायोप्लास्टिक्स. अब बायोप्लास्टिक्स है तो एनवायरनमेंट फ्रेंडली है, डिग्रेडेबल है या कंपोस्टेबल है. उन्होंने पैडी स्ट्रॉ को लिया, पल्वराइज करा, दैट्स अ मैकेनिकल ग्राइंडिंग, पार्टिकल साइज रिड्यूस करा, एल्कलाइन ट्रीटमेंट या स्टीम एक्सप्लोजन करा टू डीपॉलीमराइज लिग्निन. अब लिग्निन बहुत स्ट्रांगली बाउंडेड है सेलुलोज और हेमीसेलुलोज से, तो उसको रिलीज करने के लिए या सॉफ्टन करने के लिए, डीपॉलीमराइज करने के लिए एल्कली ट्रीटमेंट या स्टीम एक्सप्लोजन कर सकते हैं. फिर उसको वॉश करा, ड्राई करा, उसमें एडिटिव्स ब्लेंड करे और ट्विन स्क्रू एक्सट्रूडर के ऊपर लोड करा. अब एक्सट्रूडर क्या करता है, उसको डेफिनेट फॉर्म्स एंड शेप्स में बना देता है, राइट. ऑप्टिमाइज प्रोसेस करा और आप ये देखेंगे एक्सट्रीम राइट पे ये सर्कुलर के ग्रैन्यूल्स है जिसको उन्होंने आगे ब्लेंड करा with research collaboration from CFET और Central Institute of Plastic Engineering and Technology तो देहरादून में ही है, and also LRPM in भुवनेश्वर. उनसे collaboration करके they finalize this process, probe, perform, practice in converting plastics to environment friendly durable products. अब स्टूडेंट्स को ये बताना चाहूँगी कि स्टार्टअप जर्नी भी आसान नहीं था. उन्होंने कम से कम 30+ इंजेक्शन मोल्डेड प्लांट्स विजिट करे प्रूफ ऑफ प्रिंसिपल के लिए, मार्केट फीडबैक लिया, कि मार्केट किस तरह का प्लास्टिक प्रोडक्ट यूज़ करना चाहेगा। मान लो ऑटोमोबाइल इंडस्ट्री में क्या क्या प्लास्टिक की प्रॉपर्टीज़ होने चाहिए, क्योंकि वो एक बायो फ्रेंडली प्लास्टिक बना रहा था, तो उसके लिए रिसर्च करना बहुत ज़रूरी था। उसने अपना प्रोडक्ट ऑप्टिमाइजेशन करा, टेंसाइल स्ट्रेंथ पे वर्क करा। तो प्लास्टिक में इलास्टिसिटी कितना है, ड्यूरेबिलिटी कितना है, स्पेसिफिक ग्रेविटी कितना है, और लाइट वेट लाने के लिए उसने काफ़ी रिसर्च करी, राइट। एंड देन ही सेट अप अ पायलट प्लांट इन लुधियाना, और उसका फर्स्ट कस्टमर था द कैंगरू लैब। कैंगरू लैब किसने सुना है? स्टेपलर्स और स्टेपल पिन बनाता है। उस स्टेपलर में एक ब्लू कलर का पोर्शन होता है प्लास्टिक का। सो ही डिड द पायलट फॉर दैट। अब हम सुबह से सुन रहे हैं कि स्टार्टअप कब सक्सेसफुल होगा, आइडिया कब सक्सेसफुल होगा, जब उस बिजनेस को यूज करने के लिए कोई कस्टमर होगा। तो उसने काम ही ऐसा शुरू करा, जो वहाँ पे स्टार्टअप जो बेस थी कैंगरू स्टेपलर्स जो बनाती थी, लुधियाना में है। यही मैं सुबह बोल रही थी, अगर आपको लोकल एनवायरनमेंट में काम करना है तो लोकल इंडस्ट्रीज देखिए उनकी नीड्स क्या है, ठीक है। आर द स्टूडेंट्स गेटिंग इट? लोकल नीड्स को आप सॉल्व कर रहे हो तो यू विल ऑलवेज बी सक्सेसफुल। तो अपने एरिया ऑफ रेजिडेंस से ज़्यादा दूर भी नहीं जाना पड़ेगा, वहाँ इंडस्ट्रीज है, मैन्युफैक्चरर्स है, जिनका आप प्रॉब्लम सॉल्व कर पाओगे, एंड दे विल ऑल्सो प्रमोट लोकल। ये लुधियाना की स्टार्टअप थी, कंगारू की फैक्ट्री है लुधियाना में, तो उसने स्टार्टअप को प्रमोट करा। सो इन्होंने इंडस्ट्री विजिट्स करी, ओरिजिनल इक्विपमेंट मैन्युफैक्चरर्स है, OEMs जिसको कहते हैं, उनके साथ भी डिस्कशंस करा कि वो प्रोडक्ट में क्या फीचर्स चाहते हैं। सो आप देखिए, जैसे सुबह ये भी बोला गया था, इफ यू मेक द प्रोडक्ट इन डिस्कशन विद द यूजर्स, दैट हेल्प्स यू ओवरकम द वैली ऑफ डेथ ऑल्सो। सो इस स्टार्टअप ने बहुत रिसर्च करा, बहुत इनोवेशन करा, एंड ही हैड लॉट ऑफ रिसर्च कोलैबोरेटर्स एंड इनोवेटिव आइडियाज that finally led to the development of the product which had higher yields. So he could incorporate higher percentage of paddy straw while making his biopolymer. This could not have happened without research, right? So research was the key that his enterprise become successful, and to provide a moat and to protect his intellectual property, he has also filed a provisional patent for it. Right, and the plastic which he produced is lightweight, has high tensile strength for injection molding. अब plastics बनते हैं injection molding से, and he fulfilled the criteria of making them lightweight also, which came from adding unique formulation, additives and composites to his product, which came from the agri waste, जो उसको CFID ने support करा research में develop करने के लिए। Have you all, the students especially, have you understood the value chain? मतलब उसने कैसे प्रोडक्ट को डेवलप करा, एक वेस्ट को लेके रिसर्च करके एक पेटेंट तक कैसे जा पहुंचा, क्योंकि वो एक यूनिक प्रोडक्ट बना पाया। तो उसने ऑलरेडी इसके QC रिपोर्ट्स थे, क्वालिटी पैरामीटर्स थे, so he gathered all of them and he is prevailing because he made the product sustainable. Carbon dioxide emission which was coming from burning from agri residues reduced, and his lightweight plastic, bioplastic products were very well accepted by the automobile industry. And the whole country, the whole world is moving to sustainability. So he found new markets who are willing to take his product forward. Like अभी पिछले स्पीकर ने टैम, सैम और सॉम बोला, ये स्टूडेंट्स के लिए है कि आपका टारगेट मार्केट कितना है, उसमें से आप कितना एड्रेस कर सकते हो और उसमें से कितना एक्चुअली कैप्चर करोगे। दैट मेक्स और मार्स योर स्टार्टअप। सो स्टूडेंट्स के लिए ये जानना बहुत ज़रूरी था कि बायोप्लास्टिक्स का ग्लोबल डिमांड क्या है। उसके बेसिस में उसने अपने कस्टमर्स आइडेंटिफाई करे और अपने प्रोडक्ट को बेटर एंड बेटर बनाया कि वो इन मार्केट्स में, स्पेशली यूरोपियन मार्केट्स में एंटर कर सके। अब एक टर्म बोलते हैं पर्सोना। पर्सोना इज वेयर आर योर कस्टमर्स एंड व्हाट काइंड ऑफ प्रोडक्ट दे नीड। So this is also a part of our mentoring of startups. We tell them to do customer discovery and make personas कि आपके customers कहाँ हैं और किस तरह का product चाहेंगे। So circles ने ढूंढे customers who are mission driven, who have sustainability as a goal, and also people who want to send their products to European markets, क्योंकि वहाँ पे बायोप्लास्टिक्स की डिमांड ज़्यादा है, क्योंकि वो कार्बन एमिशन कम करना चाहते हैं. एंड ही फिट द मार्केट. इमेजिन इन द प्लास्टिक सेक्टर कि वो एक प्लास्टिक एक बायोप्लास्टिक भेज रहा है. सो वेंडर्स हु वांटेड टू हैव अ बेटर कार्बन फुटप्रिंट, दे वांटेड हिज प्रोडक्ट्स. तो आपको ये बताना चाह रही थी उसका मार्केट डिस्कवरी किस तरीके से हुआ. सेकेंड टाइप ऑफ पर्सोना कस्टमर्स का जो उसने ढूंढा वो थे थर्ड पार्टी मैन्युफैक्चरर्स जो मोल्डेड प्लास्टिक कंपोनेंट्स बना रहे थे. सो इफ यू डू योर मार्केट डिस्कवरी, कस्टमर डिस्कवरी एंड फाइंड कस्टमर पर्सोना जिनको प्रोडक्ट चाहिए, देन इट इज़ अ श्योर शॉर्ट रेसिपी टू बी सक्सेसफुल. राइट, गो टू मार्केट में उसने सेल्स चैनल्स लिए, वैल्यू प्रोपोजिशन इन्हांस करा, geographical focus में being in Ludhiana, he has vision to expand to European markets. Like I was telling you कि European market are looking for vendors and suppliers जिसके product में carbon footprint या carbon dioxide emission reduction strategy है. So आज सुबह मेरे पैनल पे मैम ने बताया था sustainability is also a key thing. तो मैं आपको ये बताना चाह रही हूँ कि लिग्निन को लेकर बायोप्लास्टिक्स बनाने के लिए द स्टार्टअप ऑल्सो टारगेटेड यूरोपियन मार्केट्स, क्योंकि उनका अनमेट नीड इन प्लास्टिक्स, इन सस्टेनेबल प्लास्टिक्स वो मीट कर पाया. राइट, अभी ही हैज कस्टमर्स हीरो साइकिल्स, कैंग्रू इंडस्ट्री एंड ऑटोमोबाइल कंपनीज हु आर मेकिंग डैशबोर्ड्स, मडगार्ड्स एंड accessories. So this is the value chain I wanted to tell you. Moving from one customer to larger customers in automobile industry, जहाँ पे आप bioplastics, durable bioplastics दे रहे हो, which last longer. That is also a component of decreasing the carbon footprint, that you make products which last longer. ठीक है, ESG और carbon footprint अलग session है. So I'm just touching on those terms क्योंकि बच्चों को आज ये जानना चाहिए कि ESG क्या होता है, कार्बन फुटप्रिंट क्या होता है, सस्टेनेबिलिटी क्या होता है. आर द स्टूडेंट्स विद मी? सस्टेनेबिलिटी क्या, कैसे आया इस कंसेप्ट में? एनी क्वेश्चंस? किसी को समझ आया कि हमने एक लिग्निन के कंपाउंड को लेकर वैल्यू एडेड बायोप्लास्टिक्स बनाए और उसमें सस्टेनेबिलिटी कैसे आया? इज इट क्लियर टू द स्टूडेंट्स? एनी क्वेश्चंस?
Students, do
now,
you have any questions? You are free to feel and ask, ma'am.
Because I want to conclude here because these are totally like a startup pitch also. His main thing I want to show here कि competition होना अच्छा है and how can you be better than your competition to survive. This he achieved to be a successful business venture. Right, this is about his unit economics, cost of goods. He calculated raw material procurement से लेके electricity, wages, other direct cost, और उसके बाद how he can turn profitable. That is his calculation, and annual sales price के साथ he wants to achieve a gross profit margin of up to 30%. So this, I'm not going into the details of the slide, but this is for students to understand how he turned profitable. राइट, ये उसका यूनिट इकोनॉमिक्स है. 50 kg एग्रीकल्चर वेस्ट प्लांट से बायोप्लास्टिक्स बनाने का टू अचीव अ प्रॉफिटेबिलिटी ऑफ अप टू 30%. राइट, विद दैट आई क्लोज एंड लाइक आई आस्क एनी क्वेश्चंस, बट मेरा मन कह रहा है कि प्लीज डोंट आस्क क्वेश्चन, प्लीज लेट दिस बी ओवर. बट स्टिल इफ देयर आर एनी क्वेश्चंस, यू आर मोस्ट वेलकम बिकॉज विनीता इज वेटिंग फॉर द नेक्स्ट सेशन. एनी क्वेश्चंस? हम सबका सेशन के बाद यही मन करता है कि हम लोग पूछ तो लेते हैं एनी क्वेश्चंस, बस अंदर से लगता है प्लीज लेट दिस बी ओवर, डोंट आस्क एनी क्वेश्चंस. मैम हंस रही है तो शी इज विद मी. एनी क्वेश्चंस सीरियसली फ्रॉम स्टूडेंट्स, फैकल्टी, स्पेशल डेलीगेट्स?
Ma'am, sir has a question for you.
Yes, sir. Yes, sir. This is what we were trying to show. Sir, he modified his process such that he could utilize high amount of lignin. He did research to make his extraction process viable with low specific gravity, high tensile strength, utilizing maximum raw material so that he could predict a gross revenue of 25 to 30%. So that's the R&D which CFIT helped him to do. Also, like I was showing you, he has successful customers. Yes, sir.
When people go for buying things, they normally don't care or they don't know it, most of them. So then they buy whatever the cheaper thing they get. So to reach up to last stage, will it be cheaper than the plastic material?
So, sir, I want to answer that question in a philosophical way. Indian customer is very cost conscious. Free मिल रहा है तो दो दे दो, ₹2 भी सस्ता मिल रहा है तो वही खरीदेगा. उनको carbon footprint से मतलब नहीं है.
मालूम नहीं है, उनको मालूम.
Exactly, I fully agree with you. But sir, his value proposition was targeting companies which appreciate carbon neutrality and ESG. Because that's the new thing in the market, and उसका जो production process है, to the end users, it will be comparable to the plastics which are available in the market at a reasonably high cost, क्योंकि he's working with original manufacturing equipment, original equipment manufacturers. They were willing to take the cost because they were getting a label of a sustainable product.
यूरोप में ज़्यादा पॉपुलर हो जाए.
Yes, sir. So, sir, आप देखेंगे वो लुधियाना से सीधा expansion यूरोपियन markets कर रहा है, क्योंकि वहाँ पे market है who value these sustainable bioplastics. So, this is also something for all students to understand.
एक suggestion. In addition to this, you make it SWOT. करना चाहिए.
Definitely, I agree, sir. क्योंकि mass appeal, mass recognition, mass acceptability is very much there for a startup. और एक चीज बोलना चाहूंगी, हम में से कितनों के पास आईफोन है? आप आईफोन का एक चीज देखेंगे, प्रोडक्ट वो सिर्फ 11% कस्टमर्स को टारगेट कर रहा है, वो उसी में से रेवेन्यू कमा रहा है. बट बाकी जो मोबाइल कंपनीज हैं, they are targeting the rest of the clientele 89%, क्योंकि वो 11% को eliteness चाहिए. And guys, you have to believe me, iPhone, Apple products are very high on environment sustainability. India में concept catch up नहीं हुआ है, but people who are carrying an iPhone, the battery lasts longer. The products are made from recyclable, compostable plastics. So there's an elite class of customers who will always be there for your products. So if you want to play the game of volume or if you want to play the game of eliteness, the choice is yours. But India का population इतना है 143 करोड़, there is market for everyone. Customers वो भी मिलेंगे जो ₹2 के पीछे कोई और product खरीदेंगे. Customers वो भी खरीदेंगे जो eliteness के लिए iPhone ही खरीदेंगे. But in addition, they are getting a product which is more long lasting. हम लोग branded चीज़ों के साथ क्यों जाते हैं? चाहे वो 30 वॉश में खराब हो जाए, but otherwise product 3 वॉश में ही खराब हो रहा है. So having a product that lasts longer is also saving the earth from that product to going for dumping. So sir, अब आप पूरा value chain समझे. So the mindset is changing towards sustainability. This is the collective consciousness we want to raise. Ask for products that last longer. Buy products that last longer. जूता भी आजकल 3 महीने से ज़्यादा नहीं चलता है. चाहे जितना ब्रांडेड ले ले, they want repeat customers. But अब environment के हिसाब से देखें तो वो गलत है सर. Earlier, I bought a shoe which lasted me 15 years. Now my shoe does not last even 3 months, which is totally wrong. But sir, consumerism इतना high है, हम environment को नहीं देख रहे हैं. So it is for all of us to make a collective conscious decision, invest in quality and go for people who give you better value for money. Any more questions? So this is for children. This is my Gmail, this is my LinkedIn account. I have built my LinkedIn profile very well. You can connect with me. I do a lot of mentoring sessions for startups, entrepreneurs. I love to interact with startups. सब मुझे पूछते हैं कि हम idea ही कहाँ से लाए business बनाने का. So we can have a one-on-one with you, and Professor Ajay is there, the VC of Dev Bhoomi University. He's a very good friend, and he's invited me to be interacting more with all of you, the faculty and students of Dev Bhoomi. So very fortunate to be here. And also I have my Insta profile, because youngsters are more on Insta than LinkedIn. So please do connect, and would love to stay in touch. My number is on WhatsApp. I respond to every message within 24 hours. And if you send me a LinkedIn connect, I'll ask you, good to connect, how may I help you? So thank you guys, you are the future, and the future is very safe.
Thank you, ma'am, for sharing us so much valuable thoughts for our students, and definitely that would be really helpful for their future endeavor. Now I request our Pro-Vice Chancellor, Professor Ritika Mehra ma'am, to kindly felicitate our eminent speaker, Professor Shikha Dhawan, who has delivered a beautiful session on how to have a startups. Can we have a huge applause for our eminent speaker who's delivered beautiful thoughts for our students, how they can have their own startups. Students, this was for you. Can we have a huge applause for you, guys? This side से no noise. मुझे लग रहा है मेरे को कोहाट सर को दोबारा बुलाना पड़ेगा स्टेज पे. Can we have a huge applause? Better. Thank you, ma'am. It is a time to listen from the another eminent speaker, Professor Vinita Jindal ma'am, a senior scientist at Biotechnology Industry Research at Assistant Council, BRRAC.
So let me have the honor to introduce Vinita while her slides being put up. So Vinita at BIRAC is so passionate about saving your intellectual property. अब बच्चे समझते हैं intellectual property क्या होता है? Which you have generated from your own mind, जिसका value है, उसको save करने के लिए आप patents कर सकते हो, कोई company बनाते हो, उस logo को कैसे trademark protect करना है, copyright protect करना है. So Vinita is very passionate about that, and today she's going to talk about the government schemes, government grants, which can fuel entrepreneurship and startup concepts and idea. To introduce Dr. Vinita formally, she is a distinguished biotechnologist and science administrator, currently serving as a senior scientist at Biotechnology Industry Research Assistant Council, that is BIRAC, a public sector enterprise under the Department of Biotechnology, Government of India. With a career dedicated to advancing biotechnology, innovation and entrepreneurship in India, Dr. Vinita has played a pivotal role in bridging the gap between scientific research and industrial application. Her expertise spans biotechnology innovation management, public-private partnership, and strategic program implementation aimed at strengthening India's bio-innovation system. जब तक विनीता का presentation नहीं आता, I can go on and on because she has done remarkably well at BIRAC. She has been instrumental in conceptualizing and managing key national programs that promote translational research, startup incubation, and technology commercialization in sectors such as healthcare, agriculture, and industrial biotechnology.
With a deep understanding of the challenges faced by early stage innovators, Dr. Vinita has worked extensively to develop funding and mentorship models that enable scientific discoveries to evolve into viable market-ready solutions. Over to you, Dr. Vinita. And a big round of applause for her.
Thank you, Dr. Shikha, for the nice introduction. And so my session has become, little, of course, I will talk about these schemes, but before you, Dr. Ajam, and you very well explained about, so there were two different concepts, one from the incubation side and one from the startup side. So how startup actually, think about the idea and how they work on their idea and what are the steps. She very, beautifully explained about how to take up idea and what are the technically as well as on the business side. And Dr. Azam explained about the pitching session or the importance of the pitching, how you have to present your idea within 2 minutes or within 5 minutes, whether it to the funding organization or to the investors. So it could be anybody if you have to, so what are the different points or the items you have to put as part of the presentation. So from the BIREC side, of course, you might be aware of, we are a funding organization. And so what I will be presenting here for another 15 minutes, how BIREC has helped or nurtured the biotech innovation ecosystem in the country. So basically, I will be covering about the how we are bridging research, innovation, and commercialization part. So if we talk about the creativity and the inventiveness, so these are the two components when we talk about the startups or the individual entrepreneurs. So if you have the creativity to bring out, to perform on your idea, and that has to be inventive and that has to be novel, then you are able to create a lot of prospects, not only for yourself, but for others as well. So how we do it, so before that I go into different components of what BIREC is handling or doing. So BIREC is basically a Government of India enterprise, which was set up by the Department of Biotechnology in 2012. And we have a vision and mission to promote the biotech ecosystem where we can bring entrepreneurship, entrepreneurs, as well as we promote the biotech startups. And definitely our target is to bring the affordable product to the society. So within the BIREC, we have a different programs. So with the help of the different programs, we support the biotech innovation and research ecosystem. So in morning when Dr. Mrityunjay also talking about how they are supporting the innovation ecosystem with the help of the, he talked about the TRL levels, technology readiness levels, and the mentorship program and the incubation system and other programs as well. So within BIREC, again we have a different program and for each category, and when I say each category, that means we also target the students, we target the entrepreneurs, individual entrepreneurs, as well as the startup companies, whether they are MSMEs or young startup companies. In addition to this, we have programs available for the academic institution. The only difference, if I mean, you can ask to me, what is the difference between the BIREC and other funding organization? So if we talk about the DBT and DST and CSIR, maybe we can keep out CSIR out of this segment. DBT and DST, they are promoting or they are funding basically for the academic or the basic sciences they are funding. But so there was the idea, if we can support the translational science, so that we can have, after some time, if we can have a product or technologies out of these translational research. So that's where BIREC came into picture, and we are promoting the translational research. So that is the difference where we have brought up the concept of funding the translational research, and based on which different startups in the biotech segment can be come up. So within BIREC, we have different programs, and along with the other programs, we have program management units. They also have a different functions to operate, like Mission COVID Suraksha, where with the help of this program, we were able to brought up different COVID vaccine during the COVID time. Along with this thing, we have Grand Challenges India program, where we have partnered with the Bill and Melinda Gates Foundation. So they have set up a unit within the BIREC where we are promoting socially as well as the scientific projects. Again, we have a World Bank and Department of Biotechnology supported program management unit, National Biopharma Mission. And under this mission, we have promoted development of the biopharmaceuticals as well as the medical devices and diagnostic sector. And this program also had a very important component, the skill development. Because when we talk about promoting the research and innovation to bring out the quality products, we need a skilled person who can run this show of the biotech. Because people are very important, and skilled people we have to bring out the more products in the market. So there is a program within the National Biopharma Mission to create the skill or to enhance the skill part of the individual students as well as the researcher. And then there was a program on the National Nutrition Mission, and within BIREC, of course, So we promote from the ideation stage to the product development or the product commercialization. So this is the entire chain. If you can see on this slide, on the lower part, from the ideation, where the student also become part for the ideation stage. And then from ideation to proof of concept, we have another different program which we call it as Biotech Ignition Grant, where startups can apply. And then once you have a proof of concept developed, right, after ideation you have worked on your idea, you have proof of concept, then you become a company and then you apply for the larger funding beyond the technology readiness level 3. So here is, so entire scheme is actually dependent on the technology readiness level, how you move from ideation, which is at the very nascent stage, which we call it as a 0, and then you develop or you design your idea, you come up with some data in terms of scientific research, then you develop a proof of concept which comes at technology readiness level 3, and after that early stage validation, late stage validation. So for covering each aspect of the research or the innovation cycle, BIREC has created different programs. For proof of concept, ignition grant. For early stage development, there is a program which we call it as a SIBRI program. And for the late stage development, we have BIPP. And of course, we have created bio-incubation centers. They are around 100 incubation centers across country, which BIREC has established. Of course, Roorkee is one of them. And we also have one incubation center in BHU in Lucknow as well. So if you are a startup and young entrepreneur, but you don't have a facility to work on your idea, you can become a part of these incubation center. They will guide you, they will mentor you on your how you can work on idea. So along with the instrumentation facility, which you might not be having as a individual researcher, you will get all the facilitation at the incubation centers. So again, as we grow, like we have developed equity program as well. And at the end, we have product commercialization program. When you have done all kind of validation, and you are ready to launch your product in the market, but still you feel there is some requirement to bring it to the market. So that's where the product commercialization program comes into picture. So I talked about the bio-incubation center, which we have created. So you get the incubation space where some office or a cabin or a seat is allocated for the individual researcher or the startup, and you will have access to the common instrumentation facility. They will provide you mentorship. They will also, expose to you to different networks who can help you on writing a proposal for the different investors or different funding organization. They will also, guide you. These are the organization where you can approach. So all kind of, 360-degree facilitation, you can get it as part of the bio-incubation centers. So this is the program I was talking about, the Biotech Ignition Grant. So Biotech Ignition Grant is basically, so initially we used to support individual researchers or the innovators as part of the Ignition Grant. But now we have changed because it's been like, we started this program in 2012. And since then we were supporting the individual innovators. And what happened after that, since 2012 until date, we were able to create around, more than 5,000 biotech startups. So now we thought to, change the process. So we have one program. Under that program, we will be, supporting the individual researchers. But this program will now, only allow startup companies to apply under this program. And this program is, you have idea and you want to work on that. And this grant will allow you to come up to a proof of concept stage, and grant is up to 50 lakhs. That will be given to you for a period of 18 months, right? And within the grant, there are options of, filing a patent application, and if you want to work with the university ecosystem, you can also work with them. So EUVA is the program where the undergraduate students as well as the postgraduate students can be a part of, if they want to be an entrepreneur. So there are two components. One is the fellowship, which will be provided by the centers. So BIREC under this program does, two different things. One is setting up of the EUVA centers, and they will announce the, calls to students to be a part of this program. And it will be both for undergraduate and the postgraduate students, where they will get a pre-incubation kind of, mentoring, how to work, how to, think about the idea, and they can identify the problem and how this problem can be taken up forward. So another, socially relevant, if any entrepreneur or researcher or person think about socially related needs, or we can call them the social innovation, working for the, it could be, solving the problem related to the environment, related to the older people, or, any other society related problems. So there is a program which we call it as a SIIP, Social Innovation Immersion Program. Under this program, again fellows are, they are selected, and we have also created few centers, social innovation centers. These centers select the fellows and they work on the problem and identify the solution. Again, as part of the grant, they will get the mentorship, and they will get exposure to the entrepreneurship programs. So these two programs, they are for the early stage and late stage development, and given to the specifically only for the startups, MSMEs and the companies. If they want, they have crossed like TRO3, and they can apply for the SIBRI and Biotechnology Industry Partnership Program. This is the program where the academic institution, whether it is a private university or the government university, they can apply. And under this, we have two components. So we have Ignition Grant-like component within this program, which we call it as AIR, Academic Innovation Research. So as a institute and the scientist has submitted proposal, they will get a grant up to 50 lakhs for a period of 18 months. This grant is given for the scientists to develop the proof of concept. You have to have only proof of principle, not from ideation, but you have to have, little work done on your idea, and then you can apply for the academic innovation research. Once you are able to develop the proof of concept, now you bring the industry partner, and your proof of concept, if you want to get it validated with the help of the industry, then the another component, contract research scheme, that comes into picture. So this is a way where industry academia collaboration work, and if company is able to get the good results out of your technology, a successful technology transfer and commercialization can happen. So product commercialization program fund, of course, this program is at the very end when you have crossed the early stage, late stage validation, and then you want little grant or small fund from the BIRAC to bring out the product or technology in the market. So, so I was talking about, earlier program, these programs actually meant for promoting the research and innovation. But within BIRAC, we have different groups. So I belong to the intellectual property and technology transfer group. So when we get the proposals, or when we fund the research for the startups, for the academic institution, so BIRAC has responsibility or the duty to help them, to guide them on protecting the intellectual property, as well as help them on technology transfer and commercialization. Because we have access to the startup companies, we have access to the innovation developed by the academic institutions. So we can connect them so that technology transfer, or at least a collaboration, can happen between academia and startup companies. So what we decided, let's support the academic institution whom we are funding for the research and innovation, as well as for the startup companies, for protecting their innovation in India as well as outside India. So this program, the BIRAC Path program, which is for patenting and technology transfer. So what we do, we take the request from these our supported grantees or the startup companies or academic institution. If they want to take a support for patent filing, we help them. We help them on each aspect of the patent filing, starting from the patentability search. So for every patent filing, or even if you file a trademark application, you have to perform a search. So those kind of services or the support we are providing to our grantees, our startup companies and academic institution, so that they can file, they can protect their innovation in India as well as outside India. Same as for the technology transfer. So for technology transfer, because we have access to the startup companies as well as big companies, so we try to connect them for their technology. So these are the few offerings which we provide to BIRAC innovators on patent filing as well as on the technology transfer. And there is one advisory program which we run every month. For any startup, any young innovator, any faculty, scientist who wants to join this session, every month we do it, second Friday every month. So like tomorrow, this session is planned. So this is a kind of online session. Anybody can join from anywhere, and this is run from 4 p.m. to 6 p.m. every month, second Friday. And you can ask any question respect to the how to protect patent, what are the process, any nuances related to technology transfer, IP, and national biodiversity regulation. So everything is answered during this session. And as part of the IP group, we also conduct lot of awareness program. We sensitize the young students as well as the startup companies on creating awareness on IP and technology transfer. In addition to this, as part of the National Biopharma Mission mandate, when I was talking about creating the skill development, we have created technology transfer offices. There are 7 tech transfer offices which BIRAC has created all across country. And basically these offices, like the incubation centers, these offices help the startup companies and the academic institution on protecting the IP, connecting them with the industry, sensitize them on the process of the technology transfer, helping them on drafting the license agreement or the non-disclosure agreement, because as a scientist, we don't know what to disclose to the company when we start discussing with them about the technology. So we call them as a regional tech transfer offices, and these are the location where we have set up. So I'll stop here, and if there are any question, I would be happy to take. That question could be on IP, tech transfer, of course on BIREC programs. I would be happy to take any question. Thank you so much.
Thank you so much, Dr. Vinita. Are there any questions for her? Because she has worked us very thoroughly through the environment that the Government of India is providing for startups to flourish and to get funded. There's hand-holding at each step of the value chain. So Dr. Vinita is here with us. If there are certain questions to her, you can please catch her during the sessions. And now over to Nirjala.
Thank you so much, ma'am, for this very informative talk. May I now request Professor Yogender Singh to kindly felicitate ma'am with a token of appreciation and gratitude. I would also request Dr. Shikha Dhawan to kindly assist. Thank you so much, sir. So today we have heard a lot about innovation, a lot of programs run by the government and various schemes, right? So now we are moving towards a section where we are giving chance to our young minds to present their ideas in a form of pitching round. So we have a pitching round for our students, and for that I request Rutanjya and Gunjan to kindly take over and invite our students for the pitching round. Kindly see, it's a competition. Each team will get 3 minutes time. Thank you.
A very good afternoon to one and all present here. Let's add some spark to the room, because we are about to dive into a round where creativity meets courage and ideas meet impact. Welcome to the pitching round on innovation.
That's right. This is the round where creativity meets confidence and ideas turn into game-changing possibilities. A very warm welcome to all the participants, judges, and the amazing audience here today. Innovation isn't just about technology or inventions. It's about looking at an ordinary thing in an extraordinary way. It's about those moments that make you wonder, why didn't I think about that way? And today our teams are all set to show us that spark. So without any wasting time, let's get started with our first presentation of the day. Please welcome Team Planty Soul to the stage. Everyone, a big round of applause.
So you have 3 minutes to present your idea, and Dr. Vinita and myself will be providing feedback and if the audience has any questions.
Okay.
Good afternoon, everyone. Thank you so much for giving me this platform to present my work. Recently, we initiated our company that's named with Plantisol, and the first product we are launching soon is the menstrual cramp pain relief cream. It is focusing, of course, females from the age group of 15 to 52. Yeah, menstrual pain, as majority of the population here knows, is the uterine cramps. Painful uterine cramps occur before or during the menstrual cycle, and they are of two types, that primary and secondary. Secondary is because of some underlying disease like of fibroids. So the data shows that about 45 to 90% of the female population experience this menstrual pain, and in India it's about 71% of the females, they experience this pain. So the causes of the pain is there is a change in hormone prostaglandin production on up and low level, and there is a reduced blood flow. Because of that reason, the female feels those cramps. They are really very painful. I even had
Renu, sorry to interrupt. You're still on the problem statement. Please come to your solution.
Sure.
You just have 1 more minute.
So our problem is, of course, as I told you, that is the menstrual cramps. So the solution is with the menstrual cramp cream that is FDA approved. That is a patented product. There is no hormones are used in this product. So they are all plant-based, 17 plus plant-based products, what I'll say, extracts, they are used in making this menstrual cream. So and besides this, it's a clean formula. As I told you, no hormones, no side effects, no parabens, no gluten is used. And it is even compatible with, if some other medication, the female is taking some other medication, then of course this is very compatible with that product. So this is a patented product. Actually, we are not able to disclose our main ingredient here, but it includes that chamomile and menthol in it. So that's very soothing and relieving. So clinical trials, they are already conducted, and it was found that the team which conducted the clinical trial, they are from Ukraine, Kyiv, and the sponsor was Just Barry Health Care Private Limited. So this is about the clinical trial I am showing here. This is the data which we got after our clinical trials. And these are the primary results. Here I am showing these primary results, that there is a reduction in the pain after using the female when used this cramp cream. There is a 1.48 point reduction in the pain, and further time the effect when this cream start working is the time duration is also less in this product of ours. About 22% of participants, they didn't require any further medication, and 40%
Renu, your time is up. Can you quickly move through your slides?
Yeah, sure.
Don't read, just quickly finish them off. Next. Okay, I think that's good.
Yeah.
Vinita, any questions for her?
Couple of questions.
Yes.
It seems it doesn't look like your idea. If it is already been done in USA, or I was unsure, because you have mentioned in one of the slides it is FDA cleared.
Yes.
Clinical trial done.
Yes, ma'am.
So it is first thing. It is beyond the ideation stage.
Yes.
And when you say clinical trial, was it on the animals or the human trial you have done?
It's ma'am. It's on the sample size is mentioned here. See, the females, the age group females, this 18 to 40 years of the females, they are randomly selected, and this is the inclusion criteria.
Was this clinical trial done in India?
Yeah, that's done in India here only.
So is this developed in India?
Actually, this is the patent. I'll show you. This patent is granted here in India.
Go back on maybe on the second or third slide. Go back. One more slide. No, I think it was second or third slide. That one. So you said hormone-free formula made in the USA.
So, ma'am, actually this product is just on sale in USA and in Europe also.
Already?
So yes, the member, the board member with us, he is Indian and he is from Haryana only. So he had that research done, and we are here working on the Plantisol marketing part. So
what exactly you are pitching for?
For this, actually, we are looking for funding. Like when you said that there is a product commercialization program, like, so yeah, that's the main thing I just want to have.
Okay.
Yeah.
Okay. Understood. Thank you.
So Renu, are you selling in the Indian market also?
Yes, ma'am. No, this is for the first time for the Indian market. We are planning to. So when I just heard ma'am's presentation that there is a program for product commercialization program, so definitely we are making our, we want to apply for some funding if we'll get something.
What is the cost of the product?
So ma'am, maximum we'll keep it about 200 rupees, not more than that.
Where is your production facility?
That is in Baddi. We are just that all the production and packing that is done at Baddi.
So how much units are you producing in what time duration?
For the first time, ma'am, we are ordering 2,000 units.
2,000?
Yes, it's just a very recent company actually. So we are applying for some funding at different levels. Now I'll apply with ma'am also.
All right.
Yeah.
Competitive product in the market already.
Yes, ma'am.
Who looks very close to your
yes, ma'am.
Is there any?
Ma'am, there are many, but in India it is not like we are, what is the uniqueness is that we are making it in a roll-on way. That is very easy to apply. Besides, I myself used this product, and this is really very, I'll say soothing. So I don't found that kind of product in Indian market, though it is available in European and American, US. In India, I didn't find this.
So is it a cream or a solution?
It's a solution. It's a solution that a kind of, I'll say gel that will roll on, you just move on, then that kind of gel.
Okay, I guess that's all. Thank you so much, Renu. All the best.
Thank you so much. Thank you so much.
Thank you, team.
A big round of applause for Renu and all the best for her future expansion.
Thank you, Team Plantisol. That was an inspiring presentation, full of fresh thinking and passion. Judges, we hope you noted some innovative highlights there. All right, let's keep the momentum going. Up next, we have Team NIRAXION-AI, ready to share their big ideas with us. Let's cheer them on, everyone.
बहुत सड़ गया है
Hello. Jai Hind everyone, my name is Anshul and I am the BTech student at Dev Bhoomi Uttarakhand University final year. Today I am representing NIRAXION-AI. First of all, what is NiractionAI? We are a software-based company which mainly focus on OpenCV, agentic AI, and generative AI. How we can implement these technologies nowadays in our modern products. So today I am representing one product from our company, which is NiractionAI, a PPE camera for the industries. As we know, there are around 48 to 58 lakhs industries in India. Okay, and every industry have their, like, mainly they focus on the peoples. Okay, so they are not accepting the basic PPE protective equipment for the person, which can be a helmet, gloves, or anything boot. So as we know, it is an important part for the human safety. So here we just introduce OpenCV or other technologies to enhance the safety of the workers. So as we know, every company has established their own cameras. So we can use those cameras, and by the software and the OpenCV processing, we can help those industries to maintain the level of safety for the persons. Now, what we do real-time PPE, means personal protective equipment detection, through the cameras which already established by the company. Our models is going to be evaluate and monitor the person which are working under the industries. They are going to be check out is the person is wearing the helmet, wearing the gloves or not. If not, it's going to be generate a alarm system or a alert for, or just forward to the adminal part. What is our solution? We are here for AI-powered PPE solutions, and as we know, our India is just grabbing the AI market, so we can use the AI or the other generative things to the market where we can establish the safety of the workers. Now, what is our market opportunity? What is our TAN, total addressable network? It is not necessarily like only India is the industrial where the industries are occupied. No. In the whole world, we have multiple industries with the workers, they are working there. Yeah, right now what is happening is there is a separate person who is just monitoring, but it is not efficient on that way. We have around 150 billion to 170 billion of market where we can grab this opportunity. We can grab the industries to accept our product. We have total around, we have 18 to 22,000 serviceable available market, and what is our serviceable optimal market is around 50 to 100 crores. What is our unique selling proposition? The first, what is our USP? So yeah, we done all these functions in a real time. The admin will get the real time notification where we can see how we are going, what is our production market. We can establish the basic plan to a premium plan where the industry can occupy us product, how they are going to be manageable the things. We can connect with the government to implement our product in their companies. Our current project is we just develop our hardware, along with we are creating a software for the admin or the dashboard where the admin will have a proper overview. This is our team, and we established NiractionAI as a startup firstly, but now we are just registered NiractionAI as a private limited company.
Very good, Dinesh. I would want to interrupt you. Very important use case. Can you tell me, have you taken feedback from your customers? Because
Yes,
these are the construction and manufacturing sector.
Yes, ma'am.
So do they need this technology, or let me reframe, how are they already ensuring the safety of their workers?
Ma'am, right now they have a adminal part who just monitor, like a supervisor, who just observing people through an open eye. If not, like somehow they are lacking. Okay, in India it is, the people are not accepting the safety measures. They are just, no, we have to work, we will get a payload. But we connect with three major companies like FL Smith, Maruti Suzuki and Medapet to concern it, and we also go for the Motherson to evaluate the thing.
Very good. Do you have more slides?
Yes, ma'am.
Do you have more slides?
No, ma'am, right now.
इसके आगे slides हैं?
No, ma'am, there's an open question.
Okay, all right. Vinita?
So what I understood, you will be using the camera.
Yes, ma'am, through the OpenCV or generative AI.
And how the data will be captured?
Ma'am, through YOLO technology, we're capturing the, we have already getting the live stream from the camera. We use that live stream to, as an input to our processor. Through the machine learning algorithms, we are just capturing is the person is wearing the helmet and other equipment or not. And through that, we will generate an alert.
So the supervisor will be able to access the data on his
Yes, ma'am. There is a different ecosystem or dashboard where all the industrial requirement things are already established, where it can get the data of the worker, it will get the live feed from the whole universe, from the whole industry and all.
Okay, thank you. Thank you.
Okay, thank you. Thank you, everyone.
Big round of applause for Nirakshan Startup.
Now let's welcome Team 3, BioPrick, to take the stage and share their innovative spark.
Good afternoon, all of you. I am Aniket Sandilya with my team members.
My name is Sachin Kumar.
My name is Srishti Khandelwal from BSc MM.
एंड मैं आप लोगों के सामने एक ट्रॉमा डिस्कस करने वाला हूँ, मेनली बचपन का ट्रॉमा, जैसे पेरेंट्स का एंड उनके चिल्ड्रंस का. मेनली आप लोगों ने ये चीज़ नोटिस की होगी कि जब बच्चों को वैक्सीनेशन या एंटीबायोटिक्स लगते हैं बचपन में, तो उनको काफ़ी पेन होता है विद द, वो मेनली पेन होता है इंजेक्शंस का. तो हमने एक इनोवेटिव आइडिया डिस्कस करके एक इनोवेटिव आइडिया दिया है जिसका नाम है बायोप्रिक. तो बायोप्रिक एक टाइप का इंजेक्शन है जिसमें हम गैलियम कोटिंग करेंगे, और ये एक ऐसे टाइप की कोटिंग होगी जो बॉडी के अंदर 3 लेयर टेयर करके अंदर जाएगी और सॉफ्ट हो जाएगी, जिस वजह से पेन एक हद तक बहुत कम हो जाएगा. और ये बायोकम्पेटेबल है और बॉडी के लिए इसका कोई डिसएडवांटेज नहीं है. तो इट्स अ गुड अपॉर्चुनिटी फॉर अस. प्रोडक्ट ओवरव्यू करूँगा मैं. एडवांस मटेरियल साइंस इनोवेटिव गैलियम कोटिंग ट्रांसफॉर्म्स कन्वेंशनल नीडल्स इनटू सुपीरियर मेडिकल डिवाइसेस. मतलब हम इसमें एडवांस मटेरियल साइंस यूज़ करेंगे, पेनलेस एक्सपीरियंस मिलेगा हमें, सेफ्टी फर्स्ट होगी इसमें. मेनली इट मीन्स कि इसमें कोई एक टाइप का कोई भी बैक्टीरिया कॉजिंग कुछ भी ऐसा नहीं है. प्रोडक्ट मार्केट सेगमेंट की बात करें तो इसमें हेल्थ केयर, रिसर्च फैसिलिटीज़ और बायोटेक्नोलॉजी R&D. इट मीन्स हम इसे सबसे पहले हॉस्पिटल्स, देन उसके बाद डायग्नोस्टिक लेबोरेटरीज़, फिर उसके बाद इंस्टीट्यूशन, इट मीन्स रिसर्च के लिए भी आगे सेंड कर सकते हैं. USP की बात करें तो ये इसका यूनिक सेलिंग प्रॉपर्टी मेनली होगा ये पेनलेस इसको इंसर्ट करेंगे तो ये पेनलेस होगा, सेल्फ स्टरलाइजिंग है, बायोकम्पेटिबल है और इन्हेंस सेफ्टी को इन्हेंस करता है. मार्केट साइज इसका 67.200 करोड़ है इन वर्ल्ड ग्लोबली मार्केट में कहे, और इसमें हम 5 to 10% मार्केट को एडवांस टेक्नोलॉजी से बीट करेंगे. गो टू गो मार्केट स्ट्रेटजी रहेगी इसमें पायलट प्रोग्राम, B2B डिस्ट्रीब्यूशन, इंडस्ट्री शोकेस. इसमें स्मार्टर, सेफर, पेन फ्री. इसका बायोप्रिक मेनली हमारा विज़न रहेगा स्मार्टर, सेफर एंड पेन फ्री. बाकी हमारे टीम मेंबर्स हैं ये. That's all. Thank you.
So, very good team BioPrick. I want to ask you a question. Galenium coated material ये कहाँ से लोगे, कहाँ टेक्नोलॉजी डेवलप करोगे?
मैम मेनली ये इंडिया में जैसे हम सूरत, मुंबई, इन सब में यहाँ पे इनके डिस्ट्रीब्यूटर्स हैं जो इजीली हमें ये चीज़ प्रोवाइड कर सकते हैं और हम उसका यूज़ करके हम मतलब मेनली इसको फिर कोयले स्टेनलेस स्टील में कोटिंग करके इसका यूज़ कर सकते हैं.
So, what are you coating that it make it safe to go inside the prick site?
मैम एक्चुअली हम प्योर गैलियम का यूज़ करेंगे.
Just one material.
यस मैम, ओनली प्योर गैलियम. जो ये इसका कोई डिसएडवांटेज नहीं है बॉडी के अंदर भी, ये मतलब कोई भी डिसएडवांटेज में गैलियम का नहीं है अभी तक.
प्रोडक्ट का कॉस्ट कितना रखोगे?
मैम इसका अगर हमें अच्छा कंट्रीब्यूशन मिलता है, मतलब अच्छा इसका वो मिलता है, तो हम ज़्यादा पॉपुलर इसको मतलब ज़्यादा अमाउंट में वो बनाएंगे, जिससे इसका आई थिंक 30 रुपीज़ ऑलमोस्ट होगा इसका प्रोडक्ट का कॉस्ट.
और नीडल का गॉज साइज क्या है?
21 गॉज मैम. 21 गॉज.
21 गॉज.
यस मैम.
ओके. एंड व्हाट अबाउट द इसमें हीट कंडक्शन क्यों चाहिए?
व्हाट मैम?
हीट कंडक्शन क्यों चाहिए?
मैम हीट कंडक्शन इसलिए चाहिए क्योंकि गैलियम को मेल्ट करने के लिए और स्टेनलेस स्टील के साथ बाइंड करने के लिए हमें एक हीट जनरेट करनी पड़ेगी जिससे हम उसको कोट कर पाएँ एंड देन हम कूल करेंगे उसको.
Okay, so how serious are you to take this idea further to get funded or to form a startup?
मैम अगर हमें अच्छा सपोर्ट मिलेगा तो हम इसको लॉन्च करेंगे एंड इसमें हमें हमारा अपकमिंग आइडिया भी है. इसमें जैसे हम वैक्यूटेनर्स होते हैं उसमें भी हम इसका यूज़ करेंगे. और अभी मैंने एक और अपकमिंग आइडिया सोचा था कि मेनली इंजेक्शंस जो होते हैं ना, वो बच्चों को देख के भी, जो स्मॉल चिल्ड्रंस होते हैं उनको देख के भी उससे डर लगता है, तो हम इंजेक्शंस को भी कस्टमाइज़ कर सकते हैं इस तरीके से जो मतलब दिखने में एज सच ऐसा कुछ दिखे ना, थोड़ा हल्का फनीड मेडिकल टर्म से थोड़ा अलग हो फॉर चिल्ड्रंस.
So these are not single use.
सॉरी,
These are not single use.
मैम दीज आर फॉर सिंगल यूज़. मैम सिंगल यूज़ कर सकते हैं, इसका सिंगल यूज़ ही होगा इसका बस, क्योंकि इसके बाद ना ये मेल्ट हो जाएगा, इसके ऊपर का गैलियम का वो होता है वो इतना लूज हो जाएगा, सॉफ्ट हो जाएगा, उसके बाद उसका मल्टीपल यूज़ इसका हो ही नहीं सकता है.
Good. All the best.
थैंक यू मैम.
थैंक यू मैम.
ये आप निडल भी सॉरी इंजेक्शन जो पूरा विद निडल है इसको आप बना रहे हो या सिर्फ कोटिंग कर रहे हो?
मैम ये मेनली है ना, हम स्टेनलेस स्टील का यूज करेंगे, इसके ऊपर हमने कोटिंग करेंगे.
नहीं, ऑलरेडी अवेलेबल तो निडल आप नहीं बनाओगे, जो इंजेक्शन है इसका बॉडी ये आप नहीं बनाओगे, ओनली कोटिंग पार्ट आप करोगे?
नहीं मैम, हम निडल भी बनाएंगे और कोटिंग भी करेंगे, क्योंकि जब हम निडल बनाएंगे उसका जो वो थिकनेस है वो थिनेस्ट करेंगे, तो हम कोट करेंगे तो वो ओरिजिनल गॉज पे आ सके 18 से 20 के.
तो निडल के लिए कौन बायर होगा आपके पास?
व्हाट मैम?
निडल लेने के लिए जब आप निडल पे कोटिंग करेंगे उस निडल को आपसे कौन लेगा?
मैम ये डायरेक्ट मैं पहले हेल्थ केयर सेक्टर में जाऊंगा, गवर्नमेंट से बात करूंगा, एज फॉर गवर्नमेंट हॉस्पिटल्स रिसर्च करूंगा, अगर पॉजिटिव रिस्पांस आया तो फिर मैं आगे बढ़ूंगा मैम.
अच्छा, एक और चीज़, आपने कहीं पर पेनलेस लिखा था, इसको पेनलेस कैसे बनाएंगे?
बिकॉज मैम जब हम इस स्किन में कोई भी इंजेक्शन इंजेक्ट करते हैं तो वो काफ़ी हार्ड होता है और हमारे टिश्यू को काफ़ी हद तक डैमेज करता है और टीयर करता है. बट गैलियम कोटेड निडल का यूज़ ये है कि वो इंसर्ट करते टाइम लाइट पेन होगा एंड देन वो बॉडी में जाते ही सॉफ्ट हो जाएगा, उसके कारण हमें पेन फील नहीं होगा, हमारा टिश्यू डैमेज जो है और वो टीयर कम होगा.
ओके, थैंक यू.
थैंक यू मैम.
A big round of applause for BioPrick and all the best to them.
Up next, we have Team 4. Team Providor, come forward and inspire us with your concept.
कौन से?
क्या हुआ?
आप लोग आगे बोलिए.
Good afternoon, everyone. I am Tariq Alam, a first year BMLT student. Today I am here to present our innovative project, Advanced Cooling Blood Sample Rack. बेसिकली हम लोग एक ब्लड सैंपल रैक बना रहे हैं। जैसे कि नॉर्मली अगर हम कहीं पे जैसे कैंप लगा रहे हैं तो वहाँ पे हमको रेफ्रिजरेटर ले जाना उतना पॉसिबल नहीं होता है। अभी के प्रॉब्लम्स क्या है कि टेम्परेचर कंट्रोल इशू होता है, हीमोलाइसिस हो जाता है, ट्रांसपोर्टेशन हो जाते हैं और लैक ऑफ मॉनिटरिंग हो जाते हैं। हम एक ऐसा प्रोडक्ट बना रहे हैं जिसमें हम टेम्परेचर को मेंटेन कर सकते हैं 2 to 8 डिग्री सेल्सियस तक, और उसको हम ऑर्डर ऑफ ड्रॉ में अरेंज कर सकते हैं। ऑर्डर ऑफ ड्रॉ बेसिकली होता है कि जैसे हमको जिस टेस्ट के लिए हमको जो भी सैंपल लेना होता है उसको हम ऑर्डर में करेंगे। हम लोग का प्रोडक्ट का ये है कि हम सबसे पहले उसको ऑर्डर ऑफ ड्रॉ में करेंगे। ये लो बजट का होगा और इसको हम यूज करेंगे। इसको हम लेबोरेटरी में, हॉस्पिटल्स में और ब्लड बैंक्स में यूज करेंगे। हम लोग का प्रोडक्ट का ओवरव्यू है कि ये सेंसर्स बेस्ड होंगे। सेंसर बेस्ड यानी कि अगर हमें टेम्परेचर फ्लक्चुएशन आ रहा है तो वो हमारे फ़ोन पे स्मार्ट नोटिफिकेशन भेज सकते हैं, और ये इजीली पोर्टेबल, लाइट वेट। सपोज अगर कहीं पे डिजास्टर्स हो रहे हैं तो सपोज कहीं पे अगर डिजास्टर्स हो रहे हैं तो वहाँ पे ये इजी टू कैरी होगा। सपोज कहीं पे अगर एक्सीडेंट्स हो रहे हैं, मोबाइल वैन होते हैं, वो वहीं पे हम ले जा सकते हैं। और ये सबसे पहले ये बैटरी पे चलेगी तो ये बैटरी पे चलेगी तो ये लॉन्ग लास्टिंग भी हो सकती है। जैसे ये आइस बैग्स होते हैं कि उसमें कॉन्टामिनेशन भी हो सकता है, इसमें हम कॉन्टामिनेशन से भी बचा सकते हैं। हम लोग का यूनिक से वैल्यू प्रोपोजिशन ये है कि इसमें हम मार्केट को ऐसे टारगेट करेंगे कि स्मॉल कूलिंग टेम्परेचर के साथ रहेंगे, और इसमें हम ऑर्गेनाइज करेंगे ऑर्डर ऑफ ड्रॉ के साथ, और इसमें पोर्टेबल और एनर्जी इफ़िशिएंट होंगे, और ये इको फ्रेंडली भी होंगे। और मार्केट साइड हम लोग के ये है कि $, 6.71 बिलियन से है 2023 तक। ये फ्यूचर्स में और आगे बढ़ सकते हैं। हम लोग का ये है कि हम लोग टारगेट्स करेंगे पैथोलॉजी लैब्स को, स्कूल को, मेडिकल कॉलेज को, हॉस्पिटल्स को, और हम ये एजुकेशन में भी यूज करते हैं। सपोज हम लोग अभी BMLT स्टूडेंट्स हैं, हम लोग को अभी ऑर्डर ऑफ ड्रॉ पढ़ाया जाता है। इसमें ऑर्डर ऑफ ड्रॉ भी रहेगा जिससे हमको स्टूडेंट और क्लियरिटी से समझ पाएंगे। और आगे हम लोग का बिजनेस मॉडल ये है कि पहले ये डिजाइंस करेंगे 25, प्रॉफिट मार्जिन 10% रहेंगे, और टेक्नोलॉजी और इनोवेशंस 10%, आफ्टर सेल सर्विसेज 10% होंगे। उसके बाद डिस्ट्रीब्यूशन एंड लॉन्चिंग 10%, मार्केटिंग एंड सेल्स 15%। हम लोग का ये सबसे पहले हम फंडिंग एंड सपोर्ट करेंगे, उसके बाद रजिस्ट्रेशन और पेटेंट करेंगे, और उसके बाद प्रोडक्ट डेवलपमेंट करेंगे। फिर अगर कोई कंपनी अगर हम लोग से कोलैबोरेशन करना चाहेंगे तो हम कर सकते हैं। दीज आर माई टीममेट्स। थैंक यू।
Tariq, can you tell me, laboratories have already established the process for preserving the sample. ठीक है, वो सीरा या प्लाज्मा जो भी होता है सेपरेट करके रख लेते हैं। So how do you want to penetrate a market which is already having a solution?
मैम सपोज अभी की जैसे कि रेफ्रिजरेटर चलता है लैब्स में, और जैसे अगर कैंप कहीं पे हो रहा है वो आइस बैग्स चलते हैं, तो आइस बैग्स में हम कॉन्टामिनेशन भी हो सकता है। इसमें हम लोग के प्रोजेक्ट में ये है कि इसमें बैटरी पे चलेगी, AI बेस्ड भी होंगे, और टेम्परेचर में अगर फ्लक्चुएशन आता है तो वो स्मार्ट नोटिफिकेशन भी भेज सकता है, और कॉन्टामिनेशन से हम बचा सकते हैं, और लाइट वेट भी होगा।
एक लेबॉर्ट्री के लिए कितने मॉडल्स, कितने पीसेस सेल करोगे?
मैम हम लोग ये अभी 200 सैम्पल्स के लिए हैं, 200।
Okay, I think that's a very good idea. You can develop it further. Go for customer discovery. Customer क्या चाहता है? How many samples will they be able to take? और product का MVP कितना रखोगे?
मैम प्राइस मैम 12 to 15 के।
So वो एनालिसिस कर लेना कि मार्केट फिट कितना जल्दी अचीव कर पाओगे।
मैम हम प्रोडक्ट को ऐसे भी डिजाइन, कितना डिमांड है, इसके कितने सैंपल्स की डिज़ाइन है, उस अकॉर्डिंग हम प्रोडक्ट को डिज़ाइन कर सकते हैं।
ऑल राइट, ऑल द बेस्ट।
थैंक यू मैम।
A big round of applause for team Provida.
Thank you everyone for being such a great audience. Stay tuned for more exciting session ahead. I now request Nirja ma'am to take over the stage from here.
Thank you so much. Quite interesting to see our students have innovative minds. With this, I would like to extend a heartfelt thank to our chair and co-chair, Dr. Shikha and Dr. Vanita, for assisting the session and taking it ahead so beautifully. Thank you so much, ma'am. Moving ahead with the sessions, we have the next plenary talk by Professor R.C. Kuhar. I believe everyone is waiting to hear him out. And I would like to invite Professor Yogender Singh as the chair for the session. Professor Yogender Singh is currently serving as the NASI Senior Scientist Advisor at the Delhi School of Public Health, Institute of Eminence, University of Delhi. Sir, I request you to kindly chair the session and invite our speaker, our plenary speaker for today, Professor R.C. Kuhar.
I request Dr. Kuhar to come here. बैठिए सर, आपको बुलाते हैं 1 मिनट के लिए. Good afternoon, everybody. I guess it is the last session. I think Dr. Kuhar does not need any introduction. I am not sure if I say something, many of you will say, I know more than that. So, Dr. Kuhar sir is a great teacher in microbiology, very passionate about teaching and research, developing product. He is a great mentor. Large number of people have benefited from his ideas. He is a great administrator also. I know him from last more than 30 years when he was in microbiology department of Delhi University. Later he joined as a vice chancellor in Central University of Haryana, and I have been there many times. He has got a lot of ideas. He is considered one of the few scientists in microbiology. His citation and everything is great. So with this, rather than I tell you all these things, I request Dr. Kuhar to come and tell about the title is also very nice, New Generation Microbiology. So thank you, Dr. Kuhar, please.
Thank you. Thank you, Professor Yogender Singh ji. I think as per schedule, this should have been break time. I mean tea break. Dear friends, I believe not in numbers. I believe the listeners. Those who can sit, they are most welcome. थोड़ा सा थोड़ा ऊंचा करना. I think you have already enjoyed the good session pitching startup development and how to take your startup specifically from the idea to the product and ultimately to the market. You have also listened very well about the innovations, research and entrepreneurship. Dear friends, I have been given the responsibility this time to talk about next generation microbiology. The discipline of microbiology, as we all know, previously perceived as observing microbes through microscopes and growing them in media petri plates or flasks, which is rapidly evolving and is witnessing a revolutionary change. You might have seen since morning that how microbes have been studied and kind of revolutions they have been bringing. However, the transition of next generation microbiology has opened a new path into an in-depth study of diverse microbes Next generation microbiology is the study of microorganisms using cutting-edge technologies such as genomics, proteomics, metabolomics, bioinformatics, synthetic biology, and automation techniques, and specifically artificial intelligence to understand microbial life, interactions, and applications to the society. Why I'm defining what next generation microbiology is, because many of you, after completing the course, when you will go after doing PhD for faculty position, you won't be able to define next generation microbiology. That's why it is very important. Rather, this session and this my lecture should have been in the presence of many microbiologists so that AMI could have taken a lead in defining the next generation microbiology, and Academic Society of Microbiology, the American Society of Microbiology and European Society of Microbiology, they have been leading in deciding about the phenomena, the words to be used, and how to define the subject. And we have been following. And that's the pity, I will say, that many of the microbiologists who are aware about that we will be talking about in a session on next generation microbiology, and they are away from here. Anyway, ladies and gentlemen, those who are sitting, enjoy, because I always believe in cooperate. When you cooperate, you will care. Through caring, you will connect yourself, then you will learn, then you will acquire, and then you will progress as Sikha Dhawan is progressing in a career in his profession. Dear friends, the first transformation took place in microbiology methods with the development of molecular biology and genomics. I'm just taking you to slightly the background to the microbiology and how the subject is, progressing. Techniques such as polymerase chain reaction, DNA sequencing, and genetic fingerprinting have enabled researchers to uncover microbial diversity without the need for cultivation. That is nothing but your metagenomics. The further advancement and accessibility of 16S RNA sequencing facilitated the study and identification of microbes from a wide range of environments, as you heard Professor Ruplal in the morning, ranging from the human gut to the deep sea ecosystem. And very recently, it has been reported that there is a life signature have been found in the deep sea volcanoes, only 5 days back. Nevertheless, the development of next generation sequencing enables millions of DNA fragments to be sequenced and analyzed simultaneously. The cost effectiveness and result delivery in a fraction of the time makes it preferable over conventional testing methods. Dear friends, what sort of technical advancements have taken place which will be taking this, our modern microbiology, to next generation microbiology, and what are the applications I'll be just sharing with you. The emergence of next generation sequencing facilitated rapid genome sequencing bacteria, viruses, algae, and fungi, revealing their genes, mutations, and resistance mechanisms. Dear friends, with the emergence of new microbes and diseases, the next generation sequencing has supported the development of culture-independent diagnostics in clinical microbiology. And next generation sequencing not only helps in the precise identification of pathogens, even from mixed cultures where traditional methods were not very useful, but also in tracking the source of infection and predicting antimicrobial resistance. It has emerged as a powerful tool to tackle global health challenges such as COVID-19, tuberculosis, and nosocomial infections. Metagenomic sequencing has also emerged as an efficient tool in scenarios where the study of polymicrobial infections or unculturable novel microbes is required. and provides insights into genetic makeup, community composition, and potential functions of such microbes. Another transformation from the standard approach is the integration of multi-omics, genomics, metabolomics, proteomics, and transcriptomics, which provides a comprehensive analysis of microbial systems. Genomics provides insights into genetic constitutions, transcriptomics gives information on active gene expression, proteomics indicates functional proteins, and metabolomics reflects metabolic activity of the microbial system. Utilizing all these studies together facilitates a comprehensive understanding of the kind of microbes existing in the system, their functions, interactions, and responses to the environment. The integrated framework of studies has shifted the study in microbiology from descriptive to predictive, allowing us to anticipate the behavior of microbes and facilitate the design of effective interventions. Ladies and gentlemen, this multi-framework study gets accelerated when artificial intelligence and machine learning are integrated into it. The massive data set provided by omics and sequencing is computationally analyzed using algorithms to help in the identification of microbial species, prediction of resistant genes, classification of microbes, and designing microbial consortia for industrial application. AI-based models which mimic the growth, conditions, and survival of microbes under varied environments are applied before standard applications to check their enhancement in industrial fermentation, food safety, and pathogens control strategies in predictive microbiology. Next generation microbiology also has application in environmental microbiology, as it provides information on enormous microbial diversity involved in maintaining the biochemical cycles, like the carbon cycle, nitrogen cycle, and of course, contaminant degradation. The integration of microbiology with molecular tools can be used to engineer microbes to degrade emerging contaminants and transform them into valuable products. Next generation microbiology, if you consider, is also focused on using microbes as cellular factories for the production of energy and acting as microbial fuel cells. Biochemical tools like CRISPR-Cas systems and other genome editing tools which enable precise modification in microbial genomes can be used to increase the potential and productivity of microbial cells. In India, in agriculture-based country, NGM can be used for designing microbial formulation that facilitate nutrient uptake, protect against infections, and reduce the reliance on chemical fertilizers to improve crop yield and soil health. Next generation microbiology is also helpful in understanding extremophiles and potential microbes in space. Artificial intelligence has a lot of challenges, and of course, it has future prospects. Next generation microbiology has many prospects, but it also faces several challenges. Data overload has become an issue due to quick expansion of omics and sequence data. Expert bioinformaticians and sophisticated computational resources are needed to process, store, and interpret this data. Many microbial genes still have unknown functions, and linking genetic information to observed trait remains complex. Reproducibility and data standardization are also significant issues in labs worldwide. Integrating new technologies, specifically AI-based, with classical microbiological techniques is another major challenge. Despite the power of sequencing and computational tools, culture-based approaches are essentials for confirming predictions, testing hypotheses, and studying microbial physiology in detail. If we talk about the future of microbiology thus depends on combining these traditional skills with modern analytical techniques rather than replacing one with each other. Infrastructure and training are also critical barriers, particularly in developing countries, and establishing laboratories with next-generation sequencing platforms, bioinformatics capacity, and high-speed data storage is expensive. So that's also a major challenge. Looking ahead, the field is moving toward real-time and portable microbiology, handheld sequencing devices, AI-driven microbial detection systems, and integrated biosensors will enable rapid on-site analysis of samples from hospitals, farms, or natural environments. You'll be able to take like mobile kind of the equipments and you can analyze different environments. Advances in microfluidics and single-cell analysis will allow researchers to study microbial interaction at unprecedented precision. To conclude, next-generation microbiology represents one of the most transformative shifts in biological sciences. It goes beyond observing and cultivating microbes to understand them as interrelated systems that affect every aspect of life on Earth. By merging genomics, synthetic biology, and artificial intelligence, scientists are discovering the hidden potential of microorganisms and applying this knowledge for the betterment of the society. Dear students, the application of this new microbiological era are endless, ranging from producing renewable energy and cleaning up pollution to diagnosing illness in hours instead of days. However, realizing its full potential will depend on addressing data, infrastructure, and ethical challenges while maintaining the spirit of classical microbiology. The goal is not to replace traditional methods, but to expand them with modern tools that reveal the unseen complexity of the microbial world. Next generation microbiology will transform how we understand life, health, and ecosystem, moving toward personalized medicine, sustainable bioengineering, and global biosurveillance. In doing so, next generation microbiology not only enhances our understanding of life, but also empowers us to harness microbial power responsibly for a sustainable future. Thank you very much for patience listening.
Thank you, Dr. Kuhat, for giving so much information about microbiology. Thank you very much. Request Nirjara if she is around to, yeah, Dr. Prince will be coming or?
Thank you so much, sir, for the interesting talk. I would request Professor Yogender Singh to kindly felicitate Professor Kuhar with a token of appreciation. May I request our Pro-Vice Chancellor, Professor Saini, to kindly felicitate Professor Yogender Singh to chair the session. Thank you so much, sir. We'll now be starting with our final knowledgeable and scientific session for the day before we move into the cultural night. So this is basically a panel discussion. In the morning, we're talking all about innovation, but before innovation comes education, right? So in the next session is all about next-gen microbiology education. So the panel discussion on the collaborative dialogue on next-gen microbiology education is focused on redefining microbiology, learning through innovation, technology, and interdisciplinary collaborations. The eminent academicians, researchers, and industry experts will share their insight on the modern teaching methodology, curriculum advancement, industry-academia partnership to prepare future-ready microbiologists. This session will emphasize on the hands-on learning, research integration, digital tool, and entrepreneurial thinking as key drivers for shaping the next-gen microbiology to the microbiological profession. For this session, our moderator will join us. So I request our panelists to kindly take your seats. Our first panelist is Professor Charu Dogra Rawat. She is an assistant professor at the Ramjas College, University of Delhi. Welcome, ma'am. Our next panelist is Dr. Urvashi Kuhar. She is from University of Delhi. Welcome, ma'am. Our third panelist is none other than Professor R.C. Kuhar, the founding chairman of AMSC. Sir, welcome. I again welcome you to stage, the dais. I also request Mishra sir to kindly join us for this panel. Please, sir. Our last panelist will represent industry. So we have a team from High Media. Dr. Pal is here with us. Sir, can you please join us? The moderator for the session, Professor Prince Sharma, the President AMI, is joining us. Before that, I would really request our panelists to introduce themselves. I request our panelists to kindly introduce themselves.
A very good evening to all of you. I would say that it's an interested, quality audience that we have here, and I appreciate every one of you being present here and being keen and ardent listeners, and I hope that stays through this small panel discussion of ours. I am Dr. Urvashi Kuhad, Assistant Professor, University of Delhi, Ramlal Anand College. I teach in the Department of English. Yes, I don't belong to a biotechnology or a microbiology discipline, except for we've lived through microbiology. My father is a microbiologist, and we've grown up moving around microbiology laboratories, and we don't think that what we see or we smell is a stench, but it's a way of life. Yes, but I'm somebody who imbibes and believes in an interdisciplinary kind of a learning, and hence I've been lucky to be part of this discussion on AI and education. So that's a bit about me, and I've been teaching for the last 10 years. I'm also a Fulbright, and I'm looking forward to learning something new here.
Okay, so why not you continue yourself?
Good evening, everyone. I am Professor Charu Dogra Rawat from Ramjas College, University of Delhi. I've been teaching zoology there for past 20 years, and I'm a microbiologist by research. And I must congratulate AMI as well as Dev Bhoomi Uttarakhand University for keeping this session because as they have put it in the name, the education comes before innovation and research. So the discussion about microbiology education is the need of the hour, particularly when the new education policy, National Education Policy 2020 is here and the changes that it has brought up. So I really look forward to and thank you for your august presence, and I hope that it will be a fruitful discussion that can orient ourselves, all the fellow teachers as well as the students, to gain meaningful microbiology education. So I look forward to this session and discussions.
Good evening, everybody. Myself, Dr. Rajesh Pal. I am from High Media and I am a manager in High Media Microbiome Research Center. There I am looking after the microbiome based product development part at the center. I am in industry for last 10 years. I did my PhD from NEERI in 2015, and after that I've taken different industry positions in Hyderabad, Bangalore, and now I am located in Nagpur. It's my honor to share this stage with stalwarts of the domain. Thank you, sir, for having me here. Thanks to organizing committee and AMI. Thank you, sir.
Good evening once again. I'm Ramesh Kohad, student of microbiology, and I just spoke to you people that when we were student, we studied microbiology by growing organism in media, having in petri dishes, flasks, and through microscope. And of course, thereafter, number of the biochemical techniques were developed, molecular biology-based techniques came in, and of course, we reached to the stage of engineering these microorganisms, but now you know very well that a variety of artificial intelligence-based techniques have come up. Machine learning is also being taken into consideration. So there are many modern technologies, new technology, 21st century based technologies are coming now, and how they have been affecting, making the study of microbes very fast, even from the environments where we were not able to reach. So our reach has been increased, and we'll be discussing the form of next generation of microbiology, which just now I told you that had we have, say, more microbiologists here, then we should also take up, say, in the presence of President AMI and others, that how we can define, at least Association of Microbiology of India today can define next generation microbiology. And that can go to, say, all departments of microbiology to follow it up. Thank you very much.
Good evening, dear. This is Dr. Hari Mishra. I am ex-BRC, Bioatomic Research Scientist. After superannuation, I have joined to one of the public funded university as a distinguished professor. Thank you, Professor Kuhad, for bringing me to this platform. Yeah, see, microbiology, in classical terms of microbiology, what I see here that, the molecular biology, hardcore lot of things have actually killed the basic microbiology that actually we are still very strong and we have a strong foundation and lot more can be done. I'm not saying that microbiology, molecular biology is not combinable. I'll tell you one small example. I was teaching research methodology to the PG student, and there in the curriculum there was a component of write the reviews. I'm sure many of you might be teaching. Okay, so, all right, so we'll come back and then I'll narrate you. actually what is the strength of microbiology that actually we should be bothering for?
Hello. Um, it's a little disheartening in a very beautiful evening, but that the strength is so low at this moment. But for a topic like this, where we are going to talk about or discuss about next-gen education, I was expecting that there should be students very much interested in this kind of panel discussions. More and more participation should have been there. But anyway, whatever the people are, we are going to start this session. I'm delighted that I'm having a father-daughter combo here from different fields, and that is fascinating that one is from microbiology and the other one is from another field, and that's going to make a good combo about this discussion actually. So let's hope for that, and let us start with this panel discussion, which is on the next generation education, particularly next generation microbiological education. The very first thing which comes to mind is that when we talk about that next gen, where is the need for next gen? Is our current system suffering from certain limitations, certain problems that we need a next gen education? Or is it a buzzword? Is it a fascination? Let us go to next generation. I mean, is there really a need for that actually? Let me first go to the, maybe, I don't know, the youngest of the panelists, Urvashi Koharji, that can you just, let's answer this question to the audience first. What is the need for this next gen education?
Does anybody in the audience, especially the young students, want to tell us why do we need to, call it a next gen, sort of an innovation? Why not just let it stay as a, general term and talk about innovation per se? But why do we need it to be more advanced? Is it not good enough? What is your take on this? Students, kuch bhi, whatever comes to your mind.
I feel the students should be participating, and the best participant can have some prizes also.
Yes, we hope that you are you make such an impact that we are bound to give you a prize.
Okay, let's set the ball rolling with you. And
sir, when you speak of the need, why is it to be called the the next generation or an added level? Is because we are continually looking for innovations, and the only thing that stays constant in the world is change. We need to be constantly adding newer things, not only for an interesting edge, but to advance things, to improve them, enhance them, perhaps to cater to another need, plus one. When we talk about the use of AI, because I'm going to talk about an educationist point of view also, AI has enhanced even the work of teachers like me in terms of keeping tab of something as simple as attendance. I'm probably not very convinced about carrying big registers where I keep attendance, and students come back to me and say, ma'am, meri attendance badha dijiye, and I'm probably in a space where I get influenced. Let me do it electronically. Let me do it right now in the real, what we call real time. So it helps you, it adds to administrative efficiency also, and students can also see their attendance real time. And apart from that, I can generate tests, I can schedule reports. There are often times it reduces a teacher's workload and a student's workload also. If I can assign homeworks right there and then, and I can give you a timeline. And in terms of experiments per se, if students have experiments to do, it can be assigned a timeline after which it is not to be submitted or not to be accepted. And also to add to it, AI and shapes up or aids to professional development. How it does that, it provides us with AI recommends a personalized and professional development resources, which seemingly, if you ask me, I will not know at the top of my head as to what to recommend you, what kind of course to do best, or what kind of training to enhance your skills as per your personal requirements. But AI does that, and it doesn't mean that we observe AI on a blind basis. We also keep our intervention or diligence in mind. Yes.
Point is taken very well. I think we can come back more to AI later
Yes.
on. I think when we started this conference yesterday, we emphasized on that. People are talking about new generation techniques, new research, but AMI in this particular conference decided, Dr. Kohar and myself and others, we decided that it should not be just restricted to research. Let us talk about next gen education also. Government has placed new education policy 2020 and it is working now. And can we see or can we say that the new education policy, is it the next gen education? What is your take, Dr. Charu, on that? Do you think that it is going to be a next gen or we have to add to those things, the new education policy, to improve that?
Yes, a very relevant question for today's scenario in education. First of all, what I understand by next generation education and why it's called next gen education is, if we go by genetics, then next gen is basically when the genome of both the parents combine together and have some kind of innovations and some variations to give rise to the next generation. And I think the key is this, that we need to keep the good part of what the prevalent system is and innovate and introduce variations to adapt to the changing environment. So we cannot deny the fact that the new generation is completely changing because of the advancements in the digital technology as well as AI, etc. So coming back to the new education policy. So new education policy fundamentally aims, as far as microbiology education is concerned, to basically move from the petri dishes of memorization to basically the understanding of concepts and the critical thinking, etc. So when we talk about these things and the particular aspects that are laid in the new education policy, we want our current students to be more equipped with these things rather than the typical rote memorization of just the facts, the names of various bacteria, viruses, etc. Rather than what they need is the functional, or as Kohar sir mentioned in his plenary dialogue, about the application of the microbiology. So NEP lays down many key parameters which aims to address these kind of thinking. So the critical thinking, the conceptual understanding, and with the inclusion of the digital technology, AI, etc. So definitely NEP, I feel, has laid down a very, I would say, a comprehensive as well as precise framework for what the next gen education in general and microbiology education in particular should be. So as far as the additions are concerned, I don't think at this stage, because it takes a lot of time and effort to lay down such policies, and it comes in years, it's not just a matter of some months that the things can be changed. But when you apply a policy, then definitely based on the kind of changes that is bringing up in the society as well as the feedback we are having, we can certainly modify it or tune it to the needs so that it can be adopted in a good manner. But as far as the policy is concerned, I think it is very, very apt for the next generation if executed properly.
Right. Anytime any student may want to add something, you're free to add. in any case, we have Mr. Rajesh from High Media with us. And because you are from a company who is representing a High Media, and you are so welcome into this panel. And for this, I would like that the Government of India has placed NEP 2020 in place with the mandate that they should produce more of the students who are startup oriented, entrepreneurship oriented, innovation oriented, rather than, as Dr. Charu said, rote learning, or rather than just producing the people who are seeking jobs. You are from the industry, and you think that, what is your point that the education, next gen education, when we are talking about the next gen education system, particularly microbiology, because High Media is very much involved in microbiological work. So when we talk about next gen microbiological education, how do you think that the next gen education should be adopted in schools and in colleges and in universities?
The NEP, what I understood and what I have been seeing in last 2 years in field also, that students, lot of students are visiting our lab. They are asking for, sir, whether we do conduct training programs or not. I think NEP has mandated at least 120 hours of industrial training, which I think is really a very good move.
Closer.
Which is really a good move, but as ma'am said, if it is not executed well, then it can also go in vain. I'm also seeing a lot of students, they are looking for the training programs here and there. In NAP, I think it has also been instructed to colleges and universities that there should be dedicated internship cells should be there, which should help students to find out the internship programs. Again, execution will be very important. Then only the objective will be served. I'm talking about the training, industrial training. What we see is whenever we are recruiting new employees, new staff, the freshers, so-called freshers. So what we see, they're not like we have been hearing in recent times that a lot of basic, majorly IT industries, they say that we are not getting employable, the freshers are not employable. That's, I think, but in other industries also, like in microbiology students, which we regularly interview for recruitment. So we are seeing the basic concepts itself is not, no, that's what my experience is, because for last 2 years, I've been doing the interviews whenever it is required. So what I have come across is, because I'm placed in Nagpur, and we get the these CVs from local colleges and the nearby universities. So what they are complaining is, the major of our syllabus is covered by the faculty who were senior to us last year. So this particular problem I have been seeing, which I feel if NEP can address that, that will be a very good thing to address the issues. Then come back to industrial training. So this in 120 hour minimum industrial training, if executed well, definitely there will be very good results we will be able to see in future. Then I think on the other side, industries can also be, at policy level, they should be instructed kind of, maybe now it is CSR is the compulsory for all the industries. Under that CSR fund or whatever it is, they can be, it can be place like a policy for all the industries, that it is mandatory for you to say, for example, based on your total revenue or whatever is the industry size, so many students have to be trained every year from the so and so industry. So those kind of policy, I think, will be helpful along with the NEP.
I think many of the higher educational institutes, they are trying to put this in place, that 120 hours mandate, it should be executed properly.
Yeah.
But how far is the industry receptive to that? They are also cooperating with the higher education institutes to take and fulfill these 120 hours.
They, based on the funding is the basic, the criteria what they consider whenever they are considering the interns, because behind every intern there will be a cost. So that's why I mentioned the CSR fund. So because it is mandatory for all the industry to spend some fund from their revenue under CSR activity, if for this particular thing, that this 120-hour internship, if that come under the CSR activity, I think that will be helpful.
Okay.
That's my point of view.
Okay, so we'll come back to that because CSR is an important part
Yeah.
of from the industry education interactions actually.
Yeah.
Right. Okay, so we'll come back to you, Dr. So now we move on to Professor Kohar. That one of the important criteria in NEP is when we talk about new education policy, we talk about next-gen microbiology or next-gen science, next-gen education. How important it is and how far the higher education institutes, they are doing it, the overhauling of the curricula and benchmarking of the curricula, and how much it is important, how much it is direly needed, according to you.
Very important, Professor Prince Sarmaji. And we need to understand, because everybody now is well aware about national education policy and its importance, and have felt very seriously that yes, we should develop the curricula as per NEP, you can say, aligning with the national education policy. And it is very, very important that simply developing the syllabi is not going to serve the purpose. It's very, very important to develop the curriculum. That's a system. That's an educational system, while syllabus is simply a kind of roadmap. One need to understand the difference, say, between these two. And when you have to develop the curriculum, of course, I'm taking the next gen also side by side, because when we develop the curriculum, we need to focus on more and fast delivery, more knowledge, more training, more skill, and how fast we can give more knowledge, can train our students, can make them skilled, can make them employable. That's really very, very important. What are the new technologies which can help in improving our teaching learning methods, pedagogies we are talking about? What are the different kind of the technological based aids are available so that we can improve the learning of the student, understanding of the subject, understanding the kind of training you have been giving. Also, it's very important for a teacher to decide about the various ways, various methods by which you can improve the competitiveness, the ability and the capability within students. And then equally important, I must say, that you need to define about the evaluation system. And all throughout, whether you are deciding about the teaching learning methods, you are deciding about the additional aids in order to deliver your teaching material to the students, training, or the methods you have been using for improving the competitiveness among students. All those evaluation methods should be based on the whole strategy, starting from pedagogies to the improving capabilities and training program you have been providing. Because it's very, very important that you are following different methods, different ways, and you are evaluating in a traditional methods, that will not serve the purpose. Therefore, it is very important. And the modern technology, whether new generation sequencing you talk about, artificial intelligence based techniques, machine learning, in order to enhance the speed, rate, because now you see the world is moving very fast, very rapid. There is demand, like if you talk about the environmental deterioration, it's, I think, very fast. Diseases are spreading very fast. There is an energy requirement, it's a big energy requirement, because by 2050, this oil reserve will be exhausted. Likewise, you take every field, so you have to speed up, because by the basic microbiology techniques, we have been able to sequence, say, few genes. We have been able to analyze, say, few microorganisms, but that's not going to serve the purpose. That's not going to give us the kind of product we need for, say, the market and the society. Therefore, we certainly have to go for next generation technologies which are available. And accordingly, we need to include all those sort of, say, the new gen technologies in order to modernize our curriculum, modernize our content, text, and modernize our methods of dealing with the teaching learning system and the research. So I think we need to, and Professor Prince Sharma has just mentioned about that benchmarking. It's very, very important. The curriculum we have been developing, it should match to the global standards. And the initial step, according to me, should be that, say, for example, there are 18 universities in Dehradun. Dev Bhoomi developed the curriculum, and it should match the parameters or the standards of other university in Dehradun, and then all universities of Dehradun within state, and then national universities, and then, of course, globally as well. Therefore, because the disparity of teaching learning methods and evaluation methods needs to be minimized, and you only can minimize and you can equalize your standards of education, your degree equivalence to the global universities only through this benchmarking, which is very, very important. And I would also add here that Academy of Microbiological Sciences and the Association Microbiology of India, they have together worked involving near about 100 microbiologists from this country, and has developed the complete benchmarked, say, the curriculum of undergraduate and postgraduate microbiology, and by January or February we would be able to make it available, say, those who are interested. Thank you very much, Professor Prince.
Yeah, thank you, sir. I think that was an elaborate answer that we really need to overhaul and benchmark our courses according to the needs, current needs actually. Dr. Hari Prasad, so you are a scientist. How important is the research to be integrated into this next gen education or microbiological education?
So, thank you. I think this is what I was expecting also. And I am quite well. So I'll start a bit discussion on NEP. So if I look the NEP 2020 curriculum, I divide broadly in three parts. Look it. The one that NEP 2020 has brought one component that our graduate students will have better personality growth, you know, the initial component of 1 year or 2 year. Then the second component which I see, the last year, that is 4th year, which has been included as a dissertation and project.
Yeah.
Now look at it's always happens that whenever a policy is being written or implemented, lot of thoughts have been given, you know. This cannot be a obsolete kind of thing or irrelevant. But the problem comes between the two, the one who is implementing and the one who has given the thought for formulating the policy. The people who actually implementing, they will always have tough time to ditto implement whatsoever policy has been written. Let it be any policy, NEP 2020 equally good. But whenever new thing happens, this kind of dilemma is always there, whether it is education or anywhere. But over a period of time, the things will get materialized and, you know, systematized, and then this will become one of the wonderful things. In my opinion, NEP 2020 has been well thought by looking to the vision for in the 10-20 years, not right now. Now coming to the research component, I have also seen quite good survey in this country. You go to the degree colleges, they are also thinking to bring out the NEP 2020 4 years program. State universities, you look it for them, they obviously they are under pressure one way or the other to implement. Central universities, institutions, they have no problem. They can, in a period of time, they can actually bring it out, the 4th year fulfillment of NEP. But mind, these are the underprivileged places where they don't have in 10,000, 20,000 rupees, even the petty cash to run their program. How will they be able to bring the research program which is needed for NEP 4-year program of NEP? So that's the biggest challenge, which I am sure we all are aware of it. We are working how to overcome. Now research has to be defined, precisely the question. Under this situation, when we are working in a premier institution where crores money is there, our research model could be different. But the places where we don't have enough resources and we are now under NEP 2020 to implement the research, that's one of the requirement, probably to make our graduates better equipped for the industry, for the science, for the growth. There we have to remodel the thinking of the research. Okay? The research should not be considered that, okay, if you don't do genomics, proteomics and everything, you are not doing the research. So research has to be picked up. It has to be redefined, and that is the responsibility which we older generation will have to do for the next generation education. More precisely, if I come to the microbiology, I wanted to take it now in my first round. Look at today when circular economy, how important microbiology will have to play the role in circular economy. Lot of waste are being created, whether it is lithium ion batteries being dumped, you know, in the generation, whether it is electronic waste is coming. And if our students are taught the model of research, that how are you going to handle to isolate microbes, etc., and work out if some of those microbes can be useful to actually take care of these pollution, etc. Again, I am bringing to the point. The bioremediation is the area of science which I am seeing for last 40 years in the field of microbiology. It's not that people were not doing, but that is another domain where next generation will have to be thought of. The question came, why do we need next generation? I am actually coming to that. Ours was the era, I am sure we all are quite old people sitting except 2 or 3 of you. So ours was the era where we were rushing in the academic science and everything on certain different type of mindsets. We have grown today much better in terms of economy. Let's accept it. We don't have to feel shy about it. This next generation, if they are taught how to do the research, that was my topic, then possibly they are not that much insecure in terms of the earning bread and butter, because we have grown economically. So if these people actually they are being given a model of the research, that how to conduct the research, let them generate one piece of data in 6 months time with the limited resources. But if on that piece of information, if they can stand in the future, then that is what this generation can do. So microbiology education in terms of the research, in my opinion, which is the platform we are discussing, I think we need to reach out I was telling to Professor Kuhar sometimes back that whatsoever he had spoken, I think we were not around probably that time, the different points, all these points will have to be elaborated, written in precisely in the present context of research and education, and reach out to those people and say, can you follow this? Then you will grow better in the research. That's what I said. So next generation in microbiology actually can do wonder than what we people we are doing, because of the security they have better, the information available, internet, lot of things, AI, etc., which is being talked about. So that is, I think, in education, next generation has to be a little more precisely taught, and we bear the responsibility to reach out precise, right education model of doing the research, you know.
I think the point is so well taken, Dr. Hari Prasad, that research has to be a good component of the education. And particularly when we talk about, you have taken the example of bioremediation. We have been doing bioremediation for the last so many years, but maybe the next gen education with newer tools may make this bioremediation much better. In the yesterday's talk, I was discussing about that. Now people have generated through AI the E. coli strains which can capture carbon dioxide from environment. So that's what the next generation can do. With the, I mean, that's possibly the wonder of combining the new gen research with the new gen education program. I'll just coming to that actually. I'll come back to you, sir. One more. We are not just finished with this. So with the first round of this panel discussion and the questions, lot of questions might have been coming to your mind. Anybody is having any question, or anybody having answer to this question, how far you are satisfied with this new education program? Anybody? You can come with the answers anytime while we are moving on to the next round of questions. That brings me to again Dr. Urvashi Kohar. And again, I will say that we are talking about next gen education. We are talking about new education policies, new education programs. How far our teachers are equipped for that, and how far the teachers need training and retraining so that they can train very well our new generation students.
Sir, first of all, I'd like to applaud your skills at actually reading my mind. I don't know how you got to know, but I have the same question. I wanted to share the same thought at how equipped are our teachers in colleges to train these students for research, because of course they've all carried out research at their MPhil, which is now obsolete, but they had carried it out nonetheless. I'm one of them. And also PhD research, but you know, for an undergraduate student to be in the 4th year and for them to understand what research is, and for us teachers to tell them what it is and what is to be done, is a very, very challenging prospect which I am facing nowadays, because this is the first batch that we are going to deal with, which will carry out a thesis, and for them there is no such training. I think I'm of the mind who would say that there should be a training for teachers also to how to guide these students in the 4th year, because they are not MA level, they are not MPhil level, they are definitely nothing closer to PhD level. So how do we deal with their competencies, and we should not keep the same parameters for them. हम लोग एक ही मापदंड लेके नहीं चल सकते कि ये स्टूडेंट्स हैं और ये रिसर्च करेंगे और वो किस लेवल की रिसर्च करेंगे. We should also be kind to them and probably kind to us as well. So this is a question that's been rummaging in my head, and I've been wondering how will I deal with this, and this is something that I'd be very grateful if we can find an insight into.
I think in my viewpoint that teachers also need a constant learning. Teachers also need a constant revamping through the orientations, through the workshops. They need to learn actually. I think a good teacher is who is always 2 steps ahead of the students. Otherwise he will not earn the respect of the students. So that is my viewpoint. Anybody can put his or her viewpoint at any point of time. We're having a general discussion.
Is there a teacher also who wants to suggest how these trainings can take place?
There is one, okay.
I could hand you the mic if somebody can.
I'm tired at this moment because jet lag problem, but I would like to share my
briefly, sir.
my experience about the education. Let me introduce myself. I'm Rajesh Sani. I have been teaching in the School of Mines for last 20 years, and I'm in US for last 26 years. And I'm a full professor and doing lot of experiments. So in 2017, I'm just telling you a simple story. In 2017, I proposed that I want to develop a pyramid approach for my research group. What is pyramid approach is, you have on top main supervisor or industrial advisor or program managers, then you have postdocs or junior faculty, then you have PhD students, then you graduate students, then you have undergraduate students, then you have high school students. So I propose, like my postdoc will train my PhD and master students, and my master students will train the undergraduate students, and undergraduate student will go to the high school and will do the outreach activity. So this worked. I did that experiment for 7 years and published last year that pyramid approach. It has been a successful, and like the good thing was about that, we could bring the undergraduate student early in the research, and they could learn, and and then you could ask, okay, you are a supervisor at the top, who is your supervisor? So that question came to me. I said my supervisor is my graduate students because I work very closely with them.
I think this is a very good point you have taken. And in the new education policy also, in the 4th year, those students who take up the research, they can go directly for PhD without doing the masters also. And Dr. Yogender wants to say something?
Yes, 4-year college can directly go for PhD, but although I find that Delhi University is blessed that many colleges do have projects, some people are already doing research, but there are a large number of colleges all over India. People don't have facility, space, money to buy things. I mean, unless you have a space, how will train large number of people? Because those numbers are not very small. Also, facility, money to buy small things. So I think all those things people have to, or government or funding agencies and UGC has to think that how to run these programs.
Yeah, that's a very pertinent problem actually which is coming up, because in the smaller areas, no funds how to do the research in the 4th year particularly. They don't have any infrastructure or so. Now here, I will just come back to Dr. Charu, but before that, can there be an industry step into this? You had the CSR things. Can they help the smaller institutes, education institutes, that research can be, or infrastructure can be given through the CSR programs, or industry can handhold them for doing this?
I think it is already there in place, sir, because I think 2 or 3 students, they are doing PhD, collaborative PhD. They are registered in ICT, Mumbai, and they are, the place of work is High Media, Thane Lab. So this kind of setting is already there, I think. But it should be, as I was saying before, it should be made compulsory for based on the company size. Say for example, if whatever is the number, 10 student has to be enrolled under industry, academia, PhD program. Those kind of things if come in scene, that will be better, I think.
So maybe that, in your point of view, that one of the, maybe the answer to this is that if industry can handhold such smaller institutes or poor institutes who cannot afford the research, if they can through their CSR programs can handhold them, maybe, I don't know if it is possible, it can be done, because some of the industries, they are doing the CSR programs also. But besides that, if I come to Dr. Charu, that how do you think that these research funding schemes for colleges like DBT Star College or this math, they can better support this kind of research?
Sir, absolutely.
Because there are methods.
Absolutely, absolutely. Before I just address that question, I've been waiting to just mention certain things very briefly, which is my experience basically. The last discussion ended with the discussion on research in education, and the first it started with the teacher training. So I would just like to put up an example here that under the PMMMT funding from Government of India, ISER Pune under the able guidance of Professor L.S. Shashidhara had been conducting these teacher training programs on research-based pedagogical tools since 2015. And it's basically training the teachers, and then the teachers go and train further the teachers. So there are certain teachers that are, pointed out.
Dr. Sani was also doing it.
Yeah, so I myself have been a part of like 2 remote areas in Bihar and everywhere, where, as sir said, research is not just doing the experimentation or anything, but research is an attitude. It's basically, when we talk about science, it's the curiosity and exploration that's goes into it. So what we do is basically just small topics we change into the approach of research. So where they talk about like, identifying the problem, creating the hypothesis, identifying the dependent variables, data collection, etc. So it's a research process that is taught, so that when they go higher up in the higher institutions, they are at least known about the concept of research and when they get the resources. I'm not denying that, of course, the government should work at the root level to provide such infrastructures at the remote areas as well. But that's the key concept that has been, done, teaching the research process to the masses, so that we just don't create, knowledgeable microbiology knowledgeates, but the thinking of microbiologists that they should adopt. Now, coming back to your question, which is about the funding, I cannot praise enough the DBT Star College scheme, because I've had that first-hand experience. I was the coordinator for a lot of time, again under the able guidance of Professor Sukanya Lal in our college. And I must tell you, the kind of funding they provided, we could provide to the students the smaller projects, the training, we could have the equipments which we could not have because of the subsidized fee structure that the colleges have. All we do the research or science, there is a very small funding from the government, but we basically do it from the fees. So the infrastructure fund is again taken from the school, the students. But the problem is again the subsidized structure. We cannot raise the fees. So such schemes of Government of India which aids the incorporation of research aspects at the undergraduate education are so much welcome. And not only I am saying, because I attended a lot of DBT Star College programs, the meetings and everything, many colleges in all over India have gained a lot of things, not just infrastructure, but knowledge base because of such schemes. So I am a very big pro for such schemes, and I would always want the Government of India and to encourage them to give such kind of funding to the undergraduate colleges, so that the implementation of the NEP can be done in a very beneficial manner for all the stakeholders.
Right, that's very well said actually. And I will ask Dr. Kohar that now, continuing on this, how far we both are on board, AMI and academy can handhold them, and at least not, if not, there are so many colleges, and at least a few of them, if that can handhold them and can them give the flavor of the research, and can give them a how the research is done actually, and what actually the research is. So can we play a role in that actually, or just we are talking about this? It's not just talking.
Yes.
First, it's very, very important that research needs to be integrated in our undergraduate program. Even if it could be integrated in school program, that's a matter how and how it should be, but it's very, very important. And University Grant Commission has already issued the guidelines to make a provision for research internship as well, in addition to the research component integration, which is very important. And the kind of challenges has been, shared by some of, our colleagues. It's very right. Those problems are there. Training is also equally important. But as far as the different associations, different academies' role is concerned, it's very, very important. And like INSA, like NASI, certainly Academy of Microbiological Sciences, the Association of Microbiology of India, I think should take a lead. This is the high time, and we should have a plan. Take our microbiologists from different parts of the country on board.
All our members and units?
Yes, our members and our units. And I think we should develop a kind of database and a full scheme. And the association can have its own portal. And we can make a provision and the process so that the students who needs internship, they can register themselves. And of course, we would be having the data of the institutions and individuals. And certainly, I think this is a very novel idea. And it is a kind of house. We consider it's a general body of Association Microbiologists of India. And we should take up, this scheme. And students, good luck to you. And the AMI president is here. He has accepted it, and we will develop a scheme very soon.
I have floated an idea, and I have accepted it also because of Dr. Kohar. And if he agrees to us, and then we can float it, and we can write. So I think we are trying to approach this problem of funding, which Dr. Yogender had raised through some means like DBT stars or like CSRs or like AMI's programs. So if we can.
I can add here, if we do have a system, then we can approach certain industries and they can fund us through this CSR as well as...
We can be a bridge between the industry and these.
Ministry of Education, MSME, and couple of more ministries which I know that they have been giving funds to different academies and associations. I think we need to start.
And one more thing, Dr. Kohar, I was thinking that while we were talking about overhauling the courses and benchmarking them, how much do you think that these industry people, new startup people should be a real integral part of this when we are making these courses?
I think it's very important. Otherwise, we won't be able to achieve the real goal of the curriculum development and, of course, that of new education policy. It's really very important that these startup experts, those who have very good knowledge about that, they should also be, I think, involved not only in our curriculum development, but in, universities or higher education institutions should involve them as professor of practice in teaching program as well, in research program as well. And that's the real way you can bring these, the industry to the academia and academia to the industry. Otherwise, when I was student, we started hearing that industry should come to the academia and academia should come to the industry. Oh, today this conference is a platform for bringing in, industry people and academia. And I could not see much of the progress, except like in the last 5 years, we could see some industrialists, they are coming forward. Otherwise, there is no success. And unless until we don't, make up our mind, it's only mindset. Because we are not ready to give even the visiting professorship or a guest professorship to an industry person.
Yes.
So how he will come to you? the institutions have to think twice about it. It's very serious matter. And it's very, very important. If really we have to make our NEP-based goals achievable, we will have to follow this, say, practice or this scheme, which is very important. And that will be very, very beneficial, say, for the students.
I think that's a very important thing. If we can be a bridge, or associations like us, others also, we can be a bridge between these not so much funded institutes and the industry.
We can have such kind of cell as well.
Yeah.
It's a, say, some cell of
some
say,
cell,
association.
Yeah.
So that there could be a, like industry people and our say,
Sure, sir.
researchers.
Yeah,
and
sure, sir.
That
Yeah. So, Dr. Hari Prasad ji,
Mishra, not Prasad.
Hari Mishra ji, sorry. Dr. Hari Mishra ji, again coming to that, government is now very much inclined on when they are opening the higher education institutes, it should be not just a higher education institute, it should be the multidisciplinary higher education institute. The words like AI, that's one thing. The words like high tech is another thing. But what is getting more impetus now is multidisciplinary higher education. What is your take on that? That how much the government is successful and what will be the fallout of this?
So glad, something which I have been thinking to speak.
I think I'm reading the minds of everybody.
Wonderful, wonderful, because we are almost the same resonance in the.
I think
so. So another important thing which, next generation microbiology, that's a topic of this discussion, I don't want to deviate it. The purity in the discipline is the yesterday's thought, something which you are saying. If a microbiology student thinks that I'll keep doing only microbiology, then sooner or later you are going to be faded out, from the stream. You will not be able to survive. So this 21st century, in my opinion, as the question is asked, is actually a research domain of multidisciplinary and interdisciplinary research. I'll give an example. Let's say a microbiology student has found a couple of bacteria isolated from the region. And the question is that we want to test if these microbes can actually remediate the lithium-ion battery. I'm again bringing a small component. Now we don't understand what is the composition. We cannot understand the composition of the battery and what are the components are there. So to be able to successfully test it, you need to move out and talk to the people who are actually expert in the domain. This is what called multidisciplinary domain. So the next generation should also be thinking to incorporate the mind in their thinking process, that they will not be sticking to only purity in the research and in the science, rather they will go for multidisciplinary component. That is something very important. And another thing, if I can comment it, though that's not direct question to me, it is not that economy, our fashion, our thinking process is growing and what Western world and rest of the world was growing for 30 years and 40 years ago. We are growing almost everything. Our science and research model is also growing in that direction. Look at, if you go to the Western world, Europe, America and all these places, the research funding is not coming purely from government establishment. Lot of, philanthropists, industrial people, they are actually contributing in the research. And I believe, though I have not tried so much, if industry friends are sitting over there, in India also that kind of thought started actually coming up. At least I have spoken a couple of them, I don't want to name it here, that they are willing to come and fund for our research, particularly keeping in mind that NEP 2020 fourth year curriculum, if has to be met at all level in this country for integrated growth, then research funding will have to come. So many of them, they are also started thinking that yes, if a project comes of the importance that will benefit to the industry also, then they are ready to fund it. So all our people who are in the particularly, I think I will also make a comment, if we all are expecting that government establishment will give the funding to the colleges and state universities for running NEP 2020, you have to wait several years before this dream can be realized.
Yeah.
Today
Okay.
also the system is there. Give me 10 seconds more. I am sure many of you know, those who are in the affair, that you get nearly 50,000 times more number of applications for one opening in the funding. And how much government can fund it? Filtering is still being done. I don't feel shy about, though I am from a premier institution, the filtering in this country for funding is also being done for those who are, white collar institutes and the top institute of the country, because we go for quality assessment of the project, and that obviously with the history will come from them only. So the funding coming to the underprivileged institution in this country, which is needed to actually implement and realize the mandate of NEP 2020, is going to be very difficult unless we walk out from the lab with the mindset that we'll not be banking on the government body, rather we'll approach to the industry with a written project. If a professor has worked, as Dr. Saini said, for 30 years and 40 years, I am sure you will find one particular component which you can propose by joining the chemistry, physics and engineering people. Go to the industry and you say, give me 20 lakhs, I want to give you the product.
Yeah.
If that kind of mindset starts growing, then only our funding opportunities will come, unless industry comes and fund with the objective clarity in our mind and their mind that we are going to benefit. It's not going to solve.
It's very true, very true. I have been asked.
I would like to add just one thing.
Okay, sir.
Because when we are talking about next generation microbiology itself, it's a multidisciplinary. Even I would say that it is transdisciplinary.
Yeah, it is no more in silos. It is microbiologist. As a microbiologist, if I have a developed thing, I don't know how to market it. Then what is the fun? It will be lying in my lab actually.
Very important.
So with this, we are coming to an end of this particular session. But I have a question again to the students. Students have been listening to this. How many of you want to go for research? That research should be a very good component of your education. How many of you think about this? Raise your hands who think that research should be a vital, important component of the education. Only a couple of people are there, because now there are some people, because I think that that research component can take you to the industry, can take you or make you learn how to go for startups. Or how many of you have a thought about that? After my education is completed, I will go for a startup, not for a job. Anybody? Yeah, I'm happy that a few hands have been raised that. And slowly, the number of these hands will increase with the time if we are putting this NEP 2020 or next gen education in place in the right manner. As Dr. Charu said, we have to put it in the right manner. So thank you so much for listening to us. I think it could have been a good eye-opener. Thank you so much.
Thank you so much, sir. This was definitely a very informative session for all of us. Now may I request the Registrar of Dev Bhoomi Uttarakhand University, Dr. Subhashish Goswami, to kindly come up on the dais for the felicitation ceremony. And may I request, sir, to kindly felicitate Dr. Urvashi Kuhar. May I request sir to kindly felicitate Dr. Urvashi Kuhad. I request all the panelists and the moderator to kindly stay back for the felicitation ceremony. Dr. Charu Dogra Rawat. Professor R.C. Kuhad. Dr. Rajesh Pal, who is a representative from High Media Labs, our sponsor and industry partner for the conference. Professor Hari Mishra. And our moderator for the session, Professor Prince Sharma, President AMI. Thank you so much, sir. Thank you, Dr. Subhashish Goswami. Thank you, sir. Moving on with the event, we now move towards the most exciting part of the day, that is the cultural night. So with this, I request our anchors to kindly take over and invite our very talented students of Dev Bhoomi Uttarakhand University for the cultural night of the conference.
A very good evening, ladies and gentlemen. With immense warmth and delight, we welcome you to the cultural evening of International Conference, jointly hosted by Association of Microbiologists of India and Dev Bhoomi Uttarakhand University, where tradition meets talent and culture embrace the creativity.
Indeed, Ritunjaya. Tonight is not just an evening of performances. It's a celebration of diversity, unity, and the spirit that binds us all beyond the boundaries and nations. I am Gunjan Joshi, and alongside me is Ritunjaya Arya, and together we shall be your hosts for this memorable evening filled with rhythm, grace, and color.
On behalf of Dev Bhoomi Uttarakhand University and Association of Microbiologists of India, we extend our heartfelt gratitude to all the dignitaries, delegates and faculty members and our international guests who have joined us this evening.
As the academic exchange of the conference transitions into this cultural festivity, let the melodies, movements, and moments of tonight reflect the beauty of our shared heritage. Please give a big round of applause for all the participants and audience. So, are you all excited for tonight? So, welcome to our much awaited cultural night, a night where creativity meets celebration. We are here to showcase the amazing talents of our friends, classmates, and the performers who have poured their hearts into every act. So, let's cheer out loud, clap harder, and make this night one to remember. Each performance tonight reflects the beauty of our culture and the dedication of our participants. Let us begin this evening of elegance and entertainment.
कंटिन्यू कर दे ये वाली स्किप शुरू कर दे ये कनेक्ट करना है नीचे कहीं बोला है एक ही है उस पे को दे रहे हैं वो दे रहे हैं
यही होगा
इसको बंद करो पहले ये लगते रहो मैं लगते रहा ना ऑन कर दो
Till the stage setup, I hope that you will show the patience because the upcoming lights are on, the energy is high and upcoming performances are really unbeatable. Thank you for being a patient audience. Ladies and gentlemen, we request you to please be patient for a few moments as our team prepares the stage for the next performance. Great things take a little time, and we promise the upcoming act will be worth the wait. Thank you for your kind cooperation and enthusiasm.
So the wait is over. Among us sits a person whose hidden talent has been the talk of this evening. We have received so many requests for him to take the stage. So without any further delay, let's welcome the one and only Professor Naveen Gupta to come forward and showcase his hidden talent.
Please give him a big round of applause.
Oh my God. हेलो गुड इवनिंग टू ऑल ऑफ यू। ये एक प्रोफेसर को लेक्चर देने के लिए पहले बुलाना तो ठीक है, एक प्रोफेसर को पोइट्री बुलाने के लिए भी पहले बुलाना, ये थोड़ा नाजायज हो गया। हेलो एली, आई एम ए प्रोफेसर और साइंटिस्ट बाय प्रोफेशन। पोइट्री मेरा पैशन है, तो आई राइट उर्दू हिंदी पोइट्री। तो कुछ अपने लिखे हुए शेर आप लोगों को सुना देता हूँ, क्योंकि हम माइक्रोबायोलॉजी की कॉन्फ्रेंस में हैं और इनॉग्रल सेशन में डॉक्टर कोहार ने, डॉक्टर प्रिंस ने, सभी ने ये जिक्र किया कि माइक्रो ऑर्गेनिज्म सब जगह मौजूद है। तो मेरे को ये नहीं समझ लगता, जब हर टाइम अपने साथ कुछ कोई है, फिर हम अकेले कैसे हो सकते हैं। तो मैंने ये नज़म लिखी, जब कभी तुम्हें लगे कि तुम अकेले हो, तन्हा हो, कोई तुम्हारे साथ नहीं। जब कभी तुम्हें लगे तुम तन्हा हो, अकेले हो, कोई तुम्हारे साथ नहीं, तो ये याद रखना, लाखों करोड़ों अरबों सूक्ष्म जीव हर वक्त तुम्हारे आसपास तुम्हारे साथ है। तुम कभी तन्हा हो ही नहीं सकते, तुम कभी अकेले हो ही नहीं सकते, तुम कभी अकेले हो ही नहीं सकते। तालियां मार दो यार। इसके अलावा बिना माइक्रोबायोलॉजी के मेरा एक सिग्नेचर शेर है कि कुहार सर, हार कर खुश करने की तरकीब बना रखी है, हार कर खुश करने की तरकीब बना रखी है, ये जो मैंने जीतने की बच्चों से शर्त लगा रखी है, दुनिया का सबसे अमीर शख्स हूं मैं, मैंने बटुए में बेटी की तस्वीर लगा रखी है।
हेलो 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 ठीक ठीक हेलो हेलो,
हार कर खुश करने की तरकीब बना रखी है, ये जो मैंने जीतने की बच्चों से शर्त लगा रखी है, दुनिया का सबसे अमीर शख्स हूं मैं, मैंने बटुए में बेटी की तस्वीर लगा रखी है। और बेटियों के लिए एक और शेर मेरे को याद आ गया, मेरी लिस्ट में तो नहीं है, कि बेटी मंदिर की मूरत मस्जिद के कुरान सी लगती है।
हेलो हेलो,
बेटी मंदिर की मूरत मस्जिद के कुरान सी लगती है, हंसी उसकी आरती की घंटी नमाज की आजान सी लगती है। हंसी उसकी आरती की घंटी नमाज की आजान सी लगती है। एक शेर है कि मेरे को अब 30-35 साल हो गए पढ़ाते हुए, कई बार लगता है बच्चों को जो पढ़ाना था, भाई पहले मैं ही बोलूंगा अब,
हेलो
कि
हेलो,
बच्चों को जो पढ़ाना था शायद कुछ हमने पढ़ाया ही नहीं। तो मैंने एक शेर लिखा कि
हेलो,
पेंसिल पकड़नी सिखाते सिखाते,
वही बोल रहा, चालू है।
दिमाग से पढ़ना सिखाते सिखाते किसी के दिल को पढ़ना सिखाया ही नहीं। दिमाग से पढ़ना सिखाते सिखाते किसी के दिल को पढ़ना सिखाया ही नहीं। पेंसिल पकड़नी सिखाते सिखाते किसी का हाथ पकड़ना सिखाया ही नहीं। पेंसिल पकड़नी सिखाते सिखाते किसी का हाथ पकड़ना सिखाया ही नहीं। अब बच्चे कहेंगे ये क्या सीरियस सुनाई जा रहे हैं, आपके पसंद के कुछ शेर की एक शेर है कि देखता हूं तेरे माथे की ये जो बिंदिया तो यूं लगता है। थोड़ा बारीक शेर है, जरा समझने की कोशिश करना, मैंने 2-3 दिन पहले ही लिखा। देखता हूं तेरे माथे की ये जो बिंदिया तो यूं लगता है जैसे मंदिर की चौखट पर किसी पुजारी ने कुमकुम से ओम लिखा हो। समझे नहीं? देखता हूं तेरे माथे की ये जो बिंदिया तो यूं लगता है जैसे मंदिर की चौखट पर किसी पुजारी ने कुमकुम से ओम लिखा हो। कुछ शेर और जो याद आ रहे हैं कि जिंदगी में मोहब्बत कई बार की, जिंदगी में मोहब्बत कई बार की, एक ही शख्स से हर बार की। जिंदगी में मोहब्बत कई बार की, एक ही शख्स से हर बार की। और एक शेर ये है कि मैं इसलिए भी अक्सर उससे मिलने नहीं जाता, मैं इसलिए भी अक्सर उससे मिलने नहीं जाता, गले वो नहीं मिलता, फासले से मिलना मुझे नहीं आता। मैं इसलिए भी अक्सर उससे मिलने नहीं जाता, गले वो नहीं मिलता, फासले से मिलना मुझे नहीं आता। और एक गजल के कुछ शेर ये हैं कि वो दिन मोहब्बत की कहानी याद है मुझे, वो दिन मोहब्बत की कहानी याद है मुझे, वो सारे का सारा मुंह जुबानी याद है मुझे। वो दिन मोहब्बत की कहानी याद है मुझे, वो सारे का सारा मुंह जुबानी याद है मुझे। ये बूढ़ी आंखें भूल गई शहर के सभी रास्ते, ये बूढ़ी आंखें भूल गई शहर के सभी रास्ते, मगर तेरे घर की वो गली पुरानी याद है मुझे। ये बूढ़ी आंखें भूल गई शहर के सभी रास्ते, मगर तेरे घर की वो गली पुरानी याद है मुझे। वो जिसमें चमकता चांद देखकर धोखा हुआ था तेरे होने का। सर जरा सीनरी देखना। वो जिसमें चमकता चांद देखकर धोखा हुआ था तेरे होने का, वो 14वीं की रात वो झील का पानी याद है मुझे, वो 14वीं की रात वो झील का पानी याद है मुझे। ये झूमर, ये कंगना, ये चूड़ी, ये बिंदिया, ये झांझर मेरे शर्ट से मैच करती तेरी वो बिंदिया लगानी याद है मुझे। मेरी शर्ट से मैच करती तेरी वो बिंदिया लगानी याद है मुझे। दो इसमें सीरियस शेर ये हैं कि मुझे कोई लालच बेईमान कर नहीं सकता, मुझे कोई लालच बेईमान कर नहीं सकता, वंड छको वाली नानक की कुर्बानी याद है मुझे, वंड छको वाली नानक की कुर्बानी याद है मुझे। मुआफ करना सर कट सकता है, ये दस्तार नहीं उतर सकती, मुआफ करना सर कट सकता है, ये दस्तार नहीं उतर सकती। इसके लिए दी गुरु गोविंद की कुर्बानी याद है मुझे, इसके लिए दी गुरु गोविंद की कुर्बानी याद है मुझे। दो शेर और सुना के जाऊंगा, तो बच्चों को लग रहा है कि थोड़ा सा रोमांस और सुना दो। रोज नई बिंदिया लगाते रहे, रोज नई बिजलियां गिराते रहे, रोज नई बिंदिया लगाते रहे, रोज नई बिजलियां गिराते रहे। तुम्हें क्या आशिकों पे जो गुजरी, तुम बस जुल्फ उंगली पे घुमाते रहे। तुम्हें क्या आशिकों पे जो गुजरी, तुम बस जुल्फ उंगली पे घुमाते रहे। वो भी बेवफाई करता रहा, हम भी झूठी कस्में खाते रहे, वो भी बेवफाई करता रहा, हम भी झूठी कस्में खाते रहे। और अगला शेर थोड़ा ज्यादा खतरनाक है। हम भी झूठी कस्में खाते रहे, वो भी न भूला हमें शादी के बाद, वो भी न भूला हमें शादी के बाद, हम भी उसके घर आते जाते रहे, हम भी उसके घर आते जाते रहे। माना इस जहां को गुलजार ना कर सके, माना इस जहां को गुलजार ना कर सके, मगर कुछ कांटे रास्ते से हटाते रहे। कोई भी अपना सा होकर ना मिला, कोई भी अपना सा होकर ना मिला, सभी बस रस्मन हाथ मिलाते रहे, सभी बस रस्मन हाथ मिलाते रहे। थैंक यू।
Wow, what a wonderful ghazals you are giving, sir. We really want you to continue with the ghazals because we feel like the audience is really relating and enjoying your performance. So, sir, we request you to please carry on in the stage.
मेरे को याद करना पड़ेगा। मेरे को जो भी याद आ रहा है कि लोग तुम्हारा काम आसान किए देते हैं, लोग तुम्हारा काम आसान किए देते हैं, हम अपनी हार का ऐलान किए देते हैं। लोग तुम्हारा काम आसान किए देते हैं, हम अपनी हार का ऐलान किए देते हैं, मगर याद रखना खामोश समंदर वक्त आने पे तूफान किए देते हैं। मगर याद रखना खामोश समंदर वक्त आने पे तूफान किए देते हैं। याद करना पड़ रहा है।
Come on, guys, give a big round of applause for sir.
माना एक गजल के ये देखो, ये इनोवेटिव एक्सपेरिमेंट कर रहे हैं, गजल के साथ ढोल बजा रहे हैं एक्चुअली। माना एक गजल के कुछ शेर याद आए हैं कि माना फासला दरमियां खिंचता रहा, माना फासला दरमियां खिंचता रहा, दर्द का ही सही कुछ तो रिश्ता रहा, वक्त के साथ हर जख्म भर गया, मगर एक खून एक मुद्दत अंदर रिश्ता रहा। माना फासला दरमियां खिंचता रहा, दर्द का ही सही कुछ तो रिश्ता रहा, वक्त के साथ जख्म बाहर से भर गया, मगर खून एक मुद्दत अंदर रिश्ता रहा, वक्त के साथ हर शय बदल गई। एक शेर जो फ्रंट रो में बैठे हैं उन सभी के लिए, वक्त के साथ हर शय बदल गई, मगर मेरा दोस्त फरिश्ता था, फरिश्ता रहा। वक्त के साथ हर शय बदल गई, मगर मेरा दोस्त फरिश्ता था, फरिश्ता रहा। माना फासला दरमियां खिंचता रहा, दर्द का ही सही कुछ तो रिश्ता रहा। रेडी है या नहीं? अभी भी नहीं। अब मेरे को पर्ची निकालनी पड़ेगी। एक शेर मेरी उम्र का शेर है जरा, और बच्चों के लिए एक नसीहत भी है। ये कुहाड़ सर अच्छा शेर है, जरा सुनिएगा। हमारी उम्र का शेर है, बच्चों के लिए नसीहत है। यूं बिन बोले दास्ताने जिंदगी सुना डाली, यूं बिन बोले दास्ताने जिंदगी सुना डाली, फकीर ने खाक मुट्ठी में भरी और उड़ा डाली, उन्हीं बच्चों ने आज बचत का हिसाब पूछा, जिन पर जिंदगी भर की दौलतें लुटा डाली। उन्हीं बच्चों ने आज बचत का हिसाब पूछा, जिन पर जिंदगी भर की दौलतें लुटा डाली। अब बाकी पोएट्री सीरियस है एक्चुअली। बिछुड़ना यूं गवारा तो नहीं था, बिछुड़ना यूं गवारा तो नहीं था, सिवा इसके कोई चारा भी नहीं था। बिछुड़ना यूं गवारा तो नहीं था, सिवा इसके कोई चारा तो नहीं था। यकीनन दोष अपना भी था इसमें, यकीनन दोष अपना भी था इसमें, मगर सारे का सारा तो नहीं था। यकीनन दोष अपना भी था इसमें, मगर सारे का सारा तो नहीं था। वो उसका जाते जाते मुस्कुराना कहीं कोई इशारा तो नहीं था। वो उसका जाते जाते मुस्कुराना कहीं कोई इशारा तो नहीं था। था तेरा शहर अब भी खूबसूरत, था तेरा शहर अब भी खूबसूरत, तेरे बिन वो नजारा तो नहीं था, तेरे बिन वो नजारा तो नहीं था। पलट कर देखते कैसे उसे हम, पलटकर देखते कैसे उसे हम, उसने हमें पुकारा तो नहीं था, बिछुड़ना यूं गवारा तो नहीं था, सिवा इसके कोई चारा भी नहीं था। एक शेर इससे मेरे को और याद आ गया, हाँ जरा सुनना, अच्छा शेर है कि हाँ कल तेरे शहर में ये मेरे को नया ट्राई दे रहे हैं, हाँ कल तेरे शहर में गया था, हाँ कल तेरे भूल गया, रह गया वहां, मैं भूल गया भाई, गया था वहां। No.
Wow, what a mesmerizing ghazal. Thank you so much, sir, for that soulful and heartfelt performance. Your voice truly touched our hearts and filled this hall with peace and melody. So, after listening to sir, we also want to dedicate some line to our guest and audience. महफिल में रंग तब आता है जब मेहमान मुस्कुराते हैं, महफिल में रंग तब आता है जब मेहमान मुस्कुराते हैं, और हर पल यादगार बन जाता है जब आप जैसे लोग साथ निभाते हैं।
To continue this splendid evening.
Hello, check. Hello.
To continue this splendid evening, let us welcome the heartbeat of Dev Bhoomi Uttarakhand University, our official music band, Riyaz Se Roo Tak, from Surtar Club. From the past few years, Roo from the Surtar Club has been the very soul of our university's musical spirit, from soulful party tunes to vibrant Bollywood rhythms and the groovy pop melodies. This talented band has consistently mesmerized the audience with their versatile performances.
So ladies and gentlemen, sit back and immerse yourself in this harmonious blend of rhythm and resonance as the band Roo takes the stage to set the perfect tone for the night.
Hello.
Check. Hello, check. Hello. Hello. hello, 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 check, hello.
Good evening. We are Rouse, the official band of Dev Bhoomi Uttarakhand University by Surtal Club. I'm your friend Parshuram, and we have a very beautiful female vocalist, Pratyusha. And how can we forget about our Rajat Joshi ji? And we have on keys Tanish Bhai, Karan and Shubham on percussion, Shaurya on lead guitar. So now it's time to welcome our mentor, Rajat Hindwa sir on mic. पागल मनवा है रे ऐसे वागे रे पागल मनवा ताबड़ तोड़ नाचलू 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 हे बंदारे तोड़ नाचलू सूरज चंदा मोड़ नाचलू अब तो ताबड़ तोड़ नाचलू मैं तो बंजारा रे बंदारे तोड़ नाचलू सूरज चंदा मोड़ नाचलू अब तो ताबड़ तोड़ नाचलू मैं तो बंजारा रे बन के आवारा मैं किस मेले में पहुंच गया कोई दो रोको हो जाऊं रे मस्ती का मारा मैं क्या बोलूं मैं किसे कहूं ये दिल का मैं हारा हो जाऊं रे दिल हारा रे दिल हारा हारा दिल हारा हारा मैं यारा रे ओ यारा रे दिल हारा हारा दिल हारा हारा मैं हारा रे मैं उसके रूप का शौदाई वो डूब जाओ ज़ाहर जाए वो शौक है रंग बदलता है मैं रंग रूप का शौदाई जिनके सर हो इश्क की छाओ पाग नीचे जन्नत होगी जिनके सर हो इश्क की छाओ पाग नीचे जन्नत हो शाम रात मेरी कायनात वो यार मेरा सैया सैया चल सैया 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 दिल हारा मैं यारा मैं यारा मैं यारा दिल हारा रे दिल हारा रे दिल हारा मैं यारा मैं यारा मैं यारा दिल हारा रे दिल हारा रे दिल हारा रे दिल हारा रे दिल हारा पागल मनवा है रे पागल मनवा ताबड़ तोड़ नाच लू नाच लू नाच लू नाच लू
On the egg shaker, we have Sayangi also. So now we are moving ahead to our performance for the Uttarakhand folk, and we will start with the Garhwali songs.
ये चोरी मसरेला सुरमा है जई मेरी सुरमा सरेला सुरमा
जी गाड़ी कुमाऊं की तरफ ले चलो यार अब।
तुम्हारा पीर बड़गड़ सुम्यालन जन सरोली देखे बोल बेटी के किम साड़ी हाट माइक दंड्या महल पूर्व छजाले छन मेरे वे अपना राज ले जोलू रे धिन धिन ताक तानी ताक धिन धिन ताक तानी ताक चल मेरी सरोली किम साड़ी हाट मां किम साड़ी हाट मां किम साड़ी हाट मां चल मेरी सरोली किम साड़ी हाट मां किम साड़ी हाट मां किम साड़ी हाट मां राजेण बनी रैली ठकुराण बनी रैली किम साड़ी हाट मां किम साड़ी हाट मां राजेण बनी रैली ठकुराण बनी रैली किम साड़ी हाट मां किम साड़ी हाट मां राजो कर रजवाड़ा राणियों का रानी वासा किम साड़ी हाट मां किम साड़ी हाट मां राजो का रजवाड़ा राणियों का रानी वासा किम साड़ी हाट मां किम साड़ी हाट मां चल मेरी सरोली किम साड़ी हाट मां किम साड़ी हाट मां किम साड़ी हाट मां चल मेरी सरोली किम साड़ी हाट मां किम साड़ी हाट मां किम साड़ी हाट मां धर्म देवी को तना दूध लेना वायो टिलू दूध लेना वायो धर्म देवी को तना दूध लेना वायो टिलू दूध लेना वायो धर्म देवी को तना दूध लेना वायो टिलू दूध लेना वायो हे धर्म देवी को तना दूध लेना वायो टिलू दूध लेना वायो तेरो जुटी में निकाची है चम माया ले कयालो टिलो टूटी जाने गुना हे माया ले कयालो टिलो टूटी जाने गुना हे लगा तेरी लंबी लटी दसरा का बुना आओ कहीं बजूनी है जे टूटी जाने गुना लगा तेरी लंबी लटी दसरा का बुना आओ कहीं बजूनी है जे दसरा का बुना लगा तेरी लंबी लटी टसरा का पुना आओ गई बजोनी है जे टूट जाएगी ना बेटी लगा तेरी लंबी लटी टसरा का पुना आओ गई बजोनी है जे टूट जाएगी गुना बेटी लगा तेरी लंबी लटी टसरा का पुना आओ गई बजोनी है जे टूट जाएगी गुना बेटी लगा तेरी कहीं बजूनी है जे टूटी जानी गुना बेटी लगा तेरी लंबी लटी दसरा का पुना आओ कहीं बजूनी है जे टूटी जानी गुना.
Thank you so much.
Thank you.
So much. Thank you so much. हेलो. जो दिल से निकले वो अल्फाज बन जाते हैं, जो महसूस हो जाए वो अंदाज बन जाते हैं. आपकी तालियों से मिलती है हर कलाकार को रोशनी, ये जज्बे महफिल की आवाज बन जाते हैं. सो प्लीज अगेन गिव अ बिग राउंड ऑफ अप्लॉज फॉर द ऑफिशियल क्लब ऑफ सुरताल रू. Next, we bring to you an invocation to the divine, Bharatnatyam, one of the oldest and most revered classical dance forms of India. So please welcome them on the stage.
This performance, titled Pranavalaya and Chandrachud, represent devotion, discipline, and the internal grace of Lord Shiva. The intricate movement, expressive gestures, and rhythmic precision together narrates the story of cosmic harmony. So please welcome our talented artists as they transport us to the artistic and spiritual realm through their mesmerizing art of Bharatnatyam.
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि
चन्द्रचूड शिवशङ्कर पार्वती रमणाय निनगे नमो नमो चन्द्रचूड शिवशङ्कर पार्वती रमणा निनगे नमो नमो सुन्दरतर विरागधर सुन्दरतर विराग धर गंगाधर गज चर्मांबरधर गंगाधर गज चर्मांबरधर चंद्रचूड शिवशंकर पार्वती रमणा निनगे नमो
प्रणवालय पाहि परिपालय परमेशी कमलालय श्रीदेवी कुरिपिंचवे करुणांबुरासी धीम तारा जतुलतो प्राणमे नाट्यं चेसे गतुलतो नामशतं मूल नतुलतो नापैननी चूपु आपेला शरणंटिने जननी नाद विनोदिनी भुवन पालिनी वे अनाथ रक्षणनी विधि कादटे मोरविनी चेरवटे నా ఆలోచనే నిరంతరం నీకు నివాళినివ్వాలని నాలో ఆవేదనే నువ్వు ఆదరించేలా నివేదనవ్వాలని
Performance, the perfect blend of rhythm, expression and tradition left us all spellbound.
Now we move to the vibrant confluence of rich tradition, the culture hues of the festive rhythm of Gujarat.
This fusion performance captures the essence of joy, festivity and cultural unity. From the graceful exuberant energy of Garba, this presentation beautifully blends the cultural rhythms into harmonious dance. So get ready to witness a mesmerizing amalgamation of color, culture and coordination as our performers light up the stage.
કારણ કે અને ઉતરવું પડે કારણ કે કિનારે તો ખાલી પડે નાની નાની પગલી ને નાના એવા સપનાની રેત વાળી ડગલી ને તોફાનો તરાપ મારે હાલે સાવ આપી જાય તોય જેની હિંમત અને આમ નહીં આપે એવો ખારવો ખલાસી એવો હાડનો પ્રવાસી એવો ખારવો ખલાસી એવો હાડનો પ્રવાસી ગોતી લો ગોતી લો ગોતી લો ગોતી લો ગોતી લો ગોતી લો
પોતાના જ દરિયામાં પોતાની જ ડૂબકીથી જાતનું અમૂલ મોતી લો
મોતી જે મજામાં ખાલી વાવટા ધજામાં એવો હાડનો પ્રવાસી ગોતી લો
એવો કોણ છે ખલાસી મને ઓકે ભૈયા કોણ છે પ્રવાસી મને હાય 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 કોણ છે ખલાસી મને લઈ જા નૈયા તમે ગોતી લો એવો કોણ છે ખલાસી મને ઓકે ભૈયા કોણ છે પ્રવાસી મને હાય 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 કોણ છે ખલાસી મને લઈ જા નૈયા તમે ગોતી લો
લોબડી આળિયું ભેળિયું મળિયું મડડે રાસ રમે ઓટી કાળી કમળિયું લાલ ધાબળિયું ફૂલ છાબડિયું શિર પરે હૈયે હે મહાસણિયું માણેક મટિયું હે મહાસણિયું માણેક મટિયું મોતીએ ચડિયું તેજ જરે નવ લાખાય લોબડી આળિયું ભેળિયું મળિયું મડડે રાસ રમે માં માડી મળ્યું મઠડે રાસ રમે માડી મળ્યું મોણીએ રાસ રમે
And now it's time to raise the energy and no chair with one of the most exuberant dance form of India, that is Bhangra.
Originally from the fertile land of Punjab, Bhangra is celebration of life, harvested and happiness with every beat of dhol and every step of enthusiasm. This performance is sure to make sure your heart dance along. So dear audience, let's the infused energy of Bhangra fill the air and bring alive the spirit of festive in this culture evening.
पंजाब है पंजाब चिंता मरवा देंगे पैरों तले आग मचा देंगे आग सिद्धू मूसेवाला दीप जानो
जिवें गिनती लेके फिरे गेड़ी मारदा विच अमडीर फिरे मुच्छा चाड़दा जानदी मैं भी तेरे चित्त विच की केड़ी गल्लों फिरदा तू मैनू ताड़दा चोपरा दे दिल कर दिंदी फाड़ी है ओ लेज़र निगाह मेरी जिथे ठहरदी चोपरा सिरे दी रकान में टोरंटो शहर दी ओ अगलाइया चारा दूनी आंदा आंदा नप्पी दी चोपरा नू ना यार ते आंदा पुलिस बाहर दी थक्की दी तेरी फोटो डैशबोर्ड ते तू मेरे लक्की दी ना उनका बाकी जटनी गेट खोल के रखी दी ओ गड्डी विच बालिया बचोला है नहीं होगी गल पक्की कत्ते दे महीने डेट रख लिया 25 बचोला है नहीं होगी गल पक्की कत्ते दे महीने डेट रख लिया 25 नवे फिर दे खरीद दे आ चादरे फिर दे खरीद दे आ चादरे ते मुच्छा कुंडियां करोंदा फिर लाना नी चढ़ दे सियाल तेनू मिठिए मित्रा ब्याह के ले जाना नी चढ़ दे सियाल तेनू छिंदिए जट ने ब्याह के ले जाना नी सस तेरी करे अरदासा तड़के बड़वाई टूमा तेरे करके फोटो तेरी कादी में दिखाई बेबे नूनियों ने लड्डू वंड दे
मिठे दे शकीन गब्बरू है तैनू रख के तली ते जट लैणगे नी होई ना पंजाब वल नू नी तैनू नत्तियां दिखा के पट लैणगे नी होई ना पंजाब वल नू है तैनू नत्तियां दिखा के पट लैणगे नी होई ना पंजाब वल नू तैनू नत्तियां दिखा के पट लैणगे
Dear audience, please cheer up such amazing and energetic performers of Dev Bhoomi Uttarakhand University.
As we move forward to the final dance of this evening, we take the immense pride in presenting the soul of our land, the Kumaoni and Garhwali region dance of Uttarakhand.
This performance is not just a dance, it's a portrayal of our culture, our traditions and our connection to the majestic Himalayas. With each movement, it narrates tales of harmony, simplicity and deep-rooted pride in our heritage. Let's embrace the rhythm of our mountains and celebrate the spirit of Dev Bhoomi through this captivating regional dance performance. Please give a big round of applause for our regional dancers.
ताम पीटो पीतल पीटो बिजुले झम हुआ कान में रूमा बिजुले झम
नंदा देवी म्याला डोला बिजुली झम भादो का मैना मेरो मन बिजुली झम
सरसराती हवा ठंडो पानी पक गए बेड़ू का फल की तानी देवताओं का निवास है उत्तराखंड प्यार सत्कार और संस्कार है उत्तराखंड ढोल दमों की थाप मसक बीन की छाप है उत्तराखंड होली न्योली नाखे की नथुली रंगीली पिछौड़ी उत्तराखंड पैजाबी छमा छम हुड़का घमा खम ऐसा हमारा उत्तराखंड ऐसा हमारा उत्तराखंड
रामदाई का होटला 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 रामदाई का होटला नैनीतालई मा रामदाई का होटला नैनीतालई मा हेरी हेरी फस्यूं त्यारा माया जालई मा कांची हेरी हेरी फस्यूं त्यारा माया जालई मा
ढल की टोपी काला सूट झुंगा दड़ी माँ ढल की टोपी काला सूट झुंगा दड़ी माँ हेरी हेरी फस्यूं त्यारा माया जालई माँ कांछा हेरी हेरी फस्यूं त्यारा माया जालई माँ
टुकली सजी गे आंखों चश्मा लगी गे ये मुंड में टुकली सजी गे आंखों चश्मा लगी गे चिपल चकते रुगात अन कमाल करी गे मुंड में टुकली सजी गे आंखों चश्मा लगी गे चिपल चकते रुगात अन कमाल करी गे तेरे सजीलो सिंगार ठिका देना जोहार तेरे सजीलो सिंगार ठिका देना जोहार ते महाजिया मन लगी मेरा पहाड़ के बांध मेरा पहाड़ के बांध प्रेमा माया लगी गे मेरा पहाड़ के बांध प्रेमा माया लगी गे
साड़े माँ मेरे दिपा समधने डबल गुलाब साड़े माँ मेरे दिपा समधने हाँ दन बड़ो दिपा कोरी समधने आहा क्या बली रसायन समधने अरे चम चम गणे अरे दम दम गणे हाँ चम चम गणे बल दम दम गणे अरे गुलाब साड़े माँ मेरे दिपा समधने डबल गुलाब साड़े माँ मेरे दिपा समधने
मारे तको कोड़े धोपा मारे तको कोड़े जम कबड्डी खेलिए मैं ते धोपा तेरे प्यारे मोह कोड़े जम कबड्डी खेलिए मैं ते धोपा तेरे प्यारे मोह कोड़े जम धोपा तेरी धोपा तेरी धोपा तेरी धोपा तेरी धोपा तेरी याद माँ जामनी की धारे माँ खिड़बिड़ खट वे गई मेरा दिल का काक माँ धोपा तेरी याद माँ जामनी की धारे माँ खिड़बिड़ खट वे गई मेरा दिल का काक माँ धोपा तेरी याद माँ जामनी की धारे माँ खिड़बिड़ खट वे गई मेरा दिल का काक माँ धोपा तेरी याद माँ जामनी की धारे
Hello. Finally, a performance that truly reflected the spirit and energy of our Dev Bhoomi Uttarakhand. So coming towards the ending segment, what a spectacular evening it has been. From classical devotion to cultural fusion, from the melodies of Surtal to the rhythms of our hills, tonight truly reflected the diversity and unity that defines our nation.
Indeed, Gunjan. This evening stands a testament of the fact the art and the culture speaks a universal language, one that transcends borders, unites heart, and celebrates humanity. On behalf of the Association of Microbiologists of India and Dev Bhoomi Uttarakhand University, we extend our heartfelt thanks to all the performers, faculty, organizing committee, and a wonderful audience of being a part of this vibrant celebration. As the curtain draws on the memorable night, let us carry us with the melodies, colors, and the warmth of this cultural evening has gifted us. Thank you, ladies and gentlemen, and a wonderful evening ahead.
Now, I would like to call Ms. Nirjala ma'am to please take over the stage.
Thank you so much. What a beautiful evening. With this, we come to an end to this very enriching and very beautiful day 2 of the 66th AMI Annual International Conference. I invite you all for dinner. I request all the dignitaries and the speakers to kindly move towards the HM restaurant for dinner. The student participants are requested to move towards the food pandal outside hotel management, and the faculty participants are requested to move towards the pandal outside the Ayurveda. Also, there's a small announcement. The buses for the hotel Golden Leaf as well as F Hotel and V1 will be parked outside the food pandal at 8:30 p.m. Also, the buses for the Nanda Devi Guest House will be parked near the parking area which is outside the main pandal. So I request you all to kindly take your seats in the bus on time. Thank you so much. See you all tomorrow at 10 a.m. sharp in the main pandal for day 3 of the 66th AMI Annual International Conference. Thank you everyone.